Pues si quieres, lo podemos hacer en orden y vamos saliendo.
Vale.
¿Por orden o no?
So, good morning. My name is Raquel Sánchez Valle. I'm a consultant, senior consultant of neurology at the Hospital Clinic of Barcelona and associate professor at the University of Barcelona. And I have the honor to co-chair this conference with my colleague, Juan Fortea, from the Hospital de Sant Pau. It's a true privilege to welcome you all to the second edition of this genetically determined Alzheimer's disease conference, where we aim to bring together the goals of Down syndrome-associated Alzheimer's disease and autosomal dominant Alzheimer's disease. In 2024, this conference brought together more than 150 participants from 20 countries. Today, we want to continue that mission. Our goal is not only to share knowledge and connect people, but also to generate a meaningful impact in these diseases. Over the next 2 days, we will hear from experts in basic science, clinical research, neuropsychology, and biomarker researchers. We also engage in conversation around ethics, accessibility, and the lived experience of individuals and families affected by these conditions. This conference will not be possible without the support of many dedicated individuals and organizations. I would like to begin by thanking the steering committee and the scientific committee for designing the program and reviewing the submissions. We are also deeply grateful for the support of the Alzheimer's Association, the Have Alzheimer's Barcelona, and the Foundation Jerome Lejeune. Thank you for your critical contributions. Thank you as well for our sponsor, Biogen, Ionis, Lilly, AC Immune, Biohaven, and Milan, the National Down Syndrome Society, and Roche. And finally, thank you all for being here. I hope you will enjoy these 2 days. And now it's my pleasure to give the floor to Alberto Gio, Head of Neurology of Sant Pau.
Well, good morning, everyone. I'm Dr. Alberto Yeo. I'm a neurologist. I'm the chair of the neurology department here at Hospital Sant Pau, and it's really a pleasure to have you here again for the second time for this exciting meeting. I just have a few words about the facility, the wonderful place that we are here. As you see, this is a great hospital. I did my residency here when this was a hospital with patients. Right now, there is no patient activity anymore, but it's really a wonderful place and very special place for us to have you here. This hospital it was declared by the part of the World Heritage by the UNESCO in 1997, and right now here it's really great because the neurology of Spain really started here in this hospital. Not right in this hospital, but in the old hospital that is in the old city that you can visit if you have time. It's really a great place to see. Well, I hope that you enjoy the meeting. If you have time, I would really recommend to really walk around and to see the different pavilions, because it really marks a part of the important history of neurology of Barcelona and our country. So thank you. I hope you enjoy the meeting, and we'll pass the word to Heather Schneider from the Alzheimer's Association. Thank you.
Well, good morning, everyone, and what an amazing gathering in such a beautiful setting in a beautiful place, and incredible congratulations to our organizers and our hosts that are having us here for bringing us all together. I'm really excited about all of the discussions that we'll have, and I think as you see from the agenda, and we heard a little bit about, we'll go from basic science, translation, and all aspects of the research in between, and those that are participating and have the lived experience as well in thinking about the research and bringing those voices and those perspectives together, because this is an amazing time to be in our field and doing the work that we're doing. We have new tools that we can use for our research, for diagnosis, for understanding the disease, understanding the diseases and their contributions, their intersection with one another, the clinical trials pipeline. We continue to see that, and we have an entire session focused and thinking about that as we go forward. And this together is a tremendous community, whether you're in the Down syndrome, Alzheimer's disease and Down syndrome community, or the ADAD, the dominantly inherited Alzheimer's disease community, or here together as we come together as a larger community, as a larger research community together. And community means a lot of things to a lot of people. It's a sense of belonging, it's a sense of sharing, it's a comfort that we have together in sharing our research and engaging in these conversations. And as a community, we will face some challenges. We are facing some challenges right now, and we may continue to face some of those challenges. But as we come together, like in meetings like this, like in discussions like this around the world, we're really keeping that central focus together. Not only will we survive, we will thrive. And we will continue to do the important work that those that are depending on us to move our research forward expect and are counting on us to do. So we have that opportunity over these next 2 days to share with each other, to talk to each other, to engage in how to take our research and our work forward in the global community and in the work that we're doing, really in this intersection of Alzheimer's disease in individuals with Down syndrome, dominantly inherited Alzheimer's, as we then think about that impact that we have for the entire community that we serve. So thank you to all of you for everything that you do as we come together in these conversations as part of this community. And an incredible thank you to all of my fellow panelists here in bringing this conversation together. So looking forward to the 2 days. Thank you.
Hello, good morning everyone. My name is Carla Obrados Tarragó, and I'm the Alliance Manager of the Hub Alzheimer Barcelona. It's a real pleasure for me to welcome you to the second edition of the Genetically Determined Alzheimer's Disease Conference, and also to briefly introduce an initiative that connects many of the institutions represented here today. The Hub Alzheimer Barcelona is a pioneering collaborative platform promoted by the Pasqual Maragall Foundation with the support of the Barcelona City Council. It brings together seven leading institutions in dementia research and clinical care across this city, which are ASE Alzheimer Center Barcelona, the Barcelona Beta Brain Research Center, Hospital Clínic de Barcelona, Hospital de Sant Pau, Hospital del Mar, Hospital Universitari Vall d'Hebron, and the Pasqual Maragall Foundation itself. Our mission is to catalyze scientific collaboration, accelerate the impact of research, and position Barcelona as a global reference in Alzheimer's innovation. We do this by promoting joint initiatives, supporting emerging talent, and building strong bridges between academia, healthcare, industry, and society. We are especially proud to support this conference, which actually reflects our core values: scientific excellence, interdisciplinary collaboration, and a shared commitment to improving the lives of those affected by Alzheimer's. Thank you very much for being here. I hope you enjoyed the conference, and I warmly invite you to learn more about the Hub Alzheimer Barcelona as we work together to strengthen the dementia research ecosystem. I wish you a productive and enriching experience in this amazing environment.
So, welcome all. My name is Juan Fortea. I'm the Memory Unit Director of the Hospital of Sant Pau. And I cannot overstate how happy I am to host the conference for the second time here with all of you. I think I want to extend, of course, our gratitude to the Alzheimer's Association, to the Barcelona Hub, to all our sponsors, not only for supporting the conference, but also because we are in a new time for our patients. We are envisioning for the first time the possibility of preventing this disease in Down syndrome and autosomal dominant AD. In fact, we can lead the way of preventing Alzheimer's disease, and we have to take this opportunity. This conference is about cross-fertilization and talking between us. So this year, in the design of this conference, not only do we want you all in the gala dinner talking to each other, we want to have a lot of coffee breaks and communications. We've hired 2 rooms in the upper floor where you can have tables if you need to do a meeting or a Zoom conference. And really, we want you to interact and enjoy the conference these 2 days. And with that, I won't extend anymore. I just want to do some housekeeping so that we are, this year, it's going to be 200 registries. I think in person, we're expecting about 180 persons. So it's going to be a full room. Not only do we have it in the floor, for those of you, there is another, you take the stairs back on the right, on my right, there is stairs to another part of there are extra seats up there. So at any point, you can also take it there. And just the structure of the sessions, we have several scientific sessions. Please, all the speakers go, and you need to be to get the mics, because the idea is to have all the talks one after another, to have joint discussions. And we really want to do that because last year the experience was really good for the discussions that we had, and we really want the interaction with you. There will be people with the mics. We really want to have the kind of interaction we had last year. So as I say, get every speaker should be get the mics, and we'll have a joint discussion at the end. As for the coffee breaks, stay tuned. The idea was to have the coffee breaks in the garden, but the weather is a bit unstable, so there might be changes. So before rushing into it, wait for instructions, because lunch was in -1. So we had extra communications this year. So last year we didn't have it, but we have poster sessions, and they will be at lunchtime. And lunchtime will be served in the Sala Ipostila, which is 1 level below. So when you go out the door, you go downstairs. That's where the poster sessions and lunch will be. As I say, coffees might change from that room or outside, depending on the weather. So before the breaks, we'll try to provide some information. And with that, without further ado, I think that we welcome you all, and we next to the next session. Okay, so we're moving to the Lifetime Achievement Awards. It's my great pleasure to move to this special moment in our program. This year we've decided to recognize not 1, but 2 persons, one on the field of autosomal dominant Alzheimer's disease and another in Down syndrome. Their lifelong dedication has significantly shaped our understanding of these conditions and has inspired many researchers and clinicians. The recipients were nominated by the steering committee, and we had a vote amongst ourselves, and after that we made the decision. Unfortunately, the awardees could not be here to receive the awards in person for different reasons, but we will proceed to honor them with the recognition that they deeply deserve. And we will begin with the Lifetime Award in Autosomal Dominant AD that will be presented by Raquel Sánchez del Valle.
Hi again. With deep admiration and gratitude, we present the Lifetime Achievement Award to a true pioneer in the field of autosomal dominant Alzheimer's disease, Dr. Francisco Lopera. Dr. Lopera, who sadly passed away in September 2024, dedicated more than 4 decades to the studying and providing better care to Colombian families affected by autosomal dominant Alzheimer's disease. His life's work began in the Paisa region, a region in Colombia, where as a young physician he first encountered families suffering from what at that time was an unknown form of dementia. It was then when the presenilin-1 E280A mutation, also known as the mutation Paisa, was identified. For decades, Dr. Lopera and his team at the University of Antioquia studied these families, often facing immense challenges. His research made him become one of the world's leading figures in the field. In recent years, he built numerous international collaborations to bring cutting-edge technologies and clinical trials to this unique population in Colombia. Today, we want to honor not only a brilliant neurologist and researcher, but a man of unlimited dedication to his patients. Dr. Lopera's legacy lives through his research group, his collaborators, and also on this conference. Please join me in celebrating the life and legacy of Dr. Francisco Lopera, recipient of this conference 2025 Lifetime Achievement Awards. We have a video from Dr. Lopera to celebrate his achievements and life. Please, a big applause to Dr. Lopera. As he cannot be here, we will send the award to his family afterwards.
It is now my honor to present this year's Lifetime Achievement Award in Down syndrome-associated Alzheimer's disease. This award goes to a scientist, a physician, educator, and lifelong advocate whose work has transformed the field of Down syndrome across the Spanish-speaking world and far beyond. A person whose unique combination of scientific excellence and human commitment is a true inspiration. Professor Jesus Flores is a doctor of medicine and surgery from the University of Navarra and holds a PhD in pharmacology from Dartmouth University in the United States. Until his retirement in 2007, he served as the chair of pharmacology at the University of Cantabria, where he left a profound legacy in research and education. In 1976, following the birth of his daughter with Down syndrome, he expanded his professional focus to include lifelong personal and scientific commitment to this field. In 1982, he co-founded the Fundación Síndrome de Down de Cantabria, and in 1984, he launched the first Spanish-language journal on Down syndrome, which he directed for nearly 4 decades. In 1995, he initiated one of Spain's earliest research lines on animal models of Down syndrome, supervising multiple PhD theses and laying the groundwork for ongoing scientific progress. As president of the Fundación Iberoamericana Down 21, he has spearheaded vital resources for families and professionals, including educational websites, encyclopedias, and scientific journals. Professor Flores has given hundreds of lectures across Spain, Latin America, and the US, Canada, and Europe. He has published over 400 articles and authored numerous landmark books, covering topics such as neurobiology, mental health, adult life, pharmacotherapy, and regression in Down syndrome. His impact has been recognized with a long list of honors, including the Exceptional Meritorious Service Award from the US National Down Syndrome Congress, the Career Achievement Award from the Spanish Society of Pharmacology, and the Madame Lejeune Prize shared with his wife for a lifetime dedicated to caring for the most vulnerable. And now, if I may, I'll add a personal note. Jesus Flores was my teacher at the University he was a professor in pharmacology, the best I had. when I was studying pharmacology, I did not know of all these achievements. Oftentimes, when you're a student, you're missing the full picture. And when I entered the research in Down syndrome, I recognized the immense legacy that Dr. Flores really had in the field of Down syndrome in Spain and Latin America. He really has been a pioneer in shaping and putting the foundations that we are now benefiting from, all of us, to help people with Down syndrome and their families. And although he could not be with us today, it's my immense pride and gratitude. It's with immense pride and gratitude that we present the DSAD Lifetime Award to Jesus Flores. So please join me in an applause to him.
Good morning, dear colleagues and friends. I want to express my heartfelt gratitude to the organizers of this second international conference for selecting me to receive the Lifetime Achievement Award, ADAD-DSAD. But above all, I'm deeply grateful for the research efforts of all those who participate in this conference, as they share the results of their studies with the rest of us. There is no doubt they are contributing productively to enrich our knowledge about persons with Down syndrome in such a special stage in their lives. The more we know them, the better we will serve them. Many thanks to all.
We will move to the first oral session, Basic Science. The moderators will be Heather Snyder and Marie-Claude Portier. Please come to the floor.
Hello, everybody. So it's a great honor to chair with Heather Snyder from the Alzheimer's Association the first session called Basic Science. And we will have 3 talks of 15 minutes, and then we will have the Q&A session. And I will introduce the 2 first speakers. So the first speaker is Lizzie Flores Aguilar. Everybody knows her. She already gave a talk last year at this meeting. And she is a postdoctoral research fellow in the lab of Liz Head at UC Irvine. And she will today talk about vascular remodeling and blood-brain barrier breakdown in individuals with Down syndrome and Alzheimer's disease. Please be seated.
Hola, buenos días. Good morning, everyone. I'm very jet lagged, but also very happy to be here and see familiar faces, old friends, and colleagues. So these are some shots from last year's conference. We had a blast. We had amazing science, great company, and I am hoping we can top that with science, but also socially, as Dr. Fortea was mentioning. On a very personal note, I'm also very happy because I met my nephew. He's all brand new, and he hasn't published any papers yet, but he mastered the art of loud peer reviewing at 3 a.m. in the morning. So, very cute guy. And I'm thrilled to share with you all the work that I've been doing in the HEAD lab. And we are a neuropathology lab. We study post-mortem brain tissue, and we do a lot of histological analysis. And my project relates to understanding vascular pathology in Down syndrome. So we all know that people with Down syndrome display Alzheimer's disease neuropathological changes throughout their lifespan Starting very early in life, they have diffuse amyloid plaques and intraneuronal amyloid beta, and that progresses to a full-blown AD neuropathology by age 40, in which we can observe mature amyloid plaques and neurofibrillary tangles and neuritic plaques. Cognitive dysfunction usually appears in their early 50s. People with Down syndrome are a very special population because they have a low frequency of some common systemic vascular risk factors, such as atherosclerosis and arteriolosclerosis, compared to the general population, but also to those with Alzheimer's disease. And we also know that people with Down syndrome have increased vascular injury markers, such as white matter hyperintensities, enlarged perivascular spaces, and microbleeds. And these are some of the papers that ABCDS and also the San Pau Memory Clinic have published. And I think everyone is here in the audience. People with Down syndrome also have severe cerebral amyloid angiopathy, which is a deposition of amyloid beta in the cortical and leptomeningeal vessels. And also they have more CAA than the neurotypical population and also the people with late onset Alzheimer's disease. We are about to submit this paper in which we study the frequency of mixed neuropathologies in Down syndrome in people with Down syndrome with and without dementia, and we found that CAA is the most common copathology in Down syndrome brains. So almost 90% of the brains that we analyzed have cerebral amyloid angiopathy, and only 10% have pure Alzheimer's disease, meaning tangles and amyloid plaques. People with Down syndrome also, cerebral amyloid angiopathy is more frequent in the occipital cortex than in the frontal cortex, and also they have increased microbleeds, which are more severe in the occipital cortex than in the frontal cortex. Microbleeds usually appear first in the occipital cortex and then in the frontal cortex. So the occipital cortex is a very hot spot to study vascular pathology in Down syndrome. We also know that people with Down syndrome have differential neuroinflammatory processes going on throughout their lifespan, and this could also contribute to this vascular pathology that we observe in this population. So as a summary, we know that people with Down syndrome have a low frequency of common systemic vascular risk factors. They have severe CAA, increased microbleeds, increased vascular injury markers, and brain inflammation. So we ask a very simple question, which is, how is the cerebrovascular affected in Down syndrome? So the main objective of this project is to characterize blood-brain barrier pathology, the integrity of the blood-brain barrier, the components of the blood-brain barrier, and blood vessel morphology in individuals with Down syndrome throughout their lifespan. And just as a reminder, the blood-brain barrier is composed by a monolayer of endothelial cells, a basement membrane, and pericytes. And what we want to do when assessing vessel morphology is to look at the vessel area fraction, the diameter of the vessels, the length of the vessels, the tortuosity of the vessels. So what we did was to start by examining the occipital cortex. We did immunohistochemistry to look at the blood-brain barrier components. We did the imaging, and we analyzed using Fiji and Halo. So first we look at fibrin. So fibrin is a component of the coagulation cascade and is normally found in the blood. It doesn't cross the blood-brain barrier. However, in many neurological disorders, such as Alzheimer's disease or multiple sclerosis, the disruption of the blood-brain barrier allows for fibrinogen to enter the brain. So fibrinogen, when it enters the brain, it can also promote neuroinflammation. So what we did was to analyze fibrin. So we did an immunostaining against fibrin. This is the brown staining that you can see here. And we analyze fibrin deposition in the gray matter and also in the white matter. We analyze 3 groups. So we have young controls without Down syndrome, young people with Down syndrome. The mean age of these groups is around 25 years old. We also have adults with Down syndrome with a mean age of 55 years old and neurotypical controls. And we also included a group of late onset Alzheimer's disease and their age-matched controls. So this is what we found. These are young controls. On the top panel, we see 3 brain sections, and then young people with Down syndrome. So these are around 25 years old. And I don't know if you can already see that we see an increase in fibrin deposition in the brain parenchyma. And this is just a close-up, and we see fibrin within the vessel walls, but also diffusing into the brain parenchyma. So we quantify this in the gray and the white matter, and we observed an increase in Down syndrome. And we also noticed when we were imaging that the gray matter was more affected than the white matter. So we quantified that, and we found that both in young controls and also in young individuals with Down syndrome, the gray matter is more affected than the white matter. The second group is the adults with Down syndrome with advanced Alzheimer's disease neuropathological change. And this is what we observe. So we see a severe extravasation of fibrin into the brain parenchyma in the gray matter, but also in the white matter. And again, we see that the gray matter is more affected in controls, but also in Down syndrome with Alzheimer's disease. And finally, we have all controls and people with Alzheimer's disease. And to our surprise, and contrary to what has been reported, we did not find any differences in fibrin deposition in the gray matter and in the white matter. But however, we did see that in both, in all controls and in Alzheimer's disease, there is an increase. Fibrin is more deposited in the gray matter than in the white matter. And as you were probably expecting, we also did not see any differences between those with Down syndrome and Alzheimer's disease neuropathology and those with Alzheimer's disease. We did see differences between those that were at advanced stages of Alzheimer's disease in Down syndrome compared to those with very incipient AD neuropathology in the gray matter, but not in the white matter. And finally, this is some data that Ellie will show. So we did some correlations between fibrin deposition and amyloid beta deposition, and Ellie will show her beautiful data, but I just want to show you that we found a positive association between amyloid load in the brain and fibrin deposition in Down syndrome with Alzheimer's disease, but also in aged controls. We also ran the same correlation analysis with tau, and we only found that people with Down syndrome and Alzheimer's disease have that the degree of tau deposition correlates with fibrin deposition in the brain. So then we're going to switch gears, and I'm going to show you the results that we have regarding vascular morphology in Down syndrome. And I couldn't have done this alone. I had the help of Jessie Pascual, Halima, and Annie. So I'm very thankful for their contributions. And what we did was to analyze vessel length, the area occupied by the vessels, the vessel tortuosity, string vessels, basement membrane area, and pericyte number. And we analyzed this in a very small group of controls, adult controls, and people with Down syndrome at advanced stages of Alzheimer's disease neuropathology. And what we found is that people with Down syndrome and Alzheimer's disease neuropathological change have an increase in vessel density compared to age-matched controls. We also found that the length of the vessel is greater in Down syndrome than in controls. However, we did not find any changes in the tortuosity of the vessels, meaning that these vessels are kind of healthy. We also look at the basement membrane, and we analyze the area covered by the basement membrane by looking at collagen 4, and we found an increase in people with Down syndrome and Alzheimer's disease. We also counted the number of pericytes, which are these bumpy cells sitting on top of endothelial cells, and we found an increase in Down syndrome compared to controls. However, when we normalize the number of pericytes to the area covered by collagen or to the number of vessels, we did not find any differences, meaning that the pericyte to vessel ratio is maintained in Down syndrome. And then when I was analyzing my slides under the microscope, I observed the occurrence of these very thin strands close to the vessels or forming part of the vessels. And these have been regarded as string vessels, collapsed capillaries, pericyte bridges, and they are called also pericyte bridges because we can see pericytes sitting up top of these string vessels, kind of like they are being trapped here. So we counted the number of string vessels in Down syndrome, and we found an increase of these structures in the Down syndrome brain. So we wanted to know the phenotype of these vessels, and we did a triple staining. So we stained against collagen 4, which is a component of the basement membrane. And here we can see a string vessel. So they are way thinner than a normal vessel. And you can see another strand here and here. So then we did a staining against lectin, which will stain endothelial cells. And you can see that we don't see these structures that are lectin positive, meaning that they are mainly composed by basement membrane proteins. We also look at PDGFR-beta, which is a pericyte marker, and we found that pericytes are indeed sitting on top of these basement membrane structures. So now, the results I show you on vessel morphology were done by looking at lamina 3 and 4 of the occipital cortex. So we are moving on, and we are using a digital pathology software to analyze the same measures, but in whole brain sections. So we will have data on all the cortical layers, and in the gray and the white matter. So we are currently working on that. So the take-home message is that the brains of people with Down syndrome show a vascular remodeling process, and also the fact that we see fibrin deposition in the brain parenchyma in young individuals with Down syndrome and also in adult individuals with Down syndrome highly suggests that the blood-brain barrier is compromised in this population. And finally, I would like to acknowledge my amazing PI and mentor, Dr. Liz Head, all the people that helped me in the HEAD lab, all the members of the HEAD lab, the repository team, our collaborators, and also, last but not least, all the tissue donors and their families. Thank you.
So thanks a lot, Lizzie. A lot of results since last year. Congratulations. So now we'll have the second talk of this session from Johan Gronveld. Johan is a MD and PhD student at the Alzheimer's Center at UMC, Amsterdam. And she will talk about genetic modifiers of age of onset of Alzheimer's disease in APP duplication and Down syndrome.
Thank you for the kind introduction. I am Joan from the Amsterdam UMC in the Netherlands, and I want to thank the organizers for letting me show you the results of my project on genetic risk factors for sporadic Alzheimer's disease, influence age at onset of dementia in carriers of an extra copy of APP with and without Down syndrome. First of all, I have nothing to disclose. The amyloid precursor protein gene, APP in short, located on chromosome 21, is responsible for amyloid production. A triplication of this gene causes overexpression, which leads to abnormal amyloid beta aggregation and eventually Alzheimer's disease. Having 3 copies of this gene happens in people that have a duplication of the gene called APP duplication, which is seen in cases of autosomal dominant Alzheimer's disease, but also happens when people have an extra copy of the entire chromosome 21, trisomy 21, or Down syndrome. And in both these groups, the age at onset of dementia varies widely. On the left, you see a figure of a Kaplan-Meier curve of APP duplication carriers, in which you can see that the typical age at onset of these carriers varies between the 40 and the 60 years of age. On the right side, you see the age-specific prevalence of dementia in Down syndrome patients, in which you can also see that the age at onset starts around 40 years of age as well. And this variability in age at onset makes it really difficult to predict age at onset in asymptomatic carriers. For sporadic AD, already many genetic risk factors are known. We know that the biggest risk factor for Alzheimer's disease is the ApoE4 allele. And genome-wide association studies have identified a lot of common variants that are associated with AD risk. And the most recent published GWAS by Belenges from 2022 identified 85 common variants associated with AD. And by aggregating these common variants, you can calculate a polygenic risk score, which represents the susceptibility somebody has to develop Alzheimer's disease. And this polygenic risk in sporadic AD is known to also influence the age at onset, in which a low genetic polygenic risk is associated with later age at onset of dementia, and the high polygenic risk is associated with earlier onset of Alzheimer's disease. And since the variability in people with an extra copy of APP is so high, we hypothesized that the polygenic risk factors for sporadic AD also affect the age at onset of dementia in individuals with an extra copy. From previous research into AD, AD, and DSAD, we already know that the effect of the ApoE has been inconclusive in autosomal dominant Alzheimer's disease in different studies. There's one specific study that looked into APP duplications specifically, and they did not find an effect of ApoE on the age of onset. In Down syndrome, previous studies showed that the ApoE4 allele was associated with earlier cerebrospinal fluid changes and thus possibly earlier onset of the disease. The polygenic risk score has been studied less frequently than the ApoE genotype, but in both autosomal dominant Alzheimer's disease and Down syndrome, the polygenic risk score for sporadic AD seemed to have an effect on CSF levels. So to study the effect of polygenic risk more in people with an extra copy of APP, we constructed two research questions. First, we want to know what the effect is of the ApoE genotype and a polygenic risk score for sporadic AD on age at onset in individuals with an extra copy of APP. And secondly, we want to find out if this effect is similar between APP duplications and Down syndrome individuals. To study this, we gathered information on 92 APP duplication carriers from different European countries, and also including 24 cases from literature. We gathered information on Down syndrome individuals from the Down Alzheimer Barcelona Neuroimaging Initiative cohort. We used 274 individuals for this. We gathered clinical characteristic on age at onset of dementia and also whether they had seizures or hemorrhages. For the genetics, we used genotyping data to determine the ApoE genotype and to calculate a polygenic risk score using the Belenges 2022 paper. For the statistical analysis, we coded the ApoE genotype as numeric, so individuals that carried an E2, E2 or E2, E3 genotype were coded as 0, homozygous carriers of E3 were coded as 1, E2, E4 and E3, E4 carriers were coded as 2, and homozygous E4 carriers were coded as 3. This reflects the increasing risk of Alzheimer's disease with the different genotypes. For the polygenic risk score for sporadic AD, we used the 85 significant SNPs from the Belenges paper, but we excluded 2 SNPs that corresponded to the ApoE genotype because we wanted to look at the ApoE genotype separately. And we also included 4 SNPs due to bad imputation quality and 2 SNPs that were located on chromosome 21. So we ended up with a polygenic risk score of 77 SNPs, and these were standardized within each own population. For the statistical analysis, we took the APP duplications and the individuals with Down syndrome together to analyze the effects of ApoE and PRS, but we also stratified the 2 groups in APP duplication carriers and Down syndrome individuals separately to see what the different effects are on those 2 groups. We associated the ApoE genotype plus the polygenic risk score with age at onset of dementia by using a Cox proportional hazard model to construct hazard ratios and a 95% confidence interval. And then we constructed probability curves to predict the median age at onset for the different genotypes, for the different genetic risks in our groups. So we constructed curves for the 3 APOE genotypes that were present in our cohort, for 5 different standard deviation points of the polygenic risk, and we looked at 2 probability curves for the 2 extreme scenarios. So first, having the lowest genetic risk, and carrying the APOE2 allele, and having the lowest polygenic risk score in our cohort, and having the highest genetic risk, so carrying an E4 allele and having the highest polygenic risk score in our cohort. So onto the results. Here are some patient characteristics of our cohort. In total, we included 366 individuals, and we're looking at the 2 stratified groups. The APP duplication carriers were mostly symptomatic for Alzheimer's disease, 95% of them, whereas in the Down syndrome cohort, only 34% were symptomatic for Alzheimer's disease. When looking at the APOE genotype, these were similarly distributed in both groups, in which most people had were carriers of the APOE E3, E3 genotype. We did not have any homozygous E2 carriers, and we only had 3 homozygous E4 carriers in the Down syndrome cohort, but these were all asymptomatic for dementia. Then moving on to the age at onset of dementia, the mean age at onset in this cohort was 51.5 years of age, and this did not differ significantly between the APP duplication carriers and the Down syndrome individuals. And on the right, you can see the figure with the distribution of all the age at onsets of dementia. When associating the APOE genotype and the polygenic risk score with age at onset, we found for the APOE genotype that it was significantly associated with age at onset, showing a hazard ratio of 1.37. This indicates that an APOE E2 allele is associated with later onset of the disease, and the APOE E4 allele is associated with earlier onset of the disease. When looking at the polygenic risk score, this showed a hazard ratio of 1.29, meaning that a higher polygenic risk score was associated with a higher hazard of earlier onset. When looking at the stratified cohorts, we found similar effect sizes for both of these groups, but only the polygenic risk score in the Down syndrome cohort reached the statistical significant threshold. Then we constructed the probability curves to predict the median age at onset for the different genetic risk. And first, you see here the 3 curves for the different APOE genotypes. The APOE E2, E4, and E3, E4 carriers had a predicted median age at onset of 51.1, homozygous E3 carriers of 52.5, and E2, E3 carriers of 53.9. For the polygenic risk score, you see here the 5 different standard deviation curves, and I'm only showing you the median age at onset of 3 of those. So 2 standard deviations above the mean PRS had a predicted median age at onset of 50.9. The mean PRS had a predicted median age at onset of 52.5, and 2 standard deviations below the mean had a predicted median age at onset of 55.2. Lastly, we constructed the probability curves for the 2 extreme scenarios, so either having the highest genetic risk, so carrying an E4 allele and having the highest polygenic risk score. This predicted median age at onset was 49.3, and having the lowest genetic risk, so carrying an E2 allele plus having the lowest polygenic risk score, had a predicted median age at onset of 59.6. So as you can see, it's a more than 10-year difference in predicted median age at onset between the highest and the lowest risk group. So to come to an end of my presentation, the age at onset of dementia in APP duplication carriers is comparable to that of Alzheimer's disease and Down syndrome. The APOE genotype and the polygenic risk score for sporadic AD influence age at onset in carriers of an extra copy of APP. We found a 10-year difference between the highest genetic risk group and the lowest genetic risk group for the predicted median age at onset of dementia. And our results suggest that these findings from sporadic AD may be transferable to APP duplication carriers, also including Down syndrome. And I want to thank you for your attention, and I want to thank everybody that collaborated on this project for the APP duplication carriers, and also for the individuals with Down syndrome. Thank you.
Excellent. Well, we are definitely starting off with two excellent talks, and we have two more to round out this session. Our next speaker is Elizabeth Andrews from the University of California, Irvine, who's going to talk about age and sex influence, Alzheimer's disease neuropathology, and Down syndrome leading to regional differences. Ellie?
Hi, everyone. First, I'd like to say thank you to the organizers for inviting me. I'm really excited to be here and to present some of the work I've been doing for my dissertation as part of the head lab. So as stated, the title of my talk is Age and Sex Influence Alzheimer's Disease Neuropathology and Down Syndrome Leading to Regional Differences. So thank you, Lisi, for the introduction on the connection between amyloid beta, tau, and Down syndrome. I'm not going to cover that. I'm going to jump right into my study aims. But really the purpose of this was to determine the age at which we see an increase by neuropathology in amyloid beta and phosphorylated tau, or p-tau, in the Down syndrome population, and also to investigate sex differences. So our hypothesis was that age 30, as has been reported before, would be a significant turning point in the pathological deposition, and that women with Down syndrome and Alzheimer's disease would have higher levels of p-tau compared to men with DSAD. Oh, sorry. There we go. And so to study this, we wanted to investigate, again, we are a postmortem tissue lab, so we investigate by staining individual cases and then quantifying it. The platform I used was QPath, which is an open source, freely available digital pathology software. And what we do is we train a pixel classifier to quantify the positive and negative area of these pathologies that we see. And I did this by placing random ROI, or region of interest boxes, across the tissue, and then to calculate the positivity, taking the area, dividing by the total box area. So that's what I'm going to be discussing throughout the talk, is this positivity measure. And this is the cohort of cases that we used. We focused on the frontal and occipital cortexes, because this is where we see earliest pathology, or earlier pathology develop in the frontal cortex, and then occipital is affected a bit later. And as Lisi mentioned, the occipital cortex is also of particular interest in Down syndrome and Alzheimer's disease due to the connection with vascular pathology. So we have a cohort of Down syndrome with AD, as well as young and middle-aged control cases. I'm not going to be discussing those today. I'm going to focus on the Down syndrome and Down syndrome with Alzheimer's disease results. But if anyone would like to discuss, I'm happy to. And this is what we observe in the lab, actually. We see some diffuse amyloid pathology at earlier ages. This is from a 24-year-old individual. And this progresses to mature plaques around the age of 30, as I will show a bit later, but this is an individual who's 57. And for p-tau, we see almost no labeling prior to 30 years of age. And we see very advanced tangle pathology when they're in their 50s, as I'm going to discuss. So this is what we actually see when we graph the positivity by age of the participant at death. And as you can see, the amyloid pathology increases quite exponentially across the cross-sectional age, and this appears to be a bit more continuous in the frontal cortex. However, in the occipital cortex, it shows that there's a bit more variability, where you see some individuals that hang out a little bit lower and some that are maintain very high levels of amyloid at death. I also want to point out that we did look at the distribution of males and females, and you see that there's not a lot of differences, at least in the frontal cortex and the occipital, maybe. But we also looked at p-tau here, and as you can see, the distribution is quite different from amyloid, where we see a lot of individuals who have very high levels of p-tau, which I think highlights the very progressive and quick progression of this disease. Also in the occipital cortex, as you can see, that there is a little bit more variability here as well, where we see a group of females who are kind of starting to sneak up a bit in those higher levels of p-tau. But overall, these differences were not significant. I have a little bit more to report on later. So next, we wanted to apply piecewise linear regression to estimate the change point, and if this agrees with what we see in some of the biofluid and also what we see in the clinic data. And as you can see, the change point we observed in amyloid beta in the frontal cortex was around 32 years of age. And also in the occipital cortex, we saw that that was around 36 years of age. So this is as expected, where the occipital cortex was impacted a bit later. And when we moved to phosphorylated tau, the story got a little bit muddled. So we saw a change point at around 28 years of age by these piecewise linear regressions, and about 35 in the occipital cortex. So that's not what was expected, as these appear to be shifted a bit earlier. But as I mentioned previously, there is a different, the group distribution for phosphorylated tau seems to be more of a two-group distribution. So we applied a different model to look at this and saw that the change point where we see significant progression in the pathology, this is a test that assumes less linearity. So we saw that it shifted to 48 years of age in the frontal cortex and to 53 years of age in the occipital cortex. And this is a bit more in line with what we expected, as the age of onset of dementia is around 55 in this population. So to summarize this portion before I move on to some of my other findings, we saw that amyloid beta and p-tau by neuropathology measures developed in the frontal cortex around 7 years before the occipital, and this was consistent with Dahl-Brack staging that we see in late onset AD. However, as I mentioned, the p-tau distribution was shifted a bit later, and also that age 30 was a pretty significant turning point in terms of amyloid beta and AD pathogenesis and DS, but there's more likely than not a transition zone where we see a very high amount of amyloid pathology, and that can seem to progress to p-tau during this time frame. So I do have a few sex differences results to report, but I did want to take a minute to advocate for why it is important to study this, especially as by measures of neuropathology, because there's not a lot of information out there for DSAD. But in late onset AD, we see that 2/3 of all people with Alzheimer's disease are women, so there is a sex bias that we see in the sporadic AD population. In addition, women with late onset AD exhibit higher levels of pathology at autopsy. In DSAD, there's very few reports of neuropathology, but there was one that found that women with DSAD had higher phospho-tau tangle burden. There's also been reports from neuroimaging that women with DSAD have greater white matter hyperintensities, and the occipital cortex was also impacted by this. In ADAD, I don't believe there's any neuropathology reports, but again, I would love to discuss if somebody knows of any. But we did see that there has been findings that plasma NfL is increased in women as a marker of neurodegeneration, but there have been no significant sex differences reported in amyloid and p-tau biomarkers or PET imaging. So what I found was I regressed on age these markers from neuropathology findings of p-tau and amyloid beta, and I looked at the association between them in men and women with Down syndrome. So in the frontal cortex, as you can see, there is a high association, although it did not reach significance, between amyloid and p-tau in the frontal cortex. But you see that there's a trend in positivity, or positive correlation. And this was not the case in men with Down syndrome, where we saw very little correlation between these two biomarkers. And in the occipital cortex, this did reach significance, highly significant in women, where p-tau and amyloid beta were significantly associated, and this seems to almost decrease or have a negative correlation in the occipital cortex of men with Down syndrome. So I think that there's a few different ways you could interpret this. One, the stronger association in women could mean that there is a tighter link perhaps between amyloid beta and progressing to p-tau pathology, or as Lisi mentioned, the occipital cortex is a hotspot for vascular pathology, so I think this warrants more investigation, especially in terms of how women may be differentially affected in this region specifically. And with that, I would like to thank the lab and to thank my amazing PI, Elizabeth Head, also the lab members who significantly helped me on this, my undergraduate students, Billy No and Freddie Gonzalez, as well as my funding sources. This project originally started from the Women's Alzheimer's movement, which was really helpful in getting my dissertation work off the ground. And I would also like to point out my mom's here with me. She lived in Spain for a bit and hasn't been back in over 40 years, so it's really special to be able to be here. And thank you.
Wonderful. Well, thank you so much. And our final speaker of this session before we go into the discussion portion, and so I would encourage you to start thinking about your questions and points to bring up to the panels or to raise in general. And our fourth speaker is my fellow moderator, Marie-Claude Potier from the Institut du Cerveau in Paris, France. And Marie-Claude's going to talk about neuropathological altercations in the locus coeruleus and Down syndrome compared to other neurodegenerative diseases.
So, thank you, Esther. So I'm a researcher from the CNRS at the Paris Brain Institute at the Salpêtrière. And today I will be talking about this European project that was funded by the Joint Programme on Neurodegenerative Disease between the Paris Brain Institute, the UCL London and KCL London, the CRG in Barcelona, Mara Dirsen was the coordinator, CERBM in Strasbourg with Jan Hero, University of Antwerpen, Groningen in Belgium and the Netherlands. So these are the people who have been involved in this project. The main people in my lab, the postdoc Marta Fructuoso, who did her PhD in the lab of Mara Dirsen. Susanna Boluda, who is a neuropathologist who was trained in Barcelona, who is now working at the Salpêtrière, and Lev Stimmer. In the lab of Peter de Dij and Debbie Van Dam, Yannick Vermeijen, who did all the analysis of bioamine using HPLC. And in the lab of Mara Dirsen, René Krans, who did some of the Western blot you will see with the adrenergic receptors. And also a big thanks to André Strydom, who really helped with the finding all the tissue in the UK brain banks. So why the locus coeruleus? So this is the blue part of the brain in human. You don't see it very blue in the mouse, but in human it's very beautiful. And the locus coeruleus, it sends projection to the spinal cord, the pons, the medulla, the cerebellum, the thalamus, the hippocampus, and various areas of the cerebral cortex. So all the projection, they release noradrenaline throughout the central nervous system, and you see that the noradrenergic cell, they contain this tyrosine hydroxylase, which is one of the limiting enzymes that will produce the noradrenaline, and the noradrenaline will be released and will bind in the postsynaptic adrenoreceptors. So noradrenaline is the major modulator of behavior, attention, mood, motivation, stress, and arousal. It regulates the blood flow, heart rate, sleep, neuroinflammation, and neural survival. It has been shown that the locus coeruleus degenerates and it affects many brain regions which are connected. So it's one of the earliest regions to show neurodegeneration in Parkinson's disease, Alzheimer's disease, and Down syndrome. So our hypothesis in this project was that locus coeruleus alteration in those diseases may reflect common mechanisms leading to dementia. And we asked two main questions I will discuss today. The first is, are neurodegeneration mechanisms in the locus coeruleus similar across diseases? And the second, which was not part of this program but was financed by Sisley Lejeune postdoctoral fellow with Marta Fructuoso, was to look at gene expression changes in the locus coeruleus of mouse model of Down syndrome. So first, we looked at loss of noradrenergic neuron and neuropathological feature, A-beta, p-tau, and alpha-synuclein, and we used 67 cases with control, sporadic AD, Parkinson's disease without dementia, with dementia, and Down syndrome with Alzheimer's disease. And you have here all the list of all the brain banks in Europe that provided this locus coeruleus. So this is the demographic, and you should just look that the mean age of the Down syndrome with AD was significantly lower as compared to the other groups. So we first did some hematoxylin labeling to count the number of noradrenergic cells, and then we looked at the A-beta load, p-tau load, and alpha-synuclein using specific antibodies. And what you can see quite clearly, maybe even from the back, is that A-beta deposits were present in Alzheimer's disease and Down syndrome with Alzheimer's disease, as well as p-tau, and alpha-synuclein was mostly present in the Parkinson's disease cases. So we quantified that. This is the number of noradrenergic cells in all these type of neurodegenerative diseases. We found that noradrenergic cell loss in the locus coeruleus was significant in Alzheimer's disease, Parkinson's disease, and Parkinson's disease with dementia. but not in Down syndrome with AD, although there was a trend. But as you can see, as I said, the mean age of the group Down syndrome Alzheimer was significantly lower compared to all the other groups. And we also observe in the control group a correlation between the number of noradrenergic cells and the age. So the number of noradrenergic cells decreases with age. So there is less degeneration of noradrenergic cells in the Down syndrome AD, which could be due to the fact that they are younger. So then we measured A-beta load, and we found that A-beta load is significantly increased in Alzheimer's disease and DSAD, with a higher increase in DSAD. For tau pathology, we only found a significant increase in DSAD with a trend in Alzheimer's disease. And finally, with alpha-synuclein, the only group that shows significant difference was the Parkinson's disease. Parkinson's disease with dementia, probably because of the loss of neurons, you don't see a significant change. So A-beta and tau pathology are more severe in DSAD with minimal loss of noradrenergic neurons. So then we looked at the level of bioamines and DYRK1A. Everybody knows DYRK1A in this room, I guess. It's a protein encoded by a gene from chromosome 21. So among all the bioamines, noradrenaline, dopaminergic, and serotonergic neurotransmitter, and their metabolites, these are the only significant difference that we could observe. So noradrenaline was increased in Parkinson's disease as compared to Parkinson's disease with dementia. And this metabolite of dopamine was also increased in Parkinson's disease. So we did not see many changes, which could be limited by the number of cases that we had. But the decrease of the level of noradrenaline were only observed in Parkinson's disease. While DYRK1A was shown to be increased only in DSAD locus ceruleus as compared to all the other groups, suggesting a specific role of this chromosome 21 protein in Alzheimer's disease in Down syndrome. Of course, then we looked at the level of adrenergic receptors. As I said, the locus ceruleus neuron, they project into the hippocampus, the prefrontal cortex. And we looked at the level of beta-2 adrenergic receptors, and we showed that there was a decrease of this beta-2 adrenergic receptor in Parkinson's disease with dementia, and a trend nearly significant in Down syndrome with AD. Only in the hippocampus, we could not see really this significant difference in the prefrontal cortex. So there is a reduced abundance of this beta-2 adrenergic receptor in the hippocampus, not in the prefrontal cortex. It's significant in Parkinson's disease with dementia and almost in DSAD. So specific region of the locus ceruleus projecting to the hippocampus might be affected. And the noradrenergic system in this region seems to be quite differentially affected across diseases. And the last thing that we looked at was the early endolysosomal phenotype. As you know, the group of Ralph Nixon described about 25 years ago modified morphology of the endosomal compartment in Down syndrome starting at 28 weeks of gestation. And he showed that this was also present in adults Down syndrome and also with dementia. So the number of neurons containing this enlarged endosome increases with age, but not the amplitude of the increase of size. So we did a lot of work on peripheral cells in Down syndrome and Alzheimer's disease. And in Down syndrome, we found that in peripheral blood mononuclear cell, we have an increase of the size of the endosome in Down syndrome as compared to control. You see here the endosome labeled with a specific antibody. We see a size increase of about 30% in the blood cells, 20% in the lymphoblastoid, about 30% in the fibroblast, and even more in the iPSC-derived neuron from Down syndrome. So we looked in the locus ceruleus, which had never been done before, and we labeled this endosomal compartment with a specific antibody against RAP5. And we looked specifically in the Th-positive cells. So this is the main area of the endosome. This is the density and the total area. And we saw significant difference only with the total area. and only in the Alzheimer's disease group. So we did not see any difference in the Down syndrome with Alzheimer's disease. So there are only modifications of the endosomal compartment, which have been described in the cortex and hippocampus, that are also present in the locus coeruleus, but only in Alzheimer's disease. So the endosomal phenotype seen in other brain area in Down syndrome is absent in the locus coeruleus. And finally, we did correlation analysis between all the variable studies in the same samples across diseases, and we found that the number of significant correlation was the highest in the DSAD group compared to all other disease groups. And I will just focus on the DSAD. In the DSAD, there was no significant correlation with A-beta and p-tau and other variables. The number of noradrenergic cells was positively correlated with age. And DIRC1A was also, which is increased, was negatively correlated with some of the bioamines. So the interesting result that we found is that A-beta and p-tau are not correlated to loss of noradrenergic neurons. So just to recap this part, there is less degeneration of noradrenergic cells in DSAD who are younger, and A-beta and p-tau pathology are more severe in DSAD. So the loss of noradrenergic neuron may occur independently of amyloid and tau pathologies in DS, and the noradrenergic cell in this group could be more resilient to the pathology in a way. The second result is that DIRC1A is higher only in DSAD, highlighting the specific role of this gene from chromosome 21 in Alzheimer's disease in DS. And finally, the endosomal phenotype seen in other brain area is absent in the locus coeruleus of individuals with Down syndrome and Alzheimer's disease, suggesting that alteration in the endosomal pathway may not solely result from A-beta overproduction. So the second part is on the gene expression changes in the locus coeruleus of mouse model of Down syndrome, and Marta Fructuoso continued in the lab with a postdoctoral fellowship from the Sisley Lejeune Foundation, and unfortunately this fellowship do not exist anymore. This is really unfortunate because it was really very good to continue research. So we used a mouse model which is called the TS66YAH mice, which has been done by the group of Yanero in Strasbourg. In a way we can discuss later, but it's a clean TS65DS model. So we first did bulk RNA-seq in the locus coeruleus of these mice, and we saw clearly that the locus coeruleus of the wild type and the TS was quite different. We saw on this volcano plot that there were many genes that were downregulated and less upregulated. And among the upregulated genes, a lot were on the noradrenergic pathway, Alzheimer's disease pathway, and Parkinson. And I just highlight this non-coding RNA, which is increased in the locus coeruleus of the mice, the Down syndrome mice. It's a protective factor against amyloid-induced neuronal injury. It promotes increased spine density in hippocampal neurons from AD mouse models. So it's a candidate that could be interesting to pursue. And finally, we did the single-cell RNA-seq analysis of the locus coeruleus of these mice. And you see here the TSNE that shows all the different types of cells that we could obtain and get the gene expression profile. You see here the expression of DIRC1A and APP in all cell types. We found pathway by the pathway enrichment analysis with the differentially expressed gene from the neuronal cluster, only the neuronal cluster. We found that it's involved in processes related to neuronal and mitochondrial function. And last slide, we also wanted to isolate from the neuronal cluster the noradrenergic cells. So we used like about 500 genes which are specific for noradrenergic cell, and we isolated the cells that express some of these genes. And you see here that this cluster here corresponds to the noradrenergic cell in the wild type and in the trisomic mice. These are the gene category that are modified in these cells, cytoskeleton organization, synaptic signaling, endocytosis, and ROS metabolic process. We showed that there was an increase of specific gene like the LDL receptors, one subunit of the NMDA receptor, and the calcium binding protein. So we are on the way to confirming some of these results, but we found that noradrenergic cells, they have a different gene expression profile in the locus coeruleus of the mice. And we want also to compare to the RNA-seq from human healthy samples that I presented today, which has been done by the group of Mara Dirsen here in Barcelona. I will end up thanking the collaborator in the team, the one in red, who are the ones that participated in this project, the project partners of this GPND project, and some other very important people on the platform of the Paris Brain Institute, the funding networks. And I will finish, because I'm still the president of the Trisomy 21 Research Society until the end of the year. And as you know, there will be the next meeting in Denver next year in June. So please, if you have received the survey, please answer the survey saying that you will come personally to the meeting. I hope it will be a success, as successful as here. Thanks a lot.
Excellent. So we have...
Yes, we can start the...
We have a lot of time for discussion and question and points to be raised. There were, we really covered the gambit of mechanisms and associations that may inform mechanisms going forward. So there are microphones that will be circulating the room for those that are here, and for those of you that are joining us online, please enter your question into the chat or the Q&A function, and there are folks here in the room that are monitoring those, and we'll ask the question on your behalf so that we can have you with us in not just virtually, but actually have your discussion point as part of the conversation as we go forward. So I know I have a ton of questions, but I think that I saw one. No?
We cannot see very well.
Yeah, it's and the same for up at the top. So you may have to yell at us if at one point. Yeah.
So thank you for the talks. Very, very nice talks. I have a couple of questions. One is for Dr. Flores. You show very nice data about the fibrin, the position in the brain. And I wonder whether you have data to the consequence of this in terms of function in the brain, and if there's any relation with CAA, for example, or other vascular abnormalities. And then perhaps I will give a second question to Dr. Potier. You showed there is no relation between neuronal loss and A-beta and tau. Any hypothesis or any other explanation for this? Thank you.
So we'll start with Lacey.
Yeah, so can you hear me? Yeah. Thanks for your question. So in terms of mechanisms, what we know is in mouse models. So we have seen that fibrin can activate the complement receptor 3. So it can activate microglia activation. Sorry for the redundancy, but so it's mainly a protein that will induce neuroinflammatory mechanisms. And I don't know, I forgot your second question. Oh, CAA. So Ellie is working on the classifier, so we can quantify CAA. So we are waiting for that, and we are going to correlate fibrin deposition to CAA.
Yeah. And then I think the question.
Yes. So to answer the second question, so of course we did not observe a correlation between the A-beta deposition, p-tau deposition, and loss of noradrenergic neuron. It does not mean that some soluble factors could be on stage to alter this specific cell type, but at least this is a very clear effect that we saw. In other studies where we looked in different regions of the different cortical area, we also found that, at least in Down syndrome with Alzheimer's disease, the level of course, A-beta pathology is much higher. This is quite well described, but also tau pathology is much higher. So as you know, Down syndrome with Alzheimer's disease is a kind of it's Alzheimer's disease, but it's kind of with differences, which is due to the fact that there are all these genes on chromosome 21. We think that at least for the difference with the tau pathology, DIRC1A could be involved because we are seeing this specific biomarker of p-tau 212, which is the most specific of DIRC1A. So we really believe that there are differences where there are increased pathology, but also, for example, in the locus coeruleus, where it seems to be in a way milder, although the pathology is there, but the effect on the noradrenergic cell is milder.
So maybe kind of building off of some of this discussion, I had a couple questions that tie in a little bit to your presentation, in particular on sex differences. And the first is maybe, Lisi, from your data, do you see any sex differences in the vascular contributions? Do you see anything in terms of that breakdown and the fibrin buildup or accumulation or association, I guess, between males and females?
So, no. We haven't seen any differences, but these are very preliminary findings. But so far, no.
And then with your data and thinking about the sex differences, you showed a number of different things that suggest that women with Down syndrome have higher p-tau tangle burden and white matter hyperintensity, some of that past data, but then there's also this stronger association with amyloid and p-tau. Why? Why do you think you're seeing question.
That? I think, again, there could be a lot of different answers, and there's probably a lot of different contributing factors. Some of Lisi's previous work was showing that women have higher inflammatory markers from postmortem data. It was from IL-6, one of those. Sorry, that's for you on the spot. So it could be an inflammation story that's really contributing to those differences as well.
There's been some evidence to suggest that there's different brain networks really from development all through. So I wondered, in thinking about in individuals with Down syndrome, is that potentially also a contributor here?
Yeah, and I definitely think so. And also that it's really important to look at the different regions, because there's also been neuroimaging data from the general population showing the occipital cortex might be more impacted post-menopause. So there's also another element of having the hormonal and menopause story in this as well.
Absolutely.
Yeah, so it's for Marie-Claude Potier. It's just to follow up because thank you for the presentation. I was puzzled because we see a lot of REM sleep disturbances in individuals with Down syndrome. So they have less REM sleep to start with, and then they decrease with DSAD. And locus coeruleus is a very important structure to regulate REM sleep. So do you have any hypothesis? And I don't know, because you saw also differences between the hippocampus and the cortex. Is there any way you could look specifically on the pathologic alterations and the influence on REM? Because clinically, as I say, this is significant in individuals with Down syndrome.
Yes. Well, thanks for the question. As you know, the locus coeruleus is quite an heterogeneous, although it's a small nucleus, but there are regions that project to specific regions of the brain. So the fact that we see it's only, 2 regions that we studied for the adrenergic receptor would mean that some projecting neurons are more affected, meaning that some sub-region of the locus coeruleus could be more affected. But we could not answer this question because in the collection of locus coeruleus that we had, we did not have, the different regions that we could analyze with all the samples. But it would be, of course, very important to know exactly what regions are more specifically affected. And regarding, I just wanted to mention that the sex effect, because as you know, in the locus coeruleus in human, there are about 18,000 neurons in women and less in men. And in fact, in our data set, we had mainly locus coeruleus from females. And this is also one point that, because they have more, so if they lose, they still have more. So it could be also something that we should redo in a larger cohort of locus coeruleus, but it's quite a difficult a part of the brain to get and to collect. So, and once you've done some experiment, it's really very small, so it's a bit tricky. But there are some sex differences, and probably it would affect the Alzheimer's disease in this specific region. Yeah.
It may contribute kind of across the life course and thinking about all of the factors that are playing a role. And one of those is the genetics and some of the underlying genetics and thinking about that. And maybe kind of a two-part question is, one is do you see any of the contributions from any of the X chromosome linked genes, and if that's something you've looked at? But then really my question is, there's been a lot of, to suggest, at least in late onset Alzheimer's, that the underlying ancestry might be driving the contribution, the genetic contribution of risk. And is that something that you've looked at all within your PRS and in particular within the populations? And what has that told you or hinted at that may be linking to some of the mechanisms? And
yeah, well, first of all, the X chromosome is not in the GWAS, so we're not looking at the variants on the X chromosome, so I cannot say anything about that, unfortunately. And
that's
also,
what I figured was going to be the answer.
Yeah, sorry. And about the ancestry, as I told before, we included mostly people from Europe. So about different ancestry, we don't have the data on people from outside Europe, so also that is something I cannot go into more deeply, unfortunately.
It does raise some of those questions in thinking about some of those ancestral differences, but then genetic that are tied into genetics, and then why you might see some of those, some of these sex differences, but then also what might be is it an inflammation story, is it a vascular story, and how some of that is linked in or tied together for sure. Yeah. Oh. Okay. Mike.
Great presentations. I had a question for Joan. There's still a lot of controversy about the effect of ApoE in Down syndrome. Your study seems to be really well poised to ask this question of ApoE4's effects on the biology, on the pathology in these individuals. Is that something that you'd be able to do with your data set, is to actually obtain information related to A beta and tau pathology and look at the effect, not just on clinical status, but on the underlying pathology?
Yeah, that's a good question. I look specifically into the genetics, so I don't have the data on A beta and P tau, but I think in the cohort there's more data on these people, so it could be a really nice follow-up to look into.
Is that data known in those cohorts?
Yeah, yeah.
I think there was a question over here.
I mean, Mike stole it a little bit. My question was also for Joan about this idea about, do we think the genetics is just starting the clock earlier? Is it leading to a greater kind of accumulation on amyloid, or like modifying the relationship between amyloid and tau? Because all those kind of would have different implications for, say, clinical trial and how aggressive you would treat it. And I think it's a super important question that we don't know.
Yeah, exactly. Yeah, it's really important for trials as well. Something we really want to know more about. Yeah.
I think there's our first question up top. Oh.
Hi.
Yeah.
Hi. My question is for Dr. Flores. Thank you for your presentation. It was very interesting. I have two questions. So the first one is more hypothetical on what do you think the temporality of CAA deposition, blood-brain barrier breakage, and fibrin deposition is? Like, do you think the CAA happens first and that leads to breakage of the walls and then that leads to fibrin deposition, or is it another way around? And the second one is, did you look in the vasculature and the CAA of your data? Is there any difference in the ApoE4 genotype? Thank you.
Yeah. So CAA, we see it begins to aggravate at age 40. And what we see in young people with Down syndrome is they already have some leakage before age 40. We do see more leakage in those that are close to 40 years, so between 30 and 40, but we also have 2 cases in their early 20s that have severe leakage. So we also need to consider that we obtained this tissue from the NIH Neurobiobank, and we don't have much clinical data on this. So there are other factors that could also be contributing to the leakage of the blood-brain barrier, not only CAA or not only Alzheimer's disease neuropathology. Regarding the APOE genotype, we have data on some of these cases, so we are going to analyze whether APOE4 contributes greatly to CAA and if it's also associated to severe fibrin extravasation.
Okay, thank you.
Thank you.
Can I just to continue on just to confirm that when we presented last year the result on CAA, we also did not observe any CAA in cases that were below 35 years old. So it could be something that could appear before. Yeah. But just one point. Did you use other marker of blood-brain barrier leakage?
We have tried with other markers like albumin or IgGs, but they are more soluble, so it's harder to detect than fibrin.
So, but do you think that because fibrin is depositing on the
vessel walls,
the
parenchyma,
together with A-beta or before?
Yeah, so we see it before, but we also see fibrin binding to amyloid plaques.
Okay.
It's not working. My name is Lucia Chavez from the University of Leuven. I have two questions, one for Dr. Flores and another one for Dr. Grunewald. It's about the timing, actually, of the fibrin accumulation in the brain and how it contributes to pathology. So you mentioned already that it activates or triggers the activation of microglia, and I understand that it happens more or less when A-beta is going up in the brain. So how do you connect these? Do you think that the accumulation of fibrin is triggering the activation of microglia and contributing to the formation of plaques? So is it mainly acting on the aggregation of A-beta, or do you think it's acting also in other aspects of pathology?
I don't know. So for that, we will need a mouse model, right? But we don't have like a very good mouse model to address all these questions. So I do believe that this very early aggregation of A-beta is kind of it's weakening the endothelial cells of the blood vessels, and this may contribute to very small leakage of fibrin. And then fibrin will enter the brain. It can activate microglia. We know that in Down syndrome, we have activated microglia since very young ages. And at very late stages of AD neuropathology, microglia become highly dystrophic. So it can be because of several things that are going on in the Down syndrome brain. There's also triplication of inflammatory genes in Down syndrome. So the specific role of fibrin, I think, is going to be very difficult to know.
Well, I find it very interesting because it somehow mimics the effect of APOE. So it connects microglia or the glia part with the aggregation of proteostasis, correct, stress. And I was wondering whether the accumulation of fibrin could actually act as a modifier of onset as APOE4. And I don't know, this is my question for Dr. Grunewald, whether you have taken a look at this in your cases?
At what exactly? I'm sorry?
At APOE, at the potential role of the accumulation of fibrin in your cases and how it could modulate onset.
I don't have any data on fibrin, so I cannot look into that.
Thank you. And just a second, last question. So you mentioned that the APOE, the predicted onset for APOE was, I think, 51 to 54, roughly, for APOE 2 to 4.
Yeah.
And then you provided other numbers, so 49 to 59, when you introduce other risk factors.
Yeah.
What is this telling us? Is this supporting the idea that there are more important factors than APOE in the modulation of onset?
That's a good question. I think we have to look into that more. the entire polygenic risk score had a same hazard ratio as only the ApoE genotype. So I would presume that ApoE is really an important one. We looked at the different common variants in the polygenic risk score as well. So look at the 85 individually, and then there was no single variant there that stood out as ApoE stands out. So I didn't find any more important variants there in data that I have.
Yes. Maybe a couple of years ago, when we looked at the PRS and the ApoE in asymptomatic control individual that had amyloid deposition, to look at the contribution of the PRS to the beta deposition in the brain, we had an interesting result. I don't know if you tried to apply the PRS in the ApoE4 positive and negative. Separating the, applying not in the whole population.
Yeah. We didn't do that.
Yeah, because then when you do that, what comes out is that you still have the SNP involved in the amyloid pathology and clearance, which are involved. So it's kind of, you would think it's ApoE that regulates all the genetic factors for amyloid, but then there are others that are really quite important. I think you could try to separate and apply the PRS because you have numbers that are quite high. Yeah. So it could be interesting to see whether you still have some difference even in the ApoE4. Yeah. Because you mix the ApoE4 with the other one where ApoE4 is...
Yeah, exactly, yeah.
So do you think, just to kind of build on what you said, it's really the synergy of all of the components of the PRS that's driving that linkage. Did you look within the PRS? I know you said you looked individually. Did you look almost in the buckets of the underlying biological mechanisms that the SNPs were associated with, or not? So in thinking kind of about your data and your data, there are some that are vascular related or endolysosomal or clearance related and immune related, and bucketing them that way.
Yeah, we didn't bucket in that way. The common variants that stood out the most were mostly involved in immune system and lysosomal activity, but we didn't bucket them according to these functions, but that's what we saw. Yeah.
Well, interesting, because it does tie in a little bit about mechanistically or even pathologically what you're saying to suggest the mechanism. So yeah.
So I wanted to come back to the fibrogen findings, Lizzie. And last year in the conference, Laia Montoliu presented a three-way collaboration that we had with Eric Johnson at Emory and the University of Gothenburg in the proteomic analysis that we perform in Down syndrome. And it was really interesting because, first of all, this was like the same analysis that we're performing DIAN, in autosomal dominant AD, so the same modules that were, so the same statistical analysis, and there we saw a lot of similarities as expected, but some interesting, some differences. And one of that was that we saw a very earlier changes in the blood-brain barrier, the composition of the collagen, and there. And we thought those were earlier than even amyloid. And so we interpreted the data, and it's about to be published, I hope, as like a weaker blood-brain barrier in Down syndrome. And we saw maybe when CAA kicks in, it's finding a weaker BBB. But maybe as it links to your data, that proteomic data, links very well. It could be that they already have, so it would be fantastic to see if, I know it's very complicated to have autosomal dominant AD, but I would hypothesize, seeing that this is a difference expression in this module in between Down syndrome and autosomal dominant, it could justify that these very early fibrogen changes in Down syndrome as opposed theoretically in autosomal dominant AD.
Yeah, so we also have post-mortem frozen tissue. So we want to look at proteins from the blood-brain barrier to see if this correlates with fibrin leakage in the brain. And my dream is also to have access to autosomal dominant AD brains. So if someone has, please let's collaborate.
So I have a question for Lizzie. So don't you find it weird that in the occipital cortex you get more CAA, but you get A beta later?
Yeah.
You find it weird?
Yeah, I find it weird. But also, so there are very old neuroanatomy studies, and they have reported that the occipital cortex is it's very vascularized compared to other cortical areas. So maybe that's why it's more susceptible to CAA than other brain regions.
But independently of
A beta in the parenchyma, yeah.
I think one of the things that's quite striking in just this conversation is the mechanisms that have come to the top of even the conversation we've had and the presentations. The vascular, the endolysosomal, lysosomal, immune as being drivers of the underlying pathology that you're then seeing when you're looking at the tissue. And so kind of for all of you to think about as you both gave your talk, but then heard from the other speakers, curious as to how you see those intersecting within the different populations that your studies investigated, and what's your, big ideas, out there. What are some of the questions that come to mind for you in those intersections? And I'd open that up to actually the whole audience to think about as we're looking at the DSAD population and the ADAD population, how do we think about asking some of these questions? This is also what we're seeing in late onset and more broadly early onset Alzheimer's as well as being those common mechanisms that are coming to the top.
Yeah, so last year we presented the cerebral meningiopathy in Down syndrome and also the APP duplication, where we found that the cerebral meningiopathy is more severe in the APP duplication as compared to the Down syndrome. And I think you should always see it like Down syndrome, although they have more cerebral meningiopathy than the sporadic AD, they have less than the APP duplication. So you should also see it as they could have protective factors against CAA, whatever they are they linked to the blood-brain barrier or not? And also for the blood-brain barrier, I think there are very few studies. The study of Lizzie is one of the first. And we should really, start more studies on the relationship between the blood-brain barrier, CAA, Down syndrome, because it's very interesting. We could find also protective factor. For example, the endothelial cells that we derive from IPS, they have a lower permeability. So maybe it's the pericytes. Also, you did not mention the astrocyte and the smooth muscle cells and so on. But it's looking at the vascular unit is really very important. Looking at the astrocyte that make connection with the neuron and connection with the blood vessel it's really the start of something that we should continue because it's quite important.
Yeah, I agree with what you are saying already. And when we also look into the AP duplication carriers, the E2 carriers, they have more hemorrhages and more CAA as well. And we don't really see that in the Down syndrome individuals, which is something, maybe explanation what you're already saying. So I think that's really important to look into,
And maybe also develop new imaging method for assessing the brain barrier leakage. I know in Leiden there are some people trying to do some injection of gadolinium arterial to look at blood-brain barrier leakage. So it could be
I think there's a couple of teams around the world that have been doing that and trying to understand that temporal relationship of what happens first and or does it build upon one another. Other thoughts?
I think it's also important that looking at these outliers, what we call outliers in our findings, because some of these people with Down syndrome, they have resilient or resistant mechanisms that can help us address a lot of questions in the field. So.
Yeah, that was one of the themes that we actually, we've hinted to it, but we haven't talked a ton about it, is that idea of resilience, for sure. So I think we have a question over here.
I just wanted to caution us that the changes that we're seeing with CAA and the vasculature in people with Down syndrome and trying to make a connection with ADAD, people with Down syndrome have a 20-fold increased risk of moya-moya syndrome. And they have very abnormal vasculature. Despite the fact that they don't have atherosclerosis or arteriosclerosis, they have very abnormal vasculature, and to define what's driving the changes that you're seeing, for example, with fibrin, is really dependent upon ensuring that you've diagnosed everything that's going on in those cases in those individuals. So I just caution us before we jump to
Yeah,
making conclusions.
so we do have some clinical data on some of our cases, and there was only one with moya moya. Yeah.
But I do think that speaks to, as we just look at today and tomorrow and the time here, right? So thinking about mechanism, what's some of that underlying mechanism, how does that potentially tie in to, as we think about symptomology that an individual is experiencing, and when we see that onset, what might be the underlying biology that's driving some of that onset? How do we measure it through these biomarkers? How do we intervene in potentially trials? But that idea of all the comorbidities that are playing a role, they're very different between these populations, or potentially very different between these populations. And you know, you talked a little bit, Marie-Claude, in terms of the what you're seeing in your work and the linkage in PDD. But then, Lisa, you talked about only 10% of what you see post-mortem have pure Alzheimer's. And so the number of co-pathologies is not just CAA, not just vascular, but there's quite a bit of other work. And there's been other studies that have suggested that alpha-synuclein pathology is also potentially present. I don't even know if that's something we've looked at here in these populations in the post-mortem tissue. So just there's a lot of questions, I think, is Mike, what you're getting to, a lot more questions than we may have answers.
Yes, and as Mike was insisting, I think there are no studies looking at the development of the vascular system in Down syndrome. So it's,
All right, other comments or questions before we?
Yeah, I have a last comment. I mean, I fully agree with Dr. Flores about looking at the full spectrum of the pathology. I think we have been looking a lot into the average, but now if we can look into the outliers and specifically on those factors that protect us against the different pathologies, it could be very, very informative.
Mm-hmm. But we need genetic studies with a lot of cases. Because you look at PRS, but you cannot look at all, you know, SNPs. So yeah. This is an issue.
I think it does underscore the importance of the volunteers and the participants in all of these studies.
And international studies.
International collaboration.
With
the work
clinical
that we do.
data.
Yeah. Okay, any final comments before we close out the session? Okay. All right, well, please join me in thanking our panelists. And as you heard at the opening, there's some instructions that we will receive for the coffee break. So, Dr. Fortea.
So the first. Can I? Yeah. So the first coffee break will be in the Sala Ipostila. That's where we're going to have all the lunches and the poster session. Is downstairs. So you situate yourselves in the hall. Is the room just below. So you have to take any stairs that you see on the way to the left. Thank you very much.
Thank you all.
Yes, I know.
So I've had a little time to adjust, but I actually got one full night's sleep last night.
The vascular system, I think, this is also quite.
jij kan wel zien wat ik doe. Oké, dit...
deep within. Test, test, test. Well.
Deep within the biology of the brain is a set of intricate instructions that define who we are, how we move, how we think, our very sense of self. Sometimes these instructions fail, but at Biogen, we are not afraid to go where others won't. At Biogen, we are pioneering new science that takes us deep into the body's nervous system and stretches wide across digital networks and patient communities to better understand and preserve the underlying qualities of our essential human nature. Science that transforms patient lives by improving mobility, breathing, vision, and brain health. Science that seeks to solve societal problems and create access to innovation. Biogen, where science meets humanity.
Alzheimer disconnects memories, lives, and families. Every day, millions of people around the world are affected by this disease. But at the heart of this city, a leader in cutting-edge research and new connection is born, the Hub Alzheimer Barcelona. A unique and innovative research network that brings together public and private institutions, professionals, researchers, and civil society to move forward together in the fight against Alzheimer's. The future of Alzheimer's research begins here. Our Hub Alzheimer Barcelona. Together, we can change the future of Alzheimer's. Contact us to learn more.
Hi, I'm Jason Hassensauer from WashU in St. Louis. Can we have our speakers come up? Katya, Bessie, Eric? Thank you. So I just wanted to introduce my co-chair from University of Wisconsin-Madison, Segan Hartley. And our first talk, I actually have a small part in this project. I'm really excited to see how Katya puts this together. It's going to discuss discrepancies in assessing intellectual disability levels in adults with Down syndrome and how this impacts dementia diagnoses. So Katya, take it away.
Yeah, thanks so much. Hi, my name is Katja Sankühler. I'm a PhD candidate at the LMU clinic in Munich and also currently a visiting researcher at the St. Paul Memory Unit. And I want to present a study that we've recently conducted on intellectual disability level classification in Down syndrome and how that might have an impact on the dementia diagnosis. So when it comes to diagnosing dementia in adults with Down syndrome, one of the major problems that we face is to distinguish between the new cognitive symptoms and the pre-existing intellectual disability. Compounding that, the severity level of the intellectual disability can vary widely from mild to severe ID. However, recent studies have actually shown that applying an empirically derived cutoff to a cognitive score from a single assessment only can actually reach high diagnostic accuracy for classifying AD dementia and prodromal AD, even more than a one-year longitudinal follow-up due to high variability in longitudinal data. But only if, and that's the important part for this talk, only if we take the intellectual disability level of that patient into account. So rather than having a one-fits-all approach, we would apply different cutoffs based on the intellectual disability level of that patient. And how do we classify these ID levels in the clinical setting? Traditionally, that was done with IQ testing with fixed IQ thresholds per ID level, as referred to in the ICD-10. However, in the more recent classification system, there was a conceptual shift towards more real-world abilities, such as doing groceries, having meaningful relationships, self-care, hygiene, and so on. So everyday life skills that can be summarized as adaptive functioning. And these are typically assessed via informant reports by asking questions to the caregiver or the family member. And in our context, specifically important is that we can also assess this retrospectively, even if the patient is not at their best level of functioning anymore, because we can ask the patient the caregiver, if the patient was ever able to perform a certain task when they were at their best level. And this is something that we can't do when we have to do a direct testing, IQ testing, as in the ICD-10, when they're not at their best level of functioning anymore. However, the DSM-5 does not provide specific thresholds per ID level. This is a disadvantage that we have. It gives rather descriptive guidelines of which criteria needs to be fulfilled for each ID level. And it's a quite detailed list. I want to spare you with that. Instead, I will give an example of one criterion, so to get an idea of the phrasing. So for example, for self-care, patients with mild ID are expected to be completely independent, while patient with moderate ID might need some kind of support, while patient with severe ID might need full support in this. So I think from this phrasing, we can see already that there is a bit of room of interpretation, and this is what Laura Del Hoyo from Barcelona and I wanted to look at in this study. So I wanted to see if there are differences in how we classify the ID levels, despite using the same classification system, the DSM-5, and if those differences can have an impact on the dementia diagnosis using these diagnostic cutoffs. And we wanted to answer the first question by comparing the cohorts from Munich and Barcelona on their baseline visits, retrospectively on cognition and on their ID levels. The cognitive performance was assessed with the CAMCOG-DS, which is a cognitive test battery developed specifically for patients with Down syndrome and assesses different cognitive functions ranging from 0 to 109 points. And the intellectual disability classification, according to DSM-5, was assessed by the neuropsychologist at each site as part of the clinical routine by asking standardized questions to the caregiver. Importantly, we had to exclude patients with severe and profound ID due to low completion rates on the CAMCOG score. All right, here we see some of the demographics. On the left side, Barcelona, on the right side, Munich. The asymptomatic individuals at the Barcelona cohort had a slightly but significantly higher age. Also, the sample size was higher in the Barcelona cohort. For the next slide, I will focus on the asymptomatic individuals. That is due to time constraints of this presentation. We did the same analysis also on the dementia cases, and it showed similar results. So in a first step for this analysis, we wanted to see if the cohorts are similar on their cognitive performance using the CAMCOG-DS. And we found no significant differences between the two cohorts from Barcelona and from Munich on their CAMCOG scores with regards to mean, score distribution, variance. So overall, we can conclude that the two cohorts are very similar on their cognition. However, if we split now the results by the assigned intellectual disability level according to DSM-5, we see a quite different result pattern. So for both patients with mild ID in the stripes and with moderate ID with the dots, the Barcelona cohort showed significantly higher CAMCOG scores compared to the Munich cohort. So how can it be that the cognition is so similar when we look at the total cohort, but so different when we look at the intellectual disability level? And this is probably due to the different distribution of patients with mild versus moderate ID within each site. If we look at the left side here, the Barcelona cohort, fewer patients were assigned with a mild ID level, only 37%, but with higher CAMCOG scores, overall suggesting that the neuropsychologists from Barcelona have a more restrictive approach, let's say, in assigning a mild ID level to their patients, where that was almost the opposite trend in the Munich cohort, with over 60% being assigned with a mild ID level, and overall a broader scoring range, and including also lower cognitive performance levels, suggesting a more liberal approach in assigning a mild ID level. So importantly, these differences exist even though we use the same classification system and the total cohorts were very similar on their cognitive performance. So it's not really expected to see these differences, but rather hints at some kind of different interpretation that we have with these intellectual disability level classification system. And the same results also hold true even when we try to make the chords as similar as possible by matching them on Kamcock scores, on age, sex, and sample size. However, after all, it's still two different chords, still different patients. So in the next step, we wanted to analyze if that result is also, or we see these results also when we look exactly at the same patients. So we took randomly selected case reports from the Barcelona side and from the Munich side, and then the neuropsychologists from each side had to review these clinical reports and rate the intellectual disability level according to DSM-5, and obviously was blind to the original ID level that was assigned at each side. And then we conducted inter-rater agreement within side and between side. And what we found is also the inter-rater agreement within side is not 100%, so even two neuropsychologists at the same side don't always agree on the assigned intellectual disability level, and that the inter-rater agreement between side was lower than within side. So overall, that supports our hypothesis that due to some kind of degree of freedom in this DSM-5 classification criteria, the neuropsychologists seem to have a slight different interpretation of these classification, or might also weight some adaptive skills differently. And that is even pronounced between sides. We suspect probably due to training differences at each side and also cultural differences that might have an impact. And the second question was if that has an impact on the dementia diagnosis when we apply these diagnostic cutoffs that I was talking before about. And for this, we calculated optimal Kamcock DS cutoffs to classify AD dementia versus asymptomatic cases at each site using RC analysis. Importantly, the dementia diagnosis was independent from the Kamcock scores. And what we can see here is that from the curves, that works really well. However, that's less surprising, I would say. That's already what other studies have shown. I think it's more interesting to look at the cutoffs, the optimal cutoffs that we calculated for each side. So just as a reminder, the cognitive performance of the total cohorts were very, very similar. In the asymptomatic individuals, but also the dementia cases. I have not directly shown this, so you need to trust me with this, but that was the case. And we can see here that the optimal cutoffs for the Munich cohort were significantly lower compared to the Barcelona cohort. And that probably mainly driven by the fact that we do these classification differently. And we see here that this is a difference up to 10 points, which if we remember that the Kamcock had a range from 0 to 109 points, can actually make a difference. However, we also wanted to show that this has clinical relevance when, for example, me as a neuropsychologist would see cutoffs from a published paper from the Barcelona cohort. I would say, hey, I also use DSM-5-based ID level classification, so I want to use these cutoffs as a guideline for my clinical reports. So what we did is we calculated sensitivity and specificity when we apply the cutoffs from one cohort to the other. For visualization purposes, I first plotted the sensitivity and specificity for the site-specific cutoffs. So these are the Munich cutoffs applied to the Munich cohort and the Barcelona cutoffs applied to the Barcelona cohort. And now in the next slide, you will see how this sensitivity and specificity will change when we apply the cutoffs to the other cohort. So when we apply the Barcelona cutoffs to the Munich cohort, there is a substantial drop in specificity. Due to the higher cutoff, we would classify more patients with dementia, and that would lead to more false positive cases. While here, when we apply the Munich cutoffs to the Barcelona cohort, due to the lower cutoff, we would miss a lot of dementia cases, let's say, and would create more false negatives. And that also answers our second question, if that might have an impact on the dementia diagnosis. Yes, if we apply the diagnostic cutoffs from another cohort, also meaning that these are not really generalizable. And that brings me to the future directions. The idea also, together with Laura Deloyo, is to develop a scoring system with fixed thresholds per ID level aligned with the DSM-5 criteria. But as kind of a precondition to understand first where are actually the differences? Like, what do we actually do differences? Is it cultural differences? Is it mainly training differences? To also be aware of this while developing that tool. And as in the meantime, as a kind of a take-home message, apply site-specific diagnostic cutoffs. But even if you're here not interested in diagnostic cutoffs, I would say also for multi-center studies that can have actually, or can be very meaningful when we actually use DSM-5-based ID level classification in multi-center studies, because we often compare within severity levels or between severity levels. So I think it's really important to be aware of the fact that this can have, or that there can be differences just purely because of the classification differences that we've just seen. So our recommendation would be to improve the consistency by doing joint case classification or consensus meetings for those kind of studies, and to use standardized adaptive functioning rating scales, even though they don't give thresholds, they do give some kind of consistency to make sure that we ask the same questions. And last but not least, I just want to say thank you mainly to the Barcelona team, to the Munich team, to the neuropsychologist. Also, a big thanks to Laura Del Hoyo that I could do this, this project with. And also thanks to the T21 Research Society. Because of the Scientific Research Exchange Grant, I can be here now and continue that project. Thanks so much.
Good job. Thanks so much, Katja. That was fantastic. Our next speaker is someone very special to me. It's Jitske van der Schaar. She's a PhD student at the Amsterdam Medical Center with Visa van der Vlier. And she's actually doing part of her PhD with me at WashU in the Diane sample. So I'm really excited to introduce her. Her talk is going to be about the impact of genetic testing for familial dementia. And she's going to look at the experiences of patients and families. So Jitske, take it away.
Well, thank you, Professor Hessenstep, for that very kind introduction. We're going to shift gears a little here, because in order to study autosomal dominant AD, we first have to identify the individuals who are affected by it. So I'm going to talk today about genetic testing for monogenic causes of dementia in a memory clinic setting and the impact this may have on patients and their families. These are my disclosures. So as you all know, in a minority of cases, dementia results from a pathogenic mutation in a single gene, and I will refer to those as monogenic causes. Now, over the past few decades, over 50 of such monogenic causes have been identified, not just for Alzheimer's disease, but also for other etiologies of dementia. But while screening for those monogenic causes is becoming increasingly feasible but also affordable, implementation of diagnostic testing in a memory clinic setting remains very limited and varies within and between countries. And one of the key obstacles is a lack of knowledge on the impact this may have on patients and their families. Because on the one hand, finding a genetic cause may help to establish a definite diagnosis for the patient, but on the other hand, for the relative, this can have far-reaching consequences. And while there is empirical data available on pre-symptomatic testing in at-risk relatives, far less is known about testing symptomatic patients in a memory clinic setting as part of their diagnostic trajectory. So together with Dr. Sven van der Lee, who is also with us here today, we studied monogenic causes in the Alzheimer's Center in Amsterdam, which is a tertiary memory clinic specialized in early onset dementia. And the first question we wanted to answer is exactly how rare are these monogenic causes? So using a test panel of 54 genes plus C9 ORF repeat expansions and APP duplications, so this is not just about causes of AD, but also other etiologies of dementia, we retrospectively tested a large cohort of our patients, and we established, or actually Dr. van der Lee established, that the prevalence of these monogenic causes for dementia in our memory clinic population is 3.3%. And then as a next step, he developed data-driven criteria to establish which patients should be tested for those monogenic causes based on their clinical diagnosis, their age at presentation, and their family history. But how many patients would be eligible and would they want to be tested? Now, to study this, we did a retrospective study evaluating over 500 consecutive new patients in our memory clinic using the data-driven criteria, and we found that 27% of those patients were eligible for genetic testing according to the criteria, and of patients who qualified, about half of them decided to proceed with testing, whereas the other half did not. And we found that patients who agreed to be tested had better cognition, higher quality of life, and were more often had an undetermined diagnosis. Now, when we studied how they decided whether to be tested or not, we found that the decisions in patients and their families tended to be mostly quick and intuitive, driven by their personal values, rather than extensive deliberation of factual information. And patients who decided to proceed with genetic testing wished to provide genetic information to their family to enhance actionability for their relatives, but also to reduce uncertainty. And on the other hand, patients who refrained from testing worried about the psychosocial impact on their family, what it might do to their family dynamics, or they simply felt that for them the timing was not right. And then we explored the impact of actually disclosing those test results to patients and their families. And we did this in a mixed method observational study, including participants related to 31 patient cases, and 25 of those tested negative, and in 6 genetic cause, a monogenic cause for dementia was found. So 26 patients and 29 relatives filled out quantitative questionnaires at their first visit to the memory clinic. Then after genetic counseling, then 1 week after learning the test results, and then also 3 months later. And in addition, we conducted 13 semi-structured interviews with 8 patients and 10 relatives, also after disclosing the test results. Then we used linear mixed models to calculate effects of test result time and interaction on our outcome variables, and we analyzed patients and their relatives independently. And due to the exploratory nature of our study, we did not correct for multiple testing. And then lastly, we transcribed the interviews and analyzed those inductively. All right, so these are the 31 patient cases at first visit, and you see the demographic, clinical, and psychosocial characteristics. As you see, a little less than half were female, and average age was 61. And the vast majority of them had children, and about 52% of them reported a first-degree family history of dementia. And we found that patients who would go on to test positive were less often clinically diagnosed with AD, and they also reported lower perceived social support compared to patients who would go on to test negative. And here you see the demographic and psychosocial characteristics of the relatives of those patients, the 29. After counseling, little more than half were female, and average age was 54, and we did not find differences between relatives of patients who tested negative versus those who tested positive. All right, so here you see the psychological outcomes. Green is for the patients who tested negative, orange for the patients who tested positive. The solid lines show average scores per group, and the dotted lines show the individual scores. And then on the x-axis, you see the assessments at baseline, so the first visit to the memory clinic, then after counseling, 1 week after disclosure, and 3 months later. As you can see, the average scores of anxiety, depression, and distress remained below the clinical threshold. And then we found in patients who tested negative, anxiety was lower 1 week after disclosure of the test result, and also 3 months later. Then likewise in the relatives of the patients, we also found that anxiety was lower 1 week after disclosure, but not 3 months later. Then, in interviews, the participants related to the negative cases reported being relieved, happy, or even elated to know that their children, their offspring, was not at risk of carrying a monogenic cause. And some patients even felt unburdened by worries or guilt, saying, well, they may still develop dementia, but not due to my DNA. But then there was also a little bit of ambivalence, because some patients were left wondering if there was not a genetic cause, then how did they develop dementia at such a young age? And then participants related to the positive cases tended to be overwhelmed by the news. One said, well, even though I suspected certain things, it did hit hard. Especially in children of those patients, it led to fear and uncertainty regarding their 50% risk of having inherited this monogenic cause, developing the disease, and, as one put it, ending up like their parent. But nevertheless, they did appreciate clarity, having clarity on what affected their family. Then we assessed the perceived likelihood of having a genetic cause in patients to assess their comprehension in a very basic way. And as you can see, at their first visit, patients estimated this likelihood of having a genetic cause relatively high at 43%. But then patients who tested negative scored lower 1 week and also 3 months after disclosure, whereas positive patients scored higher 3 months after disclosure. And in interviews, we found that negative results were interpreted as not hereditary, not due to a genetic cause, and some even grasped that it meant that most known causes were ruled out, but an element of uncertainty remained. On the other hand, a positive result was explained as an inherited form, meaning that if you have it, you will definitely develop dementia. Then we also assessed to what extent patients shared or talked about the genetic testing and the test result with their partners, their family, and others, and to what extent they received support from those partners, family, and others. And as you can see, patients who tested positive scored lower on sharing with others across all time points. And in interviews, participants in general said that they talked about the genetic testing and the test result with their adult first-degree family members and expected them to pass on the news to more distant relatives. Now, beyond that, some participants spoke quite openly about it with friends and acquaintances, saying, well, that's my way of dealing with difficult things, whereas most tended to be more selective or didn't share it at all, saying, well, this is a family matter, fearing stigma, or considering it no longer relevant after testing negative. And then, even though we found no significant effects regarding support, we did notice that only participants related to positive cases mentioned difficulties in finding support, especially in at-risk relatives. They described feeling emotional distance from their siblings and also feelings of isolation. One participant said, there are times when I feel unsupported by some friends, but at the same time, I know I have to go through this on my own, and no one can take this away from me. Then 3 months after disclosure, we assessed clinical utility and personal actionability in patients, asking them whether they sought out support, prepared for the future, changed their plans or priorities, adjusted their lifestyle, or applied for research. And we found that patients who tested positive more often reported that they had taken steps to apply for research. Now, beyond that, in interviews, participants clarified that clinical utility, if any, was more related to the syndrome diagnosis rather than the genetic test result. And one relative was actually disappointed about the lack of clinical utility, saying, well, what is the worth of knowing what the specific type of dementia is if it doesn't lead to a different approach? But participants agreed that actionability for at-risk relatives in general was much higher. Sorry, again. actionability for relatives was bigger, mostly regarding family and life planning, especially for their children and grandchildren. All right. And then finally, we assessed decision regret in patients, and as you can see, it was relatively low and independent of group. And then in interviews, we asked participants what they would advise other patients who were eligible to be tested for those monogenic causes. And some recommended that it should always be pursued to advance science and also to benefit relatives, whereas others emphasized that it is personal and also related to their circumstances, saying, well, it very much depends on the kind of person they are. So to sum up, this is one of the first studies exploring the impact of DNA diagnostics in memory clinic patients. As you saw, our sample was relatively small. We found it challenging to include participants due to the impact of the dementia diagnosis, the disease progression, and also the burden of care. But on the other hand, we were able to follow 31 patient cases and to collect the experience from patients and their relatives. So the take-home message here is that in this study, diagnostic testing for those monogenic causes for dementia did not cause short-term psychosocial harm. Patients demonstrated a rudimentary understanding of their test result and also valued having this information. And participants considered this to be actionable for research participation and also for decision-making in their children regarding family and life planning. But we did find that patients who tested positive and their at-risk family members may need additional psychosocial support. And then, of course, more research is needed to replicate these findings and also to better explore the impacts of these test results over longer periods of time. So with that, I would like to thank everyone who collaborated on this study, and especially the patients and their families who dedicated so much of their time to this study, sharing their experiences with us. And also thank you for your attention.
Well, great talk, and result disclosure is such an important topic, so I bet we're going to get a lot of wonderful comments later. Next up, we have Betsy Benayam, who is from the Fundació Catalina Syndrome de Down right here in Barcelona. And she will be giving a talk on factors influencing survival in adults with Down syndrome and Alzheimer's disease. So Betsy, come on up.
Thank you very much for your presentation. The work I'm going to present today is a joint work between Dr. Emer McClinchy from the Trinity College and our center. We have studied factors that are influencing survival or mortality after a diagnosis of Down syndrome and Alzheimer's disease. In recent decades, as you know, life expectancy has improved substantially in people with Down syndrome, but this has revealed an ultra-high lifetime risk of developing Alzheimer's disease as they age. Understanding how Alzheimer's disease impacts survival or mortality in this population is very important to allow informing families about prognosis, about optimizing personalized care, and also for guiding long-term planning after an AD diagnosis. Although empirical data on survival after AD diagnosis in Down syndrome and which factors are influencing this remain limited. So our aim in this study was to analyze a large, well-characterized Down syndrome cohort to identify clinical, genetic, and also care-related factors that can be related to post-diagnosis survival. Here you can see our mortality data come from individuals with Down syndrome who died between 2012 and 2024. And this data was collected from the 3 different sources. One is from here, from this hospital in the Catalan Down Syndrome Foundation and within the DAPNI cohort in Barcelona. The other cohort is from the ADAD-DSAD, which is a longitudinal study about aging in people with intellectual disability. And we have also included patients that were living in a specialized residency, specialized in people with in intellectual disability and in dementia. The variables of interest of the study were this that you can see, demographic and AD variables like date of birth, sex, severity of intellectual disability, APOE genotype. In this case, only data was available for the Barcelona cohort. The date of AD diagnosis, if they were taking medication, the date of death, fundamental cause of death, and also care setting at the time of AD diagnosis and also care setting at the time of death. You will see that there are 5 types of care setting facilities. People can live at home with relatives. They can live in a community group home. They can live in an ID residency, in nursing homes, or in a specialized ID and dementia home. Medical, we took only into account medical comorbidities like hypothyroidism, sleep apnea, psychiatric disorder, sensory impairments, hypertension, stroke, vascular conditions, and also the presence of late onset myoclonic epilepsy in Down syndrome, LOMETS. Between 2012 and 2024, a total of 264 patients died. Here you can see that there are 2 differentiated cohorts. One cohort is a clinical cohort from patients from AVISTA and DAPNI. The fundamental cause of death in this cohort was established through the reviewing of the medical records of each patient included in this cohort. And then the fundamental cause of death was determined by an expert clinician. The other cohort from the TILDA study, the diagnosis of Alzheimer's disease or the fundamental cause of death was determined using the death certificate, information that appears in the death certificates. As you can see in the clinical cohort, Alzheimer's disease was diagnosed as the fundamental cause of death in 72% of the patients. COVID was also present, cancer, respiratory infections, and others. And in the other cohort that is based on the death certificates, Alzheimer's disease was only diagnosed in 17% of the patients, which is a very high difference, no? If you see the numbers. The majority of the diagnosis was congenital malformations as the fundamental cause of death in these clinical reports, death certificates, sorry. And our final sample included the patients that were diagnosed with Alzheimer's disease as a fundamental cause of death. Statistical analysis, we use descriptive statistics, Kaplan-Meier survival analysis, Pearson correlations, or independent sample t-test and one-way ANOVA were appropriate. And finally, to identify independent predictors of survival, we also performed multivariate Cox regression models. Here you can see some of the results. 58% were female. The most frequent level of intellectual disability was moderate in 54% of the patients. Late onset myoclonic epilepsy was present in 72% of the patients, which is very high prevalence. Hypothyroidism was also highly prevalent, and sensory impairments were also frequently present. In relation to the AD medication, only 37% of the patients were taking medication against Alzheimer's disease. In relation to the care setting, as you can see at the moment of the diagnosis, the majority of patients were living at home with relatives and in ID residentials. By contrast, at the moment of death, some of the patients that were living at home transitioned to other type of settings, like ID residentials or also nursing homes and specialized ID. Many of the patients transitioned between facilities. The mean age at which AD was diagnosed was 53.7 years. Mean age at death was 59 years. And the mean survival time after an AD diagnosis was 5.1 years globally. At the first moment, we wanted to see which factors were related to AD diagnosis, and we have analyzed the type of care setting, and we have seen that subjects living at home when they are being diagnosed received an earlier diagnosis than those living in ID residential settings. This is statistically significant. Level of ID surprisingly did not affect the time of AD diagnosis. We were expecting that maybe people with profound intellectual disability it's more difficult to detect an Alzheimer's disease, but we found no differences in ID levels on AD diagnosis. And ApoE status was not, in this cohort, was not related to AD, the moment of AD diagnosis, although you can see that there are 2 years of difference between ApoE4 carriers versus non-carriers, although this is not significantly. In relation to the predictors of survival, we have seen that older age at AD diagnosis was associated with a shorter survival time, although this was not statistically significant. It was a tendency. In relation to the level of ID, intellectual disability, we have seen, as you can see here on the right, that people with mild intellectual disability live longer than those that have more severe levels of ID, although this was not statistically significant. But you can see, visually, you can see that there is a trend, a tendency to live less with more severe ID levels. We did not find differences between males and females in survival, although females lived a little bit more than males. In relation to ApoE carriers or non-carriers, we compared those carrying an E4 allele versus the reference group. In this case, we compared it with those having 2 E3 alleles. And we have seen that those non-carriers live a little bit longer than those that are carrying an ApoE4 allele, although this difference was not significant statistically. Late onset myoclonic epilepsy was present in 72% of individuals with Down syndrome. The average age of diagnosis of LOMETS was 54 years. When comparing patients having LOMETS and not having LOMETS, we didn't find differences in total survival time, but we saw that what is really important here is the time from the time of onset of LOMETS since the AD diagnosis. As you can see here, the shorter the interval between an Alzheimer's disease diagnosis and the onset of LOMETS, the shorter the subsequent survival time. What we also see, and this is also interesting, is that age of onset of LOMETS was also significantly associated to ApoE status. As you can see here, those carriers of the E4 allele, ApoE4, lived a mean time of 4 years less than those that were non-carriers. And this is, I think it's very important, no? The effect of LOMETS on survival and also the genetic factors. AD medication was not significantly related to total survival, although those having medication against Alzheimer's disease lived a little bit more than those not having these treatments. We saw that multimorbidity, defined as more than 2 comorbidities, was not related to total survival time after AD diagnosis. And in relation to type care of care setting, at diagnosis, we have seen that those living in a community group home at the moment of AD diagnosis lived longer than those living at home with a relative, although this was not statistically significant. Oh, yes, it was statistically significant, although after adjusting for other confounders, this significance disappeared. And finally, here you can see that the type of care setting at the moment of death is important, is very important, and significantly affects the total survival time. Here you can see that the survival time from those living in a specialized ID and dementia setting live much longer than those living in other types of care settings at the moment of death. So to conclude, to summarize our results, the mean age of AD diagnosis was 53.7 and the mean survival time was 5.1 years after a diagnosis of AD in our cohort, which is similar to other data reported in other cohorts. We have seen that the time of LOMETS, the time when LOMETS begins after the diagnosis of AD is very important. It's an important predictor of survival after AD diagnosis. And this condition is very prevalent in AD, in patients with Down syndrome and Alzheimer's disease. Other factors related to a reduced survival include older age at diagnosis and a greater severity of intellectual disability. And importantly, individuals living at the time of death in a specialist ID and dementia care setting had significantly longer survival times, possibly due to better detection and management protocols for late onset myoclonic epilepsy and other associated complications. So tailored dementia services for people with Down syndrome are of critical value in this population. And thank you very much for your attention.
Another great talk, and just was packed with data and so much implications for, I think, thinking about screening and care. So I think we'll get lots of great questions about that one as well. Our final talk in this session is going to be from Dr. Eric Rubenstein from Boston University. And Eric will be talking about using machine learning to identify novel predictors of Down syndrome, Alzheimer's, dementia. Uh-oh.
I'm stuck on, ah! This is the pratfall before the start.
Can only go uphill from here.
Yeah. All right, well, thank you for inviting me to talk today. So as some of you might have noticed in these talks, like the convention is to have, if you're presenting someone else's work, to have their little picture in like the corner. That's the machine that did the learning, so you all know. So this is our funding, no conflicts to report. So hopefully coming in, people knew that people with Down syndrome get Alzheimer's disease. If you didn't know that, I'm sure you've had a fun morning. Last year, we presented our data, our big US population data on Medicaid and Medicare, which I'll explain later, incidence and prevalence of dementia in those with Down syndrome, Alzheimer's dementia. And these were our findings, which I think we presented last year to show that, people with Down syndrome, if they live long enough, get Alzheimer's dementia, and it's pretty consistent, at least in the US, across sex, race, year, ethnicity, that we really see is a strong effect. It's what we know, but we can show it in population data. Consistent with work done in cohorts, which many of you lead and run, the age of onset and the age of death are super consistent, with an age of onset at a mean of about 54 and an age of death around 60, 59. And this is among 40,000 Alzheimer's dementia cases. So this is over 9 years, tons of cases, huge sample, and it pretty much lines up with, I think, Juan and your group's meta-analysis of age at death and age at onset. So we kind of see this consistency. And to put on my epidemiology professor hat, we know that the cause is the amyloid precursor protein and the trisomy of chromosome 21. And that's the cause, but it's not the singular cause. So in epidemiology, we like to call these causal pies, and it's sort of a theoretical framework for why disease develops. And it sort of looks like Trivial Pursuit, and it kind of acts like Trivial Pursuit. And in the case of Alzheimer's, dementia, and Down syndrome, everyone needs that triplicated 21st chromosome. That is a necessary cause. It is necessary to have that APP gene to develop dementia. However, there are other causes that contribute and work in tandem with that genetic cause. The one that obviously jumps to mind very quickly is longevity, living long enough to exhibit the symptoms, right? If you have the gene but don't live past 30 years old, you're not going to develop Alzheimer's, dementia in your life. So one cause is longevity, living long enough. So we know that everyone with Down syndrome will get Alzheimer's, dementia if they live long enough, but why is there some variation in onset? Not a ton, not as like we see in the general population, but why is there some? How do we account for other causes to make a full, sufficient cause? So when all the pieces of the pie are together, that's when the disease presents. So what else is needed for an individual or a population with Down syndrome to present? And we know, right, we know the APP gene is super strong, but we know that studying other dementias and Alzheimer's diseases, there are other more environmental causes. So I took this from the Alzheimer's Disease International. These were their 14 big dementia risk factors. And just sort of casually thinking about Down syndrome, you know, physical inactivity, infrequent social contact, less education, obesity, hypertension, diabetes, depression, hearing impairment, vision loss. Those are all things that affect the Down syndrome population. Most of those, some of those are, you know, biologically consistent with the genotype of Down syndrome. You know, maybe obesity, but then others are part of the social structure, the social determinants of health that a person with Down syndrome or population with Down syndrome experiences as they live going through their lives, right? There are some of these social determinants of health that we know are risk factors of Alzheimer's, dementia that people with Down syndrome experience. Low income, less education, disparity. How does that interplay with this known strong genetic cause to affect onset and maybe presentation of Alzheimer's, dementia? So this is kind of the model that we use to think about health outcomes in people with intellectual and developmental disabilities more broadly, but Down syndrome really specifically. And there are some fundamental determinants, which is included the biology of Down syndrome, right? Someone with Down syndrome has certain characteristics because of their genes. But they also live in a society, right? There's politics, there's legal rights that they're afforded or not afforded, depending on the country, that all affect them from the day that they're conceived, right? Even from before the day they're born, from the day that they're conceived, it is affected. And then there's stigma, discrimination, and policy that all sort of flows and pushes downstream to affect social status, which then develops mechanisms, which is sort of how either the genes present or how society influences one to develop these conditions, which then lead to outcomes, right? So there is this structure, this societal milieu that a person is in that causes health, right? It's not just this one gene that leads to dementia. It's a very strong thing that leads to dementia, but at the very least, someone has to live long enough to get there. So what we wanted to do was to use 9 years of Medicaid and Medicare data and a machine learning approach to identify whether the presence, timing, and combination of co-occurring conditions and demographic factors predict the timing of incident Alzheimer's dementia diagnosis in adults with Down syndrome in the US, right? So we're taking it as a given that the genetic cause is there. We know the genetic cause is there. Putting that aside, what about everything else? So we use Medicaid and Medicare data. So if you're not in the US, or even if you are in the US, this is a very complicated and annoying thing to have to understand. Briefly, Medicare, old folks, Medicaid, poor folks. And for people with Down syndrome, because the employment rate is so low, most are, or almost all, are on at least Medicaid. It is also written into the law that Down syndrome is a type of disability that can automatically make you eligible for Medicaid. And then Medicare, some folks get, in general, the population gets it when you're 65 or older. For Down syndrome, they can be eligible due to their parents' eligibility. So there's a lot of folks, even though they're younger than 65, that have Medicare in the US. But put together, especially in the older Down syndrome population, we believe our sample encompasses all adults with Down syndrome, especially those over 35. So this is our project. We call it DSAD Max. We have the data from 2011 to 2019. That's for this study. We now have the data through 2022. We can purchase the data every 2 years. So those are the previous 2 years' data. So as long as the government still allows us to do that, which who knows how long they'll let us do that for. So we get all claims, hospitalizations, procedures, we get prescription drugs and demographics from Medicaid and Medicare. And it's really a large near full US sample. There is a small percentage of adults with Down syndrome that are on private insurance. So either their parents or someone is paying for it or they have full-time employment. We sort of think that by the time they're reaching the dementia threshold, they are certainly on a government-provided healthcare. So we use established algorithms from the Centers for Medicare and Medicaid Services to identify for AD, ICD-9 codes and ICD-10 codes over a specified period of time. The algorithm that they present, that CMS uses, has got a bit of a longer window, so you need to watch over 3 years to see the development of a case, given the speed in which this occurs in Down syndrome and the consistency in enrollment in our data. We relaxed that to 1 year. So we only needed 1 year to ensure that what we see as AD is really AD. Oh, and then so we are interested here in this study for incident Alzheimer's dementia. So given our data structure, if you had a claim for Alzheimer's disease in 2011, we can't be certain that's the first time you got that diagnosis because we don't have your data from 2010. So we needed everybody to be in the data for 1 year with no claims for Alzheimer's disease. And then if they cleared that window, then their first claim would be an incident claim, a new diagnosis. So the first thing we did for this study is we got rid of all prevalent conditions because we're interested in onset. So if we can't be certain that it is the first onset, we excluded them from analysis. So these are the conditions we looked at for our machine learning. A lot of these are from CMS, from Medicaid and Medicare. They have the algorithms. You as a researcher can just take them and use them. So because the machine learning idea is kind of let's throw everything at the wall and see what sticks, we just took all of the conditions that they had. And you'll notice some like HIV, some of the cancers, you'd be like, that doesn't really make a ton of sense for Down syndrome. But given the approach, it really doesn't harm us in any way to throw out these conditions that we can easily code. And if it doesn't matter, it'll fall out in the model. We also, through clinical expertise in the literature, added some Down syndrome specific conditions that we were interested in. They're kind of down here, some sleep apneas. We did Hashimoto's, Graves', hyperthyroid. So we kind of have a mix of just general co-occurring chronic conditions that you see in people, and then some Down syndrome specific. We also put in our demographic factors, so sex, race, age, region of the country, that we put in the model as well. So we first restricted to people greater than 30. So our data set goes over 9 years. So we didn't want to have people too young that over 9 years they would never develop dementia. So if you were 21 when the study started, you would be 30 when it was over. So we wouldn't see dementia, we just dropped them. We only put people that we could conceivably see dementia over the time course. We looked for incident cases, and then we selected controls who also had Down syndrome matched to the timing of the case. So for example, if patient 1 is diagnosed with Alzheimer's dementia on January 1st, 2019, and they were in the study for 3 years, so we saw them in 2016, 2017, 2018, 2019, they get their case status, we're gonna take a randomly selected person with Down syndrome who does not have Alzheimer's dementia yet, and pick a point in time where they've had at least 3 years enrollment. So the amount of time we're watching a person, a case and control, is equal. Right? We don't want to be watching the cases for 9 years and the controls for 2 years, because then the cases have so much more time to get these conditions. We want to be sure that the amount of time we're observing each group, the cases and the controls, is the same. And we also want to sort of account for the period, sort of the ages at which people get these claims. So everything had to be coded binary for the model, and we used a benchmark for the incident condition. So we classified these predictors as sort of a prevalent condition or an incident condition. So a prevalent condition is something that is almost something that occurs at birth or in childhood. Cerebral palsy is not something new that someone is going to develop when they're 55 years old. Right? So if you have a claim for cerebral palsy, we're not really interested in when that claim occurred because you've always had it. It's not something that's predicting presenting at a time that's predicting dementia. It's just a factor of what the person, their life. However, for incident conditions like epilepsy, like late onset epilepsy, heart attacks, strokes, we're very interested in when that condition is happening in relation to when the diagnosis of dementia occurs. So we created windows to say, did the person have a stroke 3 months before their dementia diagnosis, 6 months, 1 year, 3 years. So for incident conditions, we had 4 variables to get at the timing. So this in turn gave us 488 predictors in our model. So I don't know much about machine learning, so the machine did it. But we split the data into training and test. We eliminated rare predictors. We used a flexible tree-based learning approach. We tested 960 models to optimize our parameters. I'm not really sure off the top of my head what the parameters were. We calculated something called Shapley additive explanations, which I'll show you in a minute. And we looked at interactions of the top predictors. This model took 2 weeks to run. So we were very happy that it worked, because if it didn't, it would have been 2 more weeks. So this was our sample derivation. We have 157,000 adults with Down syndrome in our cohort. We restricted to those 30 and older. That brought us down to 90,000. We wanted them to be in the study for at least a year. That was 76,000. And then we wanted to get rid of anyone with prevalent Alzheimer's. We needed to have new Alzheimer's cases in our data. So that left us with 55,606 people with Down syndrome at risk for Alzheimer's dementia in our data. This is the demographics. It's hard to read, I'm sorry. We ended up with 16,000 cases. So during this time period, we saw 16,000 new cases of Alzheimer's. Obviously, they were older than those without Alzheimer's. No sex differences, no real, they were more white compared to those without, although that's probably a reflection of age. And then there were some regional differences. Big differences in payer, where those with Medicaid, who were dual enrolled, meaning they get both Medicaid and Medicare, were more likely to develop Alzheimer's dementia. So the first thing we did before the machine learning is we did univariate prediction. So we just did odds ratios for every condition, all 488, with Alzheimer's as our outcome. So this is not factoring correlations between variables, the relationship between variables. So this is just the data, nothing machine learning yet. And our biggest odds ratio for a predictor was traumatic brain injury 3 months before diagnosis. So that had an odds ratio of 12, very small sample size. And again, right, sort of with all of this, the causal explanation is not necessarily there, right? Is it that they had the traumatic brain injury, went to a neurologist, got diagnosed, or is there something about the traumatic brain injury that is involved with the causal pathway of dementia? But the big ones, TBI, epilepsy, a schizophrenic disorder, a spinal cord injury, insomnia. So a lot of neurological type things and accidents in our just sort of crude univariate associations. So this is our model performance. So we made quintile of model predictors. So the 4th and the 5th quintiles had the strongest predictive power. I wrote this down. Our AUC, our area under the curve for the model, was 0.86. And we had a 76% sensitivity and a 73% positive predictive value for being if you're in the 4th and 5th quintile. So consistent with the literature in this machine learning area, that seems good. For other things, it seems a little underwhelming, but based on other similar studies in other populations, this seems to be about where we should be. So these are our biggest predictors based on this Shapley, which is kind of a relative weight. And not surprisingly, right, age is our number one, biggest predictor, which is good, kind of shows the model is finding the correct answer. We know it's going to be age. But after that, we have dual enrollment, so having Medicaid and Medicare, which we think might be a sort of indicator of health service use and access. Then we have a new epilepsy claim 3 years before dementia diagnosis, any epilepsy, any deafness, any hypothyroidism, ulcer 3 years at or before dementia diagnosis, schizophrenia ever, hyperlipidemia ever, a mental health condition 3 months before diagnosis, any anxiety, peripheral vascular disease, being older than 55, and white race. So these were our top 15 predictors. And then what we can do as the next step is, this is, we call a B-swarm plot. So there are all these little tiny dots, and each dot is an individual. If they're yellow, they have the condition. And then the weight, the SHAP value, is how highly that condition is predictive of your dementia diagnosis. So for example, this clear yellow person here, they had epilepsy. They're on the right side, which means they had a dementia diagnosis, and that epilepsy was a super strong predictor of their diagnosis, right? For this person, based on the model, say they didn't have that epilepsy, they probably wouldn't have gotten dementia, right? So it's just the strength of that predictor for that person. So something like age being greater than 45, the people that are 45 really, that have dementia, really it's like the big predictor. And then we sort of see that the purple side is sort of on the protective end. If you don't have it, does it prevent? So we sort of see that something like age 45 is further to the right, which means it's a bigger predictor that's consistent. Something like dual enrollment is got a less of a value, but really the people that are Medicaid and Medicare dual eligible really have a positive, but albeit small, relationship. And the problem with some of this is that the SHAP value doesn't have an intrinsic value. It's a relative measure of weight. So we can't say, oh, the SHAP value is 12, that means so-and-so. So I think that this is where the machine learning state of the art is now, but hopefully we can advance forward to actually have a number. We then took the top 15 predictors and plotted their interactions to see if the combination of two of the conditions or things mattered a lot. Most of them, didn't show anything, that there was really no interrelationship between the two unique predictors. The three that we pulled out as kind of important were epilepsy, incident epilepsy by age, dual enrollment by age, and any epilepsy by age. And these figures are showing, yellow is whether they have the condition, the x-axis is the binary of the variable, and then the SHAP value, how much it predicts. And what these kind of show is that for AD by any epilepsy, that's this one, right? So if you were over 45, and you had epilepsy, or your odds ratio for Alzheimer's dementia and epilepsy was 3 if you were older, and 1.85 if you were younger. So if you have Alzheimer's disease, or if you have epilepsy and are younger, your odds of AD is still high, but not as high as in the older group. And for AD and incident epilepsy, for old folks, the odds ratio is 5.6, and for young people, it was 4.4. So we are seeing this elevated risk in both groups, but there's something more about being older. So our biggest predictor was age, not surprising, makes perfect sense. But interestingly, the other demographic factors weren't very predictive. So race, sex were not really big in our model. Condition-wise, epilepsy really jumped to the top. Low meds tends to come after DSAD diagnosis, but we see the epilepsy before. So the question is this an issue in diagnostic timing, right? These are real-world data. These are clinicians that may see one person with Down syndrome a year. Or is this a different type of epilepsy? Or is our timing of low meds that it can precede the actual date of onset of diagnosable DSAD? We see that predictors of AD in the general population predict timing in DS. So the big general population AD predictors, deafness, hypothyroidism, PVD, ulcers, mental health. We see those for people with Down syndrome too. So our limitations, these claims data have flaws. As I mentioned, the timing is a little washy, a little iffy sometimes based on when the clinician and the system processes the payments. That goes for timing of diagnosis as well. And then we have a survival bias where we're only seeing people that live long enough, where some of these conditions, these predictors might lead to death before AD diagnosis. So I think that the implications of this work is sort of really thinking about the timing of this epilepsy and whether we need to be on the lookout for that before AD diagnosis, thinking more about cognitive reserve and these predictors that we know in the general AD population and some of those interventions that they use and whether that would be applicable for Down syndrome. And then this maybe sets us up for potential risk scores. This is still a black box type algorithm, but maybe we can take it the step further and develop a timing risk score. And I'm going to ask for you to indulge me for, as Americans. I know most of you aren't Americans in the room, but we're sort of the Down syndrome population is kind of facing an existential threat in the world these days with the attacks on the Department of Education and Medicaid and the use of the R word and all of this terrible stuff. And when we talk about Alzheimer's, dementia, and Down syndrome, we need to acknowledge that we need our folks to live long enough. And we have been able to take it for granted that, okay, with the extended lifespan of Down syndrome, this is now a concern. But we shouldn't take that for granted, especially in the US, where some of the reasons why folks with Down syndrome have been able to live longer and prosperous lives is because of these things that are being threatened to take away. So I hope that we are able to not worry about that at next year's and in the future conferences, but I think we should acknowledge that we are hoping our folks live long enough to get to this point. Thank you.
We are open for questions. If anybody has any, I think we have some microphones that will be available. Do you want to start with one, Segan? It looks like you have some written down.
Oh, sure. Well, it was a wonderful group of talks. One theme I think that emerged in Eric and Bessie's talk in particular was sort of this focus on late onset epilepsy or seizures. And I think I was definitely struck by the 70% number, I think, in Bessie's, and just wanted to know if you could comment a little bit about whether that's consistent with other samples, if it was a sample issue, or if it really is something that's that high, and we should sort of think about screening for more strategically. But also, I think you mentioned the APOE4 and seizures sort of had this interaction. Just wondering if you could talk a little bit about is that just like a double whammy where these are two separate things, or you think that somehow they're connected?
Does it work? Mm-hmm. I think it is connected. No, I think we know that APOE4 has influence on the progression of AD, and affects also the time of diagnosis. And in this case, we have found this relationship. Those APOE4 have LOMETs 4 years before those that are not carriers. So, and I think yes, there is a relationship, a biological relationship between these two conditions. Although we have to investigate more, about this. And our team is now going to start a study in epilepsy. So I hope they can answer this kind of questions. But yes, it's important. Yeah, epilepsy is the most predictor, the highest predictor of death. So it's very important to study this condition and to control the onset of LOMETs. Yeah.
Thanks, Ingrid. Eric, I have a question for you. I think I want to put out my own limitation. I'm as perplexed by, I think, some of the Medicare and Medicaid billing that happens as you described. But one of the things you did bring up are the limitations in terms of the lag, I think, in the data reporting or what's available to you in the enclave as part of the claims data, and what you're really looking at was the onset or the incident. And so I've been told that sometimes that lag can be up to even like 2 years in terms of the data that is reported into the system. And I'm wondering how much that may impact some of the trends that you're seeing. I think the fact that you didn't see any differences maybe in sex and some of the things that aren't necessarily changing with time, but some of the things related to the incidents that are going to be a bit more acute, I guess. How do you think that that might shift your understanding of the patterns that you're seeing based on that pretty big limitation from what I understand?
Just to clarify, you're talking about the lag from when the provider puts the claim in, or the temporal, like we don't have the years 2022?
I actually don't know if I know how to ask this question to you, because I just know that there's a lag, and it's not necessarily as real world in terms of the temporal timing as you expect. And when you're putting cutoffs of, you wanted to make sure that they were within a certain age of even potentially even reaching the stage of dementia, how is that impacting the trends that you're seeing if there's at least up to a 2-year lag, I guess?
Okay, so when I mentioned the 2-year lag, that's in terms of the years of data that are available. So like this year, I can buy the 2023 data. So that's just in terms of kind of keeping up to date and like things like COVID and sort of the larger temporal trends and things like that. It takes some time to get the up-to-date data. In terms of like sort of how the clinician reports the data, how the claims are processed, we do sort of have to take it with faith that they're going to put the date in right, that even if there's a delay in processing the claim, it will be backdated to the day that it occurred. We know that there are issues there. There are lots of folks trying to figure out how to best use these data to account for that. But I think that, sort of in terms of the goal of this study and kind of hypothesis generating, I don't think we can say that any of these are super causal, or I think it's sort of material to give back to the labs and the clinics and think about this. Because I think kind of you need a cohort study, something with more thorough phenotypic evaluation to kind of take it that next step. So this sort of is more in the hypothesis generating area than to take it to your clinician and say, do this.
Thank you.
I was also struck by the low meds prevalence and the incidents reported, and following up on Segan's comment, how was low meds diagnosed, Bessie, in the study that you presented? I feel like when we see epilepsy as a big driver in the Medicare data, clinicians in practice are probably calling it epilepsy when in fact it is myoclonic epilepsy. Although those of us who see patients would see complex partial seizures, simple partial seizures, and those exist. So I'm very curious how they went about making that diagnosis. And then are we seeing similar rates in ABCDS that I don't think we have, or any of the other clinicians who've seen adults? Because this is really reportable. It's very, very high.
I think we have high prevalence of epilepsy of low meds because we have analyzed the whole progression, till death. Maybe in other cohorts, epilepsy still have to develop. Epilepsy is not developed. So maybe if you analyze a cohort mortality cohort, then you detect more cases. It's true that sometimes follow-up of patients, when they are in an advanced stage of Alzheimer's disease, these patients get lost, and you don't have data about these patients. But I think that's the issue, that we have more time, to detect, to see if these symptoms appear in patients with AD diagnosis. I am not an expert in low meds, in epilepsy. Maybe you can ask some neurologists here in the unit that are working with our patients. Sorry, Lucia. Yeah, Lucia, like Lucia, says Juan. We have many neurologists in our unit that are working hard on this. So if you want to comment.
Yes, sure. So I think for For your study, you actually revised every Lomet's diagnosis one by one. So all of them were indeed late onset myoclonic epilepsy. Like some of them had seizures when they were children, but this was an actual Lomet diagnosis. So how we diagnose it in the clinical data, it's when they are already with AD dementia, either the first generalized tonic-clonic seizure or myoclonic seizures. If we don't have yet an AD diagnosis, then they would have to do 2 seizures or a seizure with an EEG that's abnormal or myoclonic seizures and generic tonic-clonic. But yeah, it is actually very high. I think it's because it's in the mortality. So when looking at the prevalence, we are now looking at the prevalence in the clinical settings. It's a bit lower, but it gets higher with the raising age and highest in the mortality samples.
And there is also a proportion of patients that in where Lomets start before AD diagnosis. I think the cause is Alzheimer's disease, but sometimes in certain patients it's difficult to do a diagnosis of Alzheimer's disease. No, in some patients, Lomets is the first symptom of AD. No, that's also what we have seen in our cohort. Although it's associated to Alzheimer's disease and it usually appears after a diagnosis, but there are some cases that the onset of Lomets is before AD diagnosis. But I think it's related to Alzheimer's disease.
I have a question for Eric. So, well, of course, for everyone, congratulations, very, very wonderful session. Eric, what I was thinking about when listening to you is how can we improve the care we do for our patients based on your data? And of course, the epilepsy discussion is a very complicated one because many of these cases it will be rather the consequence than the cause of the Alzheimer's pathology independent of onset. And then if you go to hypothyroidism, could be similar. If they're hypothyroid, if the thyroid is not controlled well, the symptoms can appear a little earlier, but maybe not causal. Or it could be causal through some metabolic things. But of course, this is something we have on our radar that this should be looked at. What stood out to me is the ulcer. So that seemed to have a very strong effect. And would you have the chance to look into this a little further if there's kind of a dose effect, so people that have ulcer or treatment for that for a longer time have a higher risk or a lower risk? And is that something we should really focus on further with the clinical picture? Because, of course, chronic inflammation and things like that, there would be some potential causes that this could influence Alzheimer's.
Yeah, so I haven't looked too deeply in the ulcer finding, but I know that it is a predictor in the general population. And I think kind of clinically speaking from our findings is I think that I would say as a non-clinician is to be aware of the general population AD risk factors and that they occur in people with Down syndrome and they might precede diagnosis like they do in the general population. So hearing loss, jumping out on top, we know folks with Down syndrome often have hearing issues, really, sort of treating those conditions. And, whether or not they affect onset of AD, they'll improve quality of life. So that's kind of the thing for all of these is that they're really, it would be great if it delayed AD onset, but even if it doesn't, it's necessary treatment that we should try to prevent, or necessary treatment that will hopefully prevent the onset of these conditions.
Yeah, but the difference between Downs and the general population is they virtually all will get Alzheimer's.
Sure.
So do we have to build a prevention program that would do some screening for ulcer for all of them, especially because in many of them it's not that easy to diagnose it like in the general population. We will have a lower sensitivity with our clinical just questioning them. Do you have?
Yeah, so I think that, I can look up the data again, but the prevalence of ulcers were not incredibly high. Those that had the ulcers, it was very predictive. So it was not something that we were seeing we weren't seeing a ton of patients coming in with ulcers, but of those that did. So probably it would not promote screening just because it's relatively rare compared to the other things. But among those that have it, that maybe that's something to look into closer for the immunocompromisation and all that.
Yeah, hi. Thank you very much. I appreciate all the panelists for all their information speaking about this. I'm in the United States. I'm a community-based neurologist, so my perspective is probably skewed in some respect. I'm going to make one comment about myoclonic epilepsy. My perspective is that it's really underappreciated by a lot of general community-based neurologists. I mean, I see so many times, I work in a lot of support groups and such, where they're having myoclonic epilepsy, and I get messages from their family members, community-based organizations, and they're saying that they're having this, they're not being treated as if they're having myoclonic epilepsy. So I think it's been, at least in some places, it's underappreciated. The second comment I have is the longitudinal information. Definitely late onset Alzheimer's, definitely, people are living a lot longer than the 5-year span, which is absolutely true. But also, I think what's interesting is the quality of life. In terms of that wasn't really looked at, about what's life like. What's that life like, looking in terms of duration of life and the quality of that life, even though in certain settings they might be living longer. But what is that life like? Are they living at a happier life? Is their quality better for themselves and their caregivers and their carers? And definitely for at least myoclonic epilepsy and for those that have associated behavioral difficulties, absolutely, I don't really see such a great, better quality of life. I think it's definitely harder, more of a hardship for them, not to be prophetic about it, but I think it's very true. And also on the longer longevity, what decision-making is made about end-of-life care? And you can extend life forever and ever, but is their quality of life different, or are people making different end-of-life decisions at a certain point? And that might be a difference also about why some are living longer or not in different locations.
I have a quick question for Katja. When you were comparing the intellectual disability assessments between Munich and Barcelona, did anything stick out as different other than what you showed us? Was there less time spent in one group or another? Were they emphasizing different things? What could account for this discrepancy? And also, when you compared the two, did you translate them to English?
Yeah.
Is that what you did? Okay, yeah, just curious.
Yeah, so for the second question, maybe, yes, we translate in English, which is also like, you could say like it's an limitation there, because obviously we're not, I can't speak Spanish, and I think Laura also can't speak German. And for the first question, the question was basically, where are the differences a bit, right? Yeah, so we haven't looked into this. That's something that we want to do, because we assume a big part of this is also like just training differences, and like basically, one neuropsychologist is teaching the other neuropsychologist how to do this. But there might be also cultural differences into this. So for, I don't know, let's take public transportation. I mean, that's now maybe less the difference between Barcelona and Munich, but public transportation, I'm asking this because it's highly relevant in Germany, why it might not be really relevant in the US, because there's not so much access to public transportation in more like other areas. Something like verbal skills, I think, also might be actually where we see differences, or how do we weight this? Because it's, I don't know, I feel like there's also not so much clarity on what it means if the patients are not verbal, but they're actually like very good in their everyday skills. How much do we weight this when they're really good in doing all the other everyday tasks, but they can't really communicate? This is such a difference. So they're kind of like, would be a severe on this communication element, but like highly skilled in other areas.
Okay. And one question for Jessica, since she didn't get one. Jessica, is the rate at which you refer people for genetic testing typical, or is it especially high at your center? And what are the factors that you use to determine if someone should go get genetic testing?
By referring, you mean when we offer them genetic testing for this monogenic process?
Is that rate similar across Europe, United States? It was like 24% or something?
Yeah, no, it varies very much within countries and between countries. I think there are not very definite guidelines on when to offer genetic testing and when not to do it. So I think by establishing these data-driven criteria, we tend to offer it more often, but also because we see a population that's younger and that's looking for a second or a third opinion. So I think it's high in our specific population, yes.
Very interesting.
Yeah.
Well, I think I'm keeping everyone from lunch, so maybe we should break now. And Raquel. Good job.
So thank you, the panelists, for this great session. It's now lunchtime. Lunch will be served in the same room. We have coffee this morning downstairs. During lunch, we will have a poster session that will be a virtual digital poster session. So we have 4 screens. In screen 1 will be basic science posters. In screen 2, clinical neuropsych posters. In screen 3, fluid biomarker posters. And in screen 4, we will have neuroimaging and other biomarker posters. We will ask to the poster presenters of basic science and clinical neuropsych to present their posters today. And for those presenting for fluid biomarkers and neuroimaging, we will ask them to present their posters tomorrow. So the screen in screen 1 will be basic science chaired by Olivia Belbin, and in screen 2 clinical neuropsych posters chaired by Adriaton. So enjoy lunch. Bon appetit.
5 minutos de rigor, nos falta uno. Un minuto de rigor. Bueno. Hello, good afternoon, everyone. Thank you for organizing this wonderful conference. My name is Maria Carmona. I'm a neurologist from here, from São Paulo, the memory unit. And I'm glad to share this, to co-chair this session with Dr. Johannes Levin from the Ludwig Maximilian University. Our first speaker is Íñigo Rodríguez Vaz, and you can start whenever you want.
Okay. So, good afternoon to everyone. Really happy to be here. Thanks to the organizer for allowing me to be here sharing these results with you. This project has been done in collaboration with Ludwig Maximilian University in Munich and also with EDBAB's Biobank here in Barcelona. So, as you already know, Down syndrome is a genetic form of Alzheimer's disease. This is because of the third copy of the chromosome 21 includes an extra copy of the amyloid precursor protein that leads to an overexpression and accumulation of amyloid in their brains. So almost all individuals with Down syndrome will develop Alzheimer's disease pathology by their 40s. In addition to Alzheimer's disease, in Down syndrome is also present often is present alpha-synuclein deposits in form of Lewy body pathology. Lewy body pathology, or LBP, is present in autosomal dominant AD and also in sporadic AD in a frequency and distribution that can vary. In AD, these alpha-synuclein aggregates are often present in the amygdala, what give a amygdala predominant form, but also with spread neocortical LBP can occur. So also LBP in genetically determined AD is proposed to be a secondary phenomenon of driven by AD. The alpha-synuclein seed amplification assay, SAA, has emerged as a highly precise tool for detecting misfolded alpha-synuclein in vivo. Previous studies have shown prevalence of alpha-synuclein seeding activity in 6 to 11% of ADAD cases and from 20 to 45% in sporadic cases. However, the presence of LBP detected by this method in Down syndrome has not been explored until now. So our main goals were to estimate the prevalence of alpha-synuclein seeding activity in CSF from individuals with Down syndrome. Also evaluate its relationships, the relationship of its results with analyze age, sex, intellectual disability, APOE4 genotype, and CSF and plasma biomarkers. Finally, we stratified the results by the presence of AD symptoms in asymptomatic and symptomatic groups, including in this last one, prodromal and dementia AD. Seed amplification assay was performed in Ludwig Maximilian University and followed with two key steps. First, the definition of whether a reaction was positive or negative, being considered positive if the fluorescent intensity was at least 15% of the average maximum fluorescence of the positive control. Second, we analyzed all the samples in quadruplicates and classified the entire sample according to these results. So if at least two reactions were positive, then the sample was classified as positive. If none of them were positive, then was classified as negative. If only one reaction was positive, then we repeated the test. And if still only one reaction was positive after retesting, we classified the sample as inconclusive and was excluded from further analysis. Just to clarify that this protocol has been recently published, showing a sensitivity of 98% and a specificity of 100% for detecting alpha-synuclein in Lewy body diseases. We also carried out a neuropathological evaluation on brain tissue from 19 individuals, donors with Down syndrome. 5 of these donors have also antemortem CSF available for SAA. Evaluations were performed according to established criteria. So in total, we analyzed 270 samples, but 31 were excluded due to inconclusive results or cognitive decline of uncertain etiology. As commented before, we included neuropathological information from 19 brain donors, and also we included a control group, haploid controls that tested negative for SAA. So, moving to the results, out of 239 samples, 22 tested positive, so give us a prevalence of 9.2%. This rate was similar across the groups, the asymptomatic and symptomatic groups, and positive results were stable across the age spectrum. Interestingly, females exhibited a trend for a higher positivity rates, although this difference was not statistically significant. Regarding to fluid biomarkers, we found a higher plasma neurofilaments level in individuals cognitively impaired that tested positive compared to those that tested negative in SAA. No other significant difference were found regarding to other biomarkers or comparing between clinical groups. In neuropathological findings, I know this table includes a lot of information, but I would like to draw your attention to these columns. In this group, we are showing the semi-quantitative burden of Lewy body pathology across the different analyzed regions. According to these results, then we classified the individuals based on McCabe criteria. So Lewy body pathology was identified in 9 individuals, 2 of them classified as olfactory bulb predominant, 5 more as amygdala predominant, and 2 more as neocortical deposition. In this other column, we are showing the results of SAA, and very interestingly, the only individual with abundant neocortical Lewy body pathology was positive for SAA. 2 cases classified as amygdala predominant were negative, and very importantly also, no false positive results were found. This model is an illustrative representation of the distribution and density of the Lewy body pathology in the 9 individuals with confirmed neuropathological data. So the color scales indicate the semi-quantitative burden of the LBP, and samples, individuals in blue boxes are those with available antemortem CSF available for the SAA. So one more time, we can see that the only individual with abundant neocortical Lewy body pathology was positive for SAA. Instead, 2 cases with rare neocortical or bulb amygdala pathology were negative for this test. With all this information, we can say that the rate for SAA positivity was similar to that shown in ADAD, but lower than in sporadic AD. The prevalence found was consistent across the age spectrum, was what is in contrast with some results shown also in sporadic AD. Even though non-statistically significant trend toward higher alpha-synuclein seeding activity in positivity in women, which also differs with previous findings from other cohorts. Positive SAA results were not related to AD symptoms, also in contrast to that shown in ADAD. And even in sporadic AD, results are controversial. Also seems that the positivity increases across the cognitive stages. Finally, only the neocortical Lewy body pathology was only found in individuals aged 61 and 64. So although early alpha-synuclein aggregates can happen, Lewy body pathology distribution could still follow an age-dependent trajectory similar to that described in ADAD. To conclude, we can say that the prevalence of alpha-synuclein seeding activity was comparable to ADAD, but lower than in sporadic AD, with a correlation between neocortical Lewy body pathology and seeding activity. These findings suggest that age-related dynamics and Lewy body pathology progression in DSAD may be shaped by earlier amyloid spread and AD neuropathological changes. Finally, a complex interplay between age, AD pathology, and alpha-synuclein aggregation does not allow for definitive conclusions regarding an age-dependent trajectory of Lewy body pathology in DSAD. If you are interested in these findings, our paper has been recently accepted for its publication in the special issue about Down syndrome in the journal Alzheimer and Dementia. And I would like to finish just giving thanks to all our participants, their relatives, also the brain donors, and also the collaborating teams here in Barcelona, also in Munich. Of course, our funding entities, also, of course, to you for your attention. Thank you.
Thank you. I'm especially happy to present today to Laura Videla, our next speaker, who will be speaking about the DAPNI trajectory in the last 10 years.
good afternoon to everybody, and thank you to the organizers for giving me the opportunity of presenting our work here today in this great conference. Well, I'm Laura Videla. I work as a neuropsychologist at the Alzheimer Down Unit, and there I combine my clinical practice with research. And today I'm presenting some of the milestones that DABNI has reached among these last 10 years. So I would like to mention that obviously this is not all my merit. There's a lot of people working behind DABNI. And I think that DABNI, it is today what it is, thank you to the efforts of each person in the Alzheimer Down Unit, in the memory unit, and of course in Fundació Catalana Síndrome de Down and all the collaborators. So at that point, we are all aware that Down syndrome has a really high risk of developing Alzheimer's disease, and this is mainly due to the triplication of chromosome 21, where the APP gene is located. So is that strong this relation that today Down syndrome is considered as a genetic form of Alzheimer's disease, and that's why we are here today. But despite this ultra-high risk of developing this disease, the clinical diagnosis of this disease is still a bit challenging because there is a baseline intellectual disability and because there are some other co-occurring conditions as epilepsy, hypothyroidism, or obstructive sleep apnea that can hinder this clinical profile. So to give response to these problematics or to this health problem, in 2012, Fundació Catalana Síndrome de Down and the memory unit in Hospital Sant Pau's created this Alzheimer Down unit. And there we developed a health plan focused on neurological pathology in adults with Down syndrome. So there, in this health plan, we offer all people with Down syndrome to have a medical check, a neurological check, and they have a visit with a neuropsychologist, a neurologist, and as well with our nurse team. And we are all have experience with intellectual disability, but also with neurodegenerative diseases. And then 2 years later, Generalitat de Catalunya, the Catalan government, recognized our center as the reference unit for neurological pathology in whole Catalonia. That means that this unit, this medical attention, is included in the public health system of here in Catalonia, and any adult with Down syndrome from all around the territory can come to our unit and get and be part of this health plan. So in 2014, we also launched the DABNI cohort, and this cohort is nested on this health plan with the permission and obviously sending the informed consent of all the adults that come to the health plan. We have this clinical cohort, and DABNI is of course focused on studying Alzheimer's disease. I haven't mentioned it. And we collect all this kind of data, plasma genetics, CSF, neuroimaging. We also collaborate with nuclear medicine here in the hospital. We also have this sleep laboratory, and there's a collaboration as well with the neurological tissue bank in hospital clinic. And since 2 years ago, we have the opportunity as well to offer our participants to collaborate or to participate in clinical trials for Alzheimer's disease. So based on that, we have, during these 10 years, we have visited more than 1,000 individuals with Down syndrome. This data is from the baseline visit. And the main age of our cohort is 43 years old. At baseline, there's a 60% of individuals who are asymptomatic for Alzheimer's, and the main age is 38 years. And then we have a 10% that they are in a prodromal stage of Alzheimer's. That is when they have clinical or behavioral decline, but there's no impact on daily living activities. And then we have another 20% of individuals who are already in a dementia stage. That means that there is a decline, that a cognitive, behavioral, or functional, that it's impacting or that have repercussion on their daily living. And then we have this 10% of individuals with an uncertain diagnosis. This is when we detect some cognitive or functional or behavioral decline, but there's no suspicion of Alzheimer's disease. The main cause can be pharmacological, mental health problems, or other medical concerns. And when talking about Down syndrome, it is really important to take into account the intellectual disability. We have seen how Katia told us, this difference between sites, but I mean, we have to improve this kind of classification, but I think it's really important because it has many implications in how we detect Alzheimer's disease and cognitive decline in this population. So based on all that, during these 10 years, we have learned a lot of things regarding Alzheimer's. For example, that before the age of onset, it's very rare to develop the clinical symptoms of the disease, and that when we follow these people for 5 years, all those who are over 50 years, 60% of them will develop the clinical symptoms of Alzheimer's during this period, and this, and that the prevalence increases thereafter. So there's more or less a 90-95% of prevalence of Alzheimer's disease dementia in the 6th decade of life. So these have obvious impact on lifespan in life expectancy, in this population. And we know that, as Bessie mentioned as well in the previous session, that most of our participants in the DAPNI cohorts are dying due to Alzheimer's disease. So this is a really important issue on health in this population. When we started with DAPNI, it was thought that the presentation of Alzheimer's disease was like an atypical type with more behavioral and frontal symptoms. But now we have seen that memory is one of the first cognitive domains in decline, and this decline is quite similar to that seen in sporadic Alzheimer's disease or in autosomal dominant forms of Alzheimer's. And another important thing that we have learned is that that neuropsychological assessment is really useful to detect these early changes in memory and in other cognitive functions, but that we have to take into account the intellectual disability of this person, because it's not, they will perform different if they have mild intellectual disability, moderate or severe. So we have also studied some risk factors or some factors that could be interfering or changing the course of the disease. In a big study, we didn't see, when we look at sex differences, we didn't find any sex difference in our cohort between male and female when referring to biomarkers or the clinical progression of the disease. But we did see that APOE4, as expected, had those who were carriers of the 4 allele had an earlier and clinical biomarker and clinical changes. I think it was 2 years earlier. And thanks to this longitudinal follow-up in these 10 years, we've been able to diagnose 144 new cases of Alzheimer's disease dementia, symptomatic Alzheimer's disease. So I think that faces know how important it is to have a tight care of this population. Then, on top of Alzheimer's disease, we have studied other co-occurring conditions that we know that isolated or in conjunction with Alzheimer's can be can have a direct impact on health, on the health of people with Down syndrome. Sandra Jimenez, who is in charge of the sleep laboratory, she has described a lot of sleep disturbances. We know that they are very common in this population. Specifically, she has described that they have a reduced REM sleep and an increased slow wave sleep, and that it gets worse along the Alzheimer's disease continuum. And she has also described a high prevalence of obstructive sleep apnea that we know that can impact on the health of people, but also it can worsen the course of Alzheimer's disease. But the good news is that it can be effectively treated and controlled with CPAP. Then moving to epilepsy, which we have seen that it's a really interesting issue now in relation to Alzheimer's. We know that people with Down syndrome can develop epilepsy at younger ages, but also in relation to Alzheimer's disease. And it's been described as late onset myoclonic epilepsy, but also we know that they develop tonic-clonic generalized seizures. And I'm really happy to share with you that in the next few months, here in Hospital Sant Pau, in charge of Dr. Maria Carmona, we will start a clinical trial to test levetiracetam in this part of the population. So I think we will have really nice insights of this topic in the next few years. And then in collaboration with Greece, we have also described some innovative biomarkers. This is HFOs, high-frequency oscillations that we have seen that can be measured during the polysomnography, and that can be a non-invasive and early marker of symptomatic Alzheimer's disease and epilepsy. Then another important element of our cohort is the neuroimaging team. It was first in charge of Dr. Fortea, and now is in charge of Dr. Bejanin. And among this year, we have described this cortical atrophy in relation to Alzheimer's, which faces, again, sporadic Alzheimer's disease, starting in temporal parietal areas, precuneus, posterior cingulate, and frontal areas. We have also correlated some cognitive measures, specifically memory, measured with the CRT and how it, different subtests of the CRT correlate with different patterns of atrophy. And we also have described this basal forebrain atrophy, which seems very specific or very characteristic from Down syndrome. And there is as well this line of research in CAA, where we have seen a more posterior pattern, which seems to be a specific phenotype of CAA in Down syndrome. And in collaboration with nuclear medicine in Hospital Sant Pau, in charge of Dr. Valle Camacho, we have learned a lot about the biology of Alzheimer's in Down syndrome. We have, with different tracers in amyloid and tau specifically, we have learned or we have seen how this amyloid and tau uptakes spreads through all the brain with a similar pattern again as in sporadic Alzheimer's disease and autosomal dominant forms. And this uptake or this amyloid accumulation in amyloid PET starts more or less a little earlier than 40 years. And another core element in our cohort, I think it's the biorepository. Here we have this big biorepository with fluid biomarkers. We have almost 400 CSF samples and almost 2,000 blood samples from people with Down syndrome. And this has allowed us to study in collaboration with many sites, of course, not only us, the core biomarkers of Alzheimer's disease, but also other biomarkers that are emerging or that can be more specific for other, not used so clinically, but maybe they can be useful in the context of monitoring in clinical trials or to better understand the biology of this disease. And thanks to all these efforts and collaborations, we now can understand a little bit better the natural history of Alzheimer's disease in this population, which is really similar to that described in autosomal dominant forms. I'm not going to go through all of it. We have seen these two figures many times, so, and it will be a bit repetitive, but I think that we have to be very proud of the advances we have done in the past 10 years. And of course, just mentioning that all this work and all the advances that there have been done in the field of Alzheimer's disease and Down syndrome, it's in part thanks to the big collaborations that we all are doing all around the world. And I think that this is very important. We have collaborated with the Horizon 21, with the ABC Consortium, and we are now part of a platform of clinical trials, the ADAD-DSAD. So I think that we have to keep working like that. And just to share a brief reflection, I think that the model that DABNI gives us, combining clinical research and biomarker research, has a direct impact on how we treat individuals with Down syndrome, on how we are diagnosing Alzheimer's, and how we are taking care of the whole process. So I think that can have a really good impact on the quality of life of this person, and that, well, we have to keep working together to improve all that. And that's it. Thank you very much for your attention. Thank you to the memory unit, and of course to all the families and participants in the DABNI cohort.
Thank you for this very nice presentation. My name is Johannes Levin from Munich. As Maria already introduced me, I want to use the opportunity to also thank Juan and Raquel for putting together this wonderful meeting. And with this, I want to hand off from the Barcelona team to the WashU crew, and to Julie from Boance's lab to take the stage and present her talk.
Sorry. So I'm going to be talking today about amyloid chronicity, or some people call it amyloid time or amyloid clocks. And this is a project, I do work for Boances, but this is a project I've been collaborating more with Brian Gordon on. This is a project that's happening in the DIAN cohort. I have no disclosures. And I know we've seen this plot a lot, but I want to highlight the x-axis on this. So when we think about the pathological development before symptoms occur, in autosomal dominant AD, we have the advantage of knowing about when someone will develop symptoms based on their genetic history and their parental history. And so what we can do then for our time variable is rather than use age, we can use estimated years to symptom onset, which is really useful in a research context because we'd like to know about all of these pathological changes that occur in the decades leading up to symptoms. The problem is that this doesn't exist for all forms of AD, right? If someone comes in with sporadic AD or is a participant in an ADRC, we don't know kind of about, we don't have as good of a timeline as we do in autosomal dominant AD. And so the idea of an amyloid clock or amyloid chronicity is that you can, rather than anchoring time to when symptoms develop, instead you could anchor it to when someone becomes amyloid positive. And so you'll see this doesn't change the curves at all, it just changes kind of what you're anchoring your timeline to. And these have been developed predominantly using PET imaging, although there have been a couple of publications that have relied on fluid biomarkers to do this. But basically with longitudinal data, if you assume that for a given baseline level of amyloid, the rate of accumulation is consistent across individuals, then you can kind of construct a curve. And so what you do is you take these average rates of change and the baseline level, and you kind of sort your data that way. And then you can fit a model to that data. And there are many publications that have fit models in many different ways. We're going to be specifically applying Toby Bethauser's method, which was published in Brain in 2022, but there are a lot of different ways you can fit a model. But then the advantage of this is once you've built that model, you can then apply the model to cross-sectional data. So then you could take someone who has maybe 40 centiloids of cortical amyloid burden, which is above kind of the threshold for positivity we'd maybe put at between like 16 and 20, and you could say, yes, this person's been amyloid positive for maybe 5 years or 10 years or whatever it is based on just that single cross-sectional value, which can be really useful then in developing a timeline for people. So as I said, we're going to be looking at SILA, or sampled iterative local approximation, as the modeling technique. This approach is available on GitHub. It's a MATLAB package that Toby Bethauser has developed. We're going to talk about a couple of key limitations and then kind of the implications for broader application outside of autosomal dominant AD. So I'm going to be presenting primarily on 278 individuals who have completed longitudinal PET in DIAN. There are 379 people who have completed longitudinal amyloid PET in DIAN in the latest data freeze, and I promise I will circle back and talk about why I didn't include everyone towards the end of the talk. So in these 278 individuals, it's important to note that 28 of them went from being amyloid negative to amyloid positive while they were enrolled in the study, and 22 of them went from being asymptomatic to symptomatic while enrolled in the study, and we're going to use both of these subsets of individuals for some validation steps. So here is just what the cortical amyloid levels look like for these participants. And you can see that amyloid begins to accumulate at a wide range of ages for individuals with the genetic mutations for autosomal dominant AD. And it is possible to reorder their amyloid burden base relative to EYO. But you can see, even with that, there's quite a bit of heterogeneity in when people start to accumulate amyloid. So instead, if we apply the SILA modeling approach and we reorder the data based on that baseline level as well as their rate of change, we get what looks like a pretty nice curve that allows you to think about how long someone has been amyloid positive based on their data. And so first then, what we did is for those individuals that went from being amyloid negative to amyloid positive, we looked at how well our predictions aligned with their actual observed age at conversion. And if the model prediction was perfect, then it would fall on that dashed line. And you can see that we're landing really close to the identity line for these folks. And the average error was 1.2 years. In DIAN, the scanning frequency is every 3 years. So we're below the sampling frequency of the study, suggesting that the SILA estimates are quite good. Then what we did is we looked at, in those 22 people who converted from asymptomatic to symptomatic, and we said how old were they when they became symptomatic and how old were they based on our model predictions of becoming amyloid positive. And so if this worked perfectly and there was a perfect mapping between developing amyloid pathology and then subsequently becoming symptomatic, you would expect to see a slope of 1 and a y-intercept of maybe 15 to 20, suggesting that people are amyloid positive for 15 to 20 years before converting to symptomatic. And so you'll see that we have a slope that's near 1, a y-intercept of 13.5 years, suggesting that on average, these autosomal dominant participants are amyloid positive for about 13.5 years prior to symptom conversion. Although there is some heterogeneity, which we would expect because we know that amyloid does not perfectly predict symptom onset. And then finally here, now these are all the DIAN participants. So these are the predicted age at symptom onset is based on their genetic mutation and their parental history. And then we've got that estimated age at amyloid positivity. And so you'll see there's more noise here in the full group of people. But we still have a slope that's near 1. And I'd say still kind of an estimated preclinical disease duration that's about the same, although it is quite a bit noisier. And then finally, the question is how well does this amyloid time variable map onto cognition? And so you'll see here this first plot just shows kind of a general cognitive composite and how it changes relative to EYO. And so as you would expect, right around that time 0, when people are developing cognitive symptoms, performance on that cognitive composite really falls off a cliff. And the R-squared value for this model is quite high. So EYO does explain a lot of the variability in changes in cognition, which is what we would expect and what we'd hope to see. When we do this with the amyloid time variable instead, that relationship is still there, but it does explain less variability. And I don't think this is surprising at all. It shouldn't surprise people that amyloid doesn't map perfectly onto cognitive changes based on the literature. But I think it's really important to keep in mind because sometimes when we want to apply these timelines, we have to think about what we're anchoring to. And so if we're anchoring to the start of pathological progression, there's still a lot of stuff that has to happen before symptoms actually develop, and there's quite a bit of heterogeneity in there. And so the amyloid time is not going to be perfectly tied to those symptoms. So as I said, I was mostly talking about a subset of participants. And the reason why I didn't include everyone is because not everyone had amyloid pathology when they were scanned. And so the issue is that when you complete a PET scan and you don't have any real amyloid pathology for the PET tracer to bind to, you might come in and have an SUVR of 0.9. And if we scanned you again later, you might have an SUVR of 0.95. And that difference isn't meaningful. It's just kind of the tracer circulating in your body, in your brain. But it's not actually sticking to anything. But that noise, the model is going to interpret, right? If you plug numbers into a model, the model is going to do the math thing. And so what happens if you apply the model to people who don't actually have meaningful pathology is you get these kind of crazy predictions suggesting that you can detect when someone is maybe 30 years before conversion to amyloid positive. That's not meaningful, but it's important to remember that I think not everyone always does when they're applying these approaches. And so we developed we borrowed an approach from another group that is more data-driven and I think is helpful here when you think about who you should include in this type of modeling and who you shouldn't. So we borrowed these methods from the AMIPAD consortium. And basically the idea is that we can estimate who is actively accumulating amyloid and consider those the participants who should be included in the model. And so the way you do that is you can take the longitudinal data, estimate the annualized rate of change for these individuals. Then they apply a Gaussian mixture model, and so you can kind of define what the upper bounds of kind of a normal annualized rate of change is. And then you just do an ROC analysis, and so we come up with an estimate of 1.16 SUVRs as a baseline threshold where you are likely to have a higher amyloid PET value if you come back in for a subsequent visit. And so that's how we that's how we limited our cohort to the 278 people, was we only included people who were above that 1.16 threshold at their baseline scan. And so then that allows us to predict time from amyloid positivity, actually a little bit before people are amyloid positive. You're able to work maybe 3 years out before conversion to amyloid positivity, but certainly not 30 or 40 years out. And so I think this has two major implications. The first one is that, yes, this amyloid clock does seem to work pretty well for people who are actively developing amyloid pathology, but it is most informative at the earlier stages of pathological progression because a lot of stuff has to happen for someone to go from being amyloid positive to actually symptomatic. And so the picture gets a little fuzzier and a little noisier by the time in terms of the relationship between amyloid and symptoms. I think this is also interesting specifically within autosomal dominant AD, because if we know the estimated age at which someone will become symptomatic, and we can now estimate the age at which they were amyloid positive, then we can have an estimate of how long someone was in that preclinical disease phase, and I think that will be a useful variable as we kind of think about heterogeneity in autosomal dominant AD as well. So in conclusion, I think we have evidence to suggest that amyloid chronicity does have good accuracy, and it may be useful when you're thinking about timing of application of drug, but with the important caveats that we can only model meaningful signal. And so with an amyloid PET, that means we need to be only modeling amyloid time in people who actually have plaques that the tracer can bind to. And also, the other folks that were excluded, we did look into the people who were excluded, and of those 100-ish individuals, about 75 of them were just really early. Their EYOs were -10 or greater. So these were just people who are enrolled in DIAN. They don't have any pathology. We wouldn't expect them to. But that other quarter of people, many of them are near symptom onset, but still don't have meaningful amyloid pathology. And so we see a higher representation of APP mutation carriers who we know develop amyloid a little more slowly than the presenilin 1 and presenilin 2. So this only applies to people who are following kind of the median pattern of disease progression, and that's an important caveat as well. And so with that, I'd like to thank my collaborators at WashU, especially Brian Gordon, as well as my collaborators at the University of Wisconsin, especially Matt Zamet and Toby Betthauser, as well as my team in the ANSYS lab. Thank you.
Thank you very much. And to close the session, I call Omar about APOE4. We discussed it a little bit, and we'll learn much more now.
Okay, hello everyone. My name is Omar Abdelmoidi, and I'm currently a rising senior at Washington University in St. Louis. And I have the privilege of being mentored by Dr. Julie Wish and Dr. Bo Ansys in the ANSYS Bioimaging Lab. And today I'll be talking about the effects of APOE4 on amyloid accumulation and tau phosphorylation in adults with Down syndrome. I have no financial disclosures to reveal. Okay, so to start out with, within sporadic AD, it's known that the APOE4 allele is a major risk factor. And so work published by Dr. Forte in 2024 showed that across the ATN, starting with amyloid markers, that the APOE4 allele has a significant effect. So you see the red line, there's lower CSF AB42 across age, which is indicative of AD pathology. And then looking at the centiloid scale, you see across age that amyloid accumulation is significantly affected by the APOE4 allele. With tau markers, we see the same trend with p-tau 181 being significantly affected by the presence of the E4 allele. And finally, with neurodegenerative markers, again, looking at hippocampal volume and plasma NFL, we see that the APOE4 allele has a significant effect within the sporadic AD population. But now moving on to the genetic forms, or Down syndrome more specifically, it gets a little more nuanced. And so work published by Dr. Benjamin in 2021 showed that across age, APOE4 carriers had lower CSF AB4240 levels, which is indicative of AD pathology. And also he was able to show that across age, there is significant impact on centiloid. Within ABCDS, work by Boerwinkle, and I want to point attention specifically to plot B, showed that across age, there wasn't a significant effect on amyloid accumulation that was able to be noticed. And Dr. Fagan recently published work showing across neurotypical controls, ADAD, and then what I want to focus on within Down syndrome, that there was a lower level of AB4240, indicative of AD pathology. She had looked at the effect of APOE4 on this level and did not see an APOE4 effect. But I do want to point your attention that this was only in a cohort of 41. And so all the analysis that I'll be showing later on this presentation has more individuals that are being analyzed within both DABNE, ABCDS, and then I will be showing a combined cohort when I'm discussing amyloid accumulation in the future of the talk. Alongside recent work by Dr. Jana Lise in 2022 showed that p-tau217 has a strong association with tau pathology in Down syndrome, and I'll get into what these implications mean for some of the results as I talk about them in the future talk. Okay, so the aim of the study. I was looking at characterizing the effect of ApoE status on Down syndrome, Alzheimer's disease pathology, using a combined cohort of both ABCDS and DABNE data to increase the power of the analysis. First, I'm going to show the effect of ApoE dosage on centiloids across age, and this is in a combined cohort. The effect of ApoE4 carrier status on CSF AB4240 across age, and this is in each cohort analyzed separately. And then the effect of ApoE4 carrier status on phosphorylated tau217 or p-tau217 across age. And again, this is in each cohort independently, and I'll get into why for the first analysis we were able to combine the cohorts, while the second and third, they're separate on the next slide. So these are the tests performed within ABCDS and DABNE respectively. For amyloid PET, the tracers for ABCDS are PIB and FBP. Within DABNE, it's FBP and FLU. And these were both the amyloid PET were converted to the centiloid scale, and so we were able to then combine them and do a cross analysis. And on first pass, there wasn't a significant distribution difference when looking at these cohorts independently, which I'll be showing as I get into the results for amyloid. For CSF AB4240, ABCDS uses Lumipulse, while DABNE uses ELISA, and we were not able to combine these cohorts because for each of these assays, they showed different measurements. And so while the meaning independently is important to look at, we weren't able to combine it into a single cohort. And then finally, for p-tau217, it had the same story since ABCDS is run on Lilly and then DABNE on Simoa. Okay, so now to get into the results that we found for amyloid. So for ABCDS, we had 241 individuals, DABNE 124. Then we were able to combine it for a total of 365. And this was really important to increase our power, specifically if you look at within our ApoE measurements for 2.4 and 4.4, those are significantly lower in the cohorts independently. So it was really nice to be able to combine it into a larger cohort. So looking at these two independently, looking at DABNE and ABCDS, both of them we see that across age, that amyloid goes up, and at first pass, the distribution was similar between both cohorts. And when we combined them onto a single plot, we saw that there was not a significant difference. So we went ahead and combined both cohorts into a single one for analysis, and we found that across age, looking at amyloid, there seems to actually be a protective effect with E22, 2.3 when analyzing amyloid accumulation. So next, we looked at CSF AB4240. And within ABCDS, we had 71 individuals. Within DABNE, 227. And then we stratified it based on whether they were an E4 carrier or not. And looking at within DABNE, we saw that across age, there is a significant effect in CSF AB4240 with E4 carriers having a lower level, which is indicative of AD pathology. Within ABCDS, while we did not see a statistically significant effect, it is of note that it is in the right trend of it does seem lower, and that there were only 71 participants in the ABCDS cohort, which is significantly less than the DABNE cohort for this analysis. Then moving into p-tau217, we had 297 individuals within ABCDS and 497 within DABNE, and again, we broke it down whether they were E4 carriers or not. And within both cohorts, we see that there is not a significant effect of ApoE4 status on p-tau217 across age. So the summary of all this is that we found a small ApoE effect on amyloid markers. This was seen through an E22, E23. They show protective effects in terms of amyloid accumulation across age. We saw that ApoE4 carriers experienced slower decline in AB4240 across age and a larger DABNE cohort. We also found that tau pathology in Down syndrome does not seem to be affected by ApoE4 status, as for our p-tau217, increase was not affected by ApoE4 carrier status in either of the cohorts. And then finally, I want to point to continued collaboration with the DABNY research program will allow for future analysis with larger cohort sizes as we aim to increase the power behind the analysis that we are able to perform related to APOE4. Thank you.
So I think we open the panel for questions. If there are no questions right now, I might ask you directly a question to Orma. Of course, we all know that in the ATN, p-taus are largely listed under A. So would you, and what would be very interesting, I think, to see is then the downstream consequences of ApoEs, the different one, especially also the protective one. So would you have the chance also to look at tau pads with the tangle pathology and see if there are, or are there plans to do that, to look for potential effects?
Yeah, so within the ABCDS cohort, we did not have enough symptomatic individuals with tau PET to do that analysis. I do know that Dr. Fortea's group is looking at tau PET, and there should be some results with that coming out soon.
Can I ask a question?
Sure.
So about your atypical folks, do you see atypical symptoms, or is it more atypical pathology, like spatial layout in the brain? Like what?
In the clinics, you mean? Yeah. The most typical pattern is episodic memory, like typical Alzheimer's disease. But we do see some participants which have some other manifestations, and we have, well, Dr. Rodriguez-Vaz have published a PCA case in Down syndrome.
Interesting.
And, yeah,
okay.
It was really interesting. Well, maybe he can explain
it. Are you the
better.
lead author on that paper? I will go find it. But yeah, tell me about it.
Yeah, it's, so far we have only found one participant with this presentation. It was very interesting because the participant come to our clinic with the typical presentation of the PCA, and when we performed the neuroimaging, also the atrophy were predominant in the occipital and parietal lobes. Also, even the tau PET uptake was prominent also in the typical areas where amnestic variants are present, but also with more posterior deposition uptake. And yeah, it was really interesting because reviewing the literature, we only found one case reported previously. So we always this new one to the science community, the science knowledge. And probably we thought that there are many more. The problem is that the tools we have for identifying, we have a very specific unit for doing that. Even for us, it's difficult to identify that kind of cases. So I assume that people that is not in their settings, they treat individuals with Down syndrome very frequently. They have much more difficulties for recognize this kind of atypical presentation. But yeah, for sure,
it's cool.
Exists that disease.
We have to be prepared for this atypical presentation because they are anecdotic, but they do exist, and well, they are there.
Yeah.
I just want to compliment Bo and Juan for doing this type of collaboration where you have DABNY and ABCDS work coming together to answer some of these questions, and doing it with Diane as well. It's terrific. My comment is that it seems now the question that I asked earlier this morning has kind of been brought a little bit closer to being settled, where maybe ApoE doesn't have, ApoE4 specifically, doesn't exert such a huge and profound impact on AD pathogenesis as it does in the general population. But my next question will be to look at things like microhemorrhages and ARIA with ApoE4 in this group. I think that would be really important as we stratify for clinical trials, thinking about the impact of ApoE4 on cerebrovascular disease.
And maybe another thing with ApoE4 is the co-pathologies. And I think what we just saw presented by Inigo is kind of opens many new questions, right? How can we have seeding activity before there is amyloid positivity? And maybe is the amyloid positivity as we know it just also something very down the line, because we know that there are vascular changes very early with microbleeds that might reflect already some seeding going on. And so maybe we discussed it quite a bit, but you can share a few thoughts.
For sure, this study has make more questions than answers. But yeah, also the definition of what amyloid positivity is depends on the tool that you have for measure that. And we know that individuals with Down syndrome accumulate amyloid deposition in their brains time earlier before we can detect on CSF or plasma, and we have also the experience of extravesical analysis where some changes are reported before these typical CSF changes, and also the neuropathological data also indicate us that individuals in their 30s have amyloid in the position, so even we cannot determine it by the current diagnostic tools. That does not indicate that that exists, this amyloid exists. So maybe something that happens previously that we can even to detect.
A microphone for Juan.
I was going to say, for Mike's question about
sorry.
For Mike's question about the ApoE for autosomal dominant, we heard earlier it tends to impact the age of onset, and I think that seems to be clear. But we don't see a huge effect on the amyloid accumulation, but we do see an impact on the ARIA rates. And Eric might talk on this later, McDade, that the individuals in the clinical trials who had ARIA were overwhelmingly E4 carriers, so that affects there, even though the amyloid effect isn't.
Hi, thanks for all the talk. Sven, Amsterdam UMC. I have a question on the panel, but maybe also the room. You grouped ApoE 2223, and also in Joan's work, we have not seen a single ApoE 22 carrier. Are there people that have observed in maybe at all a ApoE 22 Down syndrome individual? I mean, epidemiologically wise, every 200 persons should be a 22 individual, and if it stays silent, then it means that likely the 22s either do not reach the age that they can get Alzheimer's disease, likely due to comorbidities that they develop earlier in life, or it's even non-compatible with life. If nobody has seen, so it's just an open question, I don't see anybody answering. And did anybody see in this room an ApoE 22 individual, Down syndrome?
So yeah, we had seen there were ApoE 22 carriers.
Yeah, there were?
Yes, that were included in the study.
Okay, there were, but I didn't, you grouped them, but how many were there? Just a question.
So yeah, that was probably to increase power, and we didn't see a difference, like a group difference between the 22 and 23s, and so they were grouped together, and it was stratified by putting together the 3 4s, 2 4s, and 4 4s in a separate group.
Yeah, just how many were there? Just,
Those.
Are there 6 22s?
That's normal.
In the total, yeah, it was, I believe, less than 10.
Yeah, okay, that is normal. Right.
No, it's just to follow up because I think that Johannes, I was following on Inigo's presentation, and this is very intriguing finding that I think that is worth maybe discussing, and if anyone has ideas, if to us it was very surprising to find these seeding activities in up to 9-10% across the board in all ages. And I think that Johannes has shown with his technique. He's analyzed hundreds of participants now, and he doesn't have issues with specificity, because of course the first question would be to think that maybe this is false positives, but it doesn't seem the case in all the samples that he's analyzed in his cohort. And I think this brings a very interesting pathophysiological question, and in the divergence of something that we measure aggregates, like in PET, in the nice presentations that you've presented, and something that is seeding activity, that it is a completely different technique. And for whatever reason, the body might be able to deal with these aggregates or seeds in the body for many years, because Lotta Granholm, we had the opportunity to collaborate with her. She analyzed, as Inigo was saying, the extracellular vesicles, and you could see the gene dose effect in amyloid expression, but interestingly, also in phospho-tau. So you had children with Down syndrome, and you analyzed the extracellular vesicles of neuronal origin, and they had the increase in dose of APP. That's far to be expected. But it was interesting in those children, they also had increased phospho-tau levels within the extracellular vesicles. So that, linking it together to this seed amplification assay results in the CSF, to me is very intriguing that maybe we have to differentiate when we're measuring activity and when we're measuring tangles or amyloid plaques, aggregates. And I think that's a very interesting and intriguing. We don't claim to have answered anything. We were surprised. But I think that it's very interesting to open a discussion.
And maybe to follow up this, I think I mentioned in my presentation last year that I was like scientifically raised by neuropathologists, and now we need Lizzie and others to look systematically, I think, at very young brains with Lewy body pathology staining, because at least in the ones that we have studied, we didn't find a single case that was SAA positive and didn't have Lewy bodies. For the pathology. So it should be, if that's true, we should find brain areas with Lewy bodies. And so that's something that maybe has not been done sufficiently systematically to look, to do these kinds of staining also in the very young Down syndrome cases. And I think this is it would be a suggestion that if the capacity to do that, I think it's worth looking at.
What is that? Sorry, is it a question for me? No? Ah, okay. Sorry, this was related to the ApoE2 question earlier, but I think it's already answered. We were sort of looking at trying to identify, it's a project with John Hardy and other colleagues at UCL, just trying to identify all the individuals with Down syndrome who are ApoE22 cases known in the literature or across the cohorts. And it looks like there are a few, so it'll be interesting to put it all together. ABCDS, Dabney, Landowns, and a few of the other cohorts, and see, at least at the clinical level, to see what the effect is of having ApoE22 alleles.
I think is there more questions? Yeah. Okay. I was about to say I think the room needs coffee, but
well, now they won't want to answer my question. Okay. This is for Julie. I was just wondering if you could talk a little bit about chronicity and the differences between DSAD and ADAD, if you've thought about that. Like, are the limitations the same or the strengths the same? What's the comparison?
Yeah. So, so in ADAD, like you've got, pre-SEN1, pre-SEN2, and APP, and like we have stratified it by mutation, and we're kind of limited on numbers, but like there is heterogeneity in rates of accumulation based on the mutation. The APP carriers accumulate more slowly, and so then like when you're applying that model in a perfect world, when you're doing that annualized rate of change estimation step, you would calculate group level annualized rates for those 3 mutations separately, but we just don't have quite enough of the APP carriers. We also looked at like pre and post codon 200, and there are some differences. Sporadic AD is surprisingly much more uniform. So I've done this in the Night ADRC cohort and was genuinely surprised. I thought there would be more heterogeneity just because like I think of sporadic AD as being more messy, but specifically with amyloid accumulation, it actually seems much more uniform than Diane, where there's more heterogeneity based on mutation.
And maybe because we have time, I would like to ask you a question regarding the DABNI cohort. In Germany, we face problems with radiation authorities all the time, just because we have one, which I think is a very unique German thing, and we don't need one, actually. At least not for human research. It makes things, at times, very much more complicated. My question is, do you have kind of a limit of radiation that you could expose your individuals to when I see all this fantastic work with acquiring all the PET scans of the different quantity? Is there a limit that is set for research purposes, or do you use it as a clinical readout and then there is no limit? How do you handle that?
I think each country has its own limitations and his own rules for that. I think that here in Spain it is 3 PET scans per year, no?
I'm not sure, I will say more.
More?
Yeah.
Maybe I think Juan Fortea have this answer, can answer this question. I'm not sure of the limit. I know there is a limit. I'm not sure which
but
number it is.
It's
but
not
we don't
strictly
perform.
regulated.
Yeah.
We don't have any authority controlling how many PETs we are doing.
But we don't.
We try
to
not to perform more than 3 each year, but no one is controlling. I don't know by law. Is there a limit of radiation?
I don't know by heart, but for example, in the health study, there are three PETs at least. So at least 3 has to be.
At least 3. Yeah.
So I will say 7, something like that, but I'm not really sure. Yeah, maybe Juan.
El micro, por favor.
So, no, I think that what we do in DABNI, as you know, is a maximum of 2 per year, and when we have a third one, we do it in the off year, so that's what we do. I think that in Europe, the legislation is very varied. I think that in Germany, you can do as much radiation as you can do even in 1 month, but it's lifetime limited. I think this is the most precise that I've been. I think in the US, you have, I don't want to talk about the US, but there is a different regulation. And in Spain, it's more on the IRB, especially it's limited in the research purposes. And we usually follow the US, which is this 2 max for research purposes if there is no clinical, and all the 3rd and 4th, if needed, they are in the off years, so to speak. So that's how we usually do that. But it's less tightly regulated as in Germany, where it's a dose, and it doesn't really matter when you do it. Is that you have like a lifetime, a lifetime dose that you can do for research, and you cannot exceed it. And then it's on the investigators, and of course the subject, if he understands that, what he can do in a given. So it's very heterogeneous, I think.
Okay, thank you. I think with this, I thank you all for your talks, for the interesting discussion, and I think if that's okay with the organizers, we earned ourselves an early coffee break.
Fantastic. I think that there is still another video, that very short video that for those that haven't seen it, because we just didn't, and then we can go for the coffee, a longer coffee, and we continue at 4. Thank you.
Alzheimer disconnects memories, lives, and families. Every day, millions of people around the world are affected by this disease. But at the heart of this city, a leader in cutting-edge research and new connection is born, the Hub Alzheimer Barcelona. A unique and innovative research network that brings together public and private institutions, professionals, researchers, and civil society to move forward together in the fight against Alzheimer's. The future of Alzheimer's research begins here. Hub Alzheimer Barcelona. Together, we can change the future of Alzheimer's. Contact us to learn more.
Oh, then, yeah.
So if they can give you a hand one.
Yeah, have a hand. Yes, but they said I should have a hand mic too.
Yeah.
Yeah, that's fine.
Good afternoon. Now we have a very exciting symposium, maybe controversial, let's see, entitled What is Genetically Determined Alzheimer's Disease? So we consider including new forms. We have a last-minute change. Alberto, you cannot do it. So we thank Brian Gordon from the Washington University in St. Louis to accept being co-chairing this session with me. So the first speaker will be John Hardy. That's chair of the molecular biology of neurological diseases at the Institute of Neurology at UCL London.
Shall I start?
Okay, yeah, please,
okay,
please.
Sorry. Okay, thanks for... Thanks very much for inviting me. I have to say, I've been to the Sacra Familia about 5 times, and I have never been to this building, which is so incredibly beautiful. I should say that the first time I went to the Sacrofamiglia, this is of course off topic, it was not complete. Of course, it's never, ever probably going to be complete. And I'm so terrified of heights, they had to send someone up to actually bring me down because I froze at the top. Anyway, but it's a bit easier now, it's a bit more complete. Anyway, that's a bit off topic. I'm going to start, before I start my slides, by suggesting you think of an alternative reality where people with Down syndrome developed, let's say, bowel cancer at the age of 50. And we decided as a community that we weren't going to treat the Downs people with bowel cancer because, after all, we didn't know if it would be safe in the treatments which have been approved for others would be safe in them. So we just sat around, maybe tried some other therapies, but we didn't try the therapies which were being allowed in the rest of the community. I don't think we'd stand that, and yet we're in that position, I think, with Down syndrome. We know, and I'm spoiling my own talk here, but we know that people with Down syndrome are going to die of Alzheimer's disease if they get over the age of 40. And we're still doing, if you like, clinical trials, but we have, I mean, they're not perfect, but we have effective medications. Why are we not? I just don't really get it. I understand the processes, but I don't get it. Why, as a community, we don't say, enough's enough. These guys are dying of a disease, and we're saying to protect them, we're just going to let them die. I think it's mad. I personally think it's mad. Anyway, that's a bit off topic, but actually, if you remember that, the rest of the talk's irrelevant, because that's what I think. Okay. Well, this is my disclosures. I've consulted for Eisai, Lilly, and Roche in the last 3 years. So this is what I'm going to talk about. The amyloid hypothesis is actually a Down syndrome hypothesis. It started with Down syndrome. I get both the credit and the blame for it with Dennis Selko, but actually I'm a geneticist, and when I started to study genetics, I did it. I'm a pharmacologist originally. I started to do genetics because there were all these competing theories of Alzheimer's disease, which, no agreement. Everybody had their own theory. And I thought, when I saw Jim Gusella's Huntington paper on chromosome 4, I thought, what a cool paper this is. I've been studying how the disease ends, and he's going to find out how Huntington's start. And really, if we could do that with Alzheimer's disease, we can see where it starts. I'm going to show you a pathology slide, but I'm sure you've seen them before. And we'll know how it starts, and we'll be able to draw a stupidly simple idea, a simple line between how it starts and how it finishes, and then we'll be able to intervene in the disease. And so when Jim Gusella published that, I was very fortunate to get a job for reasons which I'll tell people in the bar, but nothing to do with my talent, in a genetics department, even though I was a pharmacologist. And the quid pro quo is I would teach the students biochemistry and pharmacology, and my colleagues in the department would teach me how to run a molecular genetics lab. That was how I started. And I saw that really there were all these different ideas about how the disease started, and genetics was going to choose which of them was right. I've already said this, but Alzheimer's disease, and other people have said it today, is almost universally the cause of death of Down syndrome individuals who live past the age of about 40. We know when amyloid deposition starts. I've known this from the, really getting way ahead of myself, but I've known this since working a little bit alongside Tony Holland and Dr. Zeman, who's here. So I've been following their data when I was part of the Londowns Consortium. I'll talk about ARIA. ARIA, of course, the amyloid-related imaging abnormality, that is clearly a problem. But I think it's a surmountable problem, and now we're getting real clinic data, and I'll just show one paper about this from the States. I think it's a surmountable problem. It's a surmountable problem. I will suggest while care is needed, because we know what I've just said, we know when amyloid deposition starts, it's a surmountable problem and may even be avoidable in Down syndrome. And as I said in my rather emotional intro, if this was a cancer drug, there'd be no question about us giving it. I just don't understand why we're tying ourselves in knots. Of course, I've been following and actually commented on the approval processes, which are, certainly idiosyncratic and result in different administrations across what in England we laughingly call Europe. And also, of course, we have two processes within England. We have the approval process, is the drug okay to give, and then is it cheap enough for us actually to give? So it's two different processes, and of course those two committees have reached different decisions. So this is a complicated mess. I think it's such a shame we don't have a more well-informed and unified system. Then we can start to pool data between let's call them the allies in the West, for the sake of a few months when we might still be allies. Okay, so this is the first statement on the, sorry to the Americans in the room. This is the first statement of the amyloid hypothesis, and it's from George Glenner. George Glenner wrote this. He first sequenced the peptide. He sequenced it from the cerebral blood vessels, and he got a sequence from an Alzheimer case. And then later in the same year, he sequenced it from a Downs case and realized it was the same sequence. And he wrote, this is the first chemical evidence of a relationship between Down syndrome and Alzheimer's disease. It suggests Down syndrome may be a predictable model for Alzheimer's disease, assuming the name's changed. Assuming the amyloid peptide is a human gene product, it suggests that the genetic defect in Alzheimer's disease is located on chromosome 21. He didn't say it quite here, but he clearly thinks that the reason that Down syndrome individuals get Alzheimer's disease is because they've got 3 copies of the gene. And I should say that this paper was submitted to Nature. Like all good papers, it was rejected, and it ended up I think, in BBRC. So that's where it ended up, but it's really all here. This is the pathology. We always focused on plaques and tangles. Plaques in the middle there, they're about 1/10 of a millimeter across, made up of basically A-beta, but there's also a lot of membranes there, and I think that's important. And tangles there on the left, made up of the tau protein, and then on the right, the pathology we always ignored, which is the congophilic angiopathy, which of course has come back to bite us now. I'll just say a couple of things about it. So I started working on Alzheimer's disease, well, in 1976. I don't know how many people in the audience were born in 1976. It was a good year. Anyway, but you used to go to meetings, and at the meetings, there'd be 30 people, 40 people working on Alzheimer's disease, and that was it. If there were 30 or 40 people, there were maybe 20 different theories about what caused the disease among the 40 people. It was chaotic, and so it really needed, if you like, a unifying, and that's what was the attraction of genetics. Before I move on, I'm just going to make a more general point, which is this idea that protein overproduction causes, in the main talk, Alzheimer's disease. Protein overproduction of A beta causes Alzheimer's disease. The same is also true, as shown by shown by us in top left, in Parkinson's disease. Synuclein overproduction causes Parkinson's disease, and Dominic Campion showed that amyloid overproduction, simple overproduction in the cases I'm sure you know about with the APP gene duplications, cause Alzheimer's disease. And he also showed that tau gene duplications cause Tangle disease. These are really simple ideas. You make too much of it and it falls out of solution. I'm making the point from a genetics point of view. This paper from Michaela Vendruscola in Cambridge makes exactly the same point from a protein chemistry point of view. He says that the genes, these proteins, they're all highly expressed and they're all close to what one might think of as their crystallization threshold. The other point implicit in this really, if marginal overproduction is going to give you the deposition, then what that's also saying implicitly is if you're not very good at clearing the protein, you're also going to get the same problem. So, and in fact, that's as we've done all the GWAS for, in fact, all of the diseases that are up here, that we're finding that in the majority of late onset cases, the major problem is we're not very good at clearing them. And so, if you like, a simple overstatement is early onset disease, make too much, late onset disease, you're not good enough at clearing it. Of course, probably the majority of people who get these diseases are making a little bit too much and they're degrading just a little bit too little. So that it's probably that balance. So these are the APP gene duplications. With Dan Nizitek, I don't know if Dan's here, with Dan Nizitek, Dan did quite a lot of work on organoids and found that base 2, which is also a chromosome 21 gene, and had been, if you like, ignored because in the resting state, in the control brain, it did not really contribute to the metabolism of APP. In fact, when you get the microglial and astroglial upregulation around plaques, you get upregulation of base 2, and it contributes quite considerably to the metabolism. That's all I'm going to say. His paper is in the EMBO journal in that year. So, other people have shown similar data today. This is the work that we did in Londowns, showing that the vast majority, 80%, I heard a very similar figure from other studies today, that 80% of people with Down syndrome are going to die of dementia. And when we say dementia, we mean, of course, Alzheimer's disease. So this is a fatal disease. Why are we not treating it when we have drugs? And then now I'm going to talk about the GWAS just briefly. When we started to do the GWAS, which, when you work on Mendelian diseases, you're carnivores. Every group who works on a Mendelian disease keeps their data to themselves, and it's a race to the finish line. And then suddenly when GWAS came along, all those carnivores had to work nicely together to pool their data sets together and do GWAS together. So we all had to switch from red in tooth and claw to vegans. It was really a shock to us all. And this is how we did, this is one of the GWAS. They get bigger and bigger and bigger. Now the biggest GWAS are all done in Amsterdam on the Amsterdam computer, but this is the result. The first thing that comes out, I think, really confused us, and this paper on the left was revealing, is that we'd expected that all of them would be APP metabolism genes. And a couple of them are. ADAM10 is alpha secretase here. ACE is also involved probably in APP metabolism. But the vast majority of these are microglial and involved in lipid metabolism. And almost certainly that reflects the fact that A-beta is metabolized mainly through the microglia. It's not going to be part of my talk, but we have some ideas as to about how that might be. This was first pointed out by Leslie Jones, and Leslie wrote this title, which I would just disagree with a little bit. She says the immune system, in the context of the brain, of course, what that means largely is the microglia, and she writes cholesterol metabolism. And in fact, the majority of genes are, there are some cholesterol metabolism genes. ABCA1 is there, for example, but the majority of them are phospholipid metabolism genes. And I won't go into it, but what I think is the reason for that is that they're recycling membranes containing A-beta stubs. So that's what I think this is pointing at. Oops, sorry, wrong button. Now we've been a part of these big clinical GWAS. I personally have not been a fan of these clinical GWAS, and even less of a fan of the GWAS where we use UK Biobank and all this sort of stuff, because as the sample numbers go up, the sample quality inevitably goes down. And even in the clinical GWAS from the Alzheimer's centres in the UK and the US, the clinical accuracy of diagnosis has been historically around about 70%. So even if you went, to one of the clinical centres in the States, let's say WashU to piss people off in the audience, your accuracy of diagnosis in those days, it's better now, your accuracy of diagnosis was around about 70%. The same at Queen Square, by the way. I'm not picking only on WashU, but it was about 70%. So we decided, and this was Valentina Escott-Price and I, we decided to do GWAS based entirely on path-confirmed samples. And of course, the trouble with that is only a proportion of the numbers are much smaller. In the last slide I showed, the numbers were something like 80,000. The numbers are far bigger than that now. But in the path-confirmed GWAS, there were only about 2,000. So what we decided to do was we decided to supplement the path-confirmed GWAS by doing GWAS of amyloid positivity in the ADNI and actually later also in the ABLE Australian cohort. My feeling about this was we were adding what was clearly path-confirmed Alzheimer's disease to path-confirmed Alzheimer's disease, but it was PET-confirmed, not path-confirmed. And actually what we found was different, and that is that the genetic risk for amyloid positivity was separable from the genetic risk of Alzheimer's disease. And what the result really showed us was that the ApoE locus was almost entirely responsible for amyloid deposition, and that dementia in that context depended upon the rest of the GWAS hits, which are largely microglial. So if you like, we've separated Alzheimer's pathogenesis into two stages. Amyloid deposition, nearly entirely in the general population, ApoE dependent, and dementia in that context, which depends upon the microglial response. Now, let's move on to how I started. What we hear is that ARIA is a problem. I'll just say what ARIA is, just for those maybe half a dozen people in the audience who don't know. What happens is when you give an amyloid antibody, the first thing it hits is that amyloid angiopathy. And it hits that at full dose concentration because that's not behind the blood-brain barrier. And so you get an inflammatory response, and that is the ARIA. What happens in that is that response is usually after the first couple of doses. Because after the first couple of doses, a lot of that amyloid angiopathy has been removed, and so you don't have that. It's not really a side effect. It's a main target effect, but you don't get that unpleasant reaction. So ARIA is usually after the first 2 or 3 amyloid inoculations. But this is the first data, and it is from WashU. I think it's from WashU. Yeah, it is. I can see half the audience nodding, so it must be. So this is their conclusion, though. A single specialty memory clinic initiated lecanemab treatment. The frequency of significant events, including ARIA, was manageable. These results may inform discussions about the risks of anti-amyloid treatment. I think this is really an important paper. Really an important paper because, you know, everyone reads clinical trials with an air of suspicion. And this is just a clinical report of how it's working in somebody's real clinic. And I think it's a very important paper for that reason. And the conclusions are really important too. It is manageable. It is manageable with the resources of WashU and the resources of, I've seen similar data from Kentucky and the Mayo Clinic too. So, but these are large, well-run operations. Now, again, this has been discussed earlier. Now, I've just said that, you know, what's the problem is that the amyloid hits the blood vessels. But we know in Down syndrome when the amyloid deposits. Here is, and again, other people have presented similar data. Andre gave me this slide today from our old Lundown, because we were working together on Lundowns. And here is the slide of amyloid positivity. It starts typically in the 30s, and then builds up, and dementia comes in, and I think I heard someone say 59 later. And then this is similar data on a slightly larger set. And dementia, again, a very high proportion get dementia. In this slide, it looks to be pretty close to 100% at the age of about 73. So really, we know what's going to happen to our Downs individuals. Now I'll talk about the history of amyloid immunization, which has never been properly told, and now it's going to be slightly improperly told. And that is that this is the key paper from Dale Schenk and Peter Subert, showing that immunization with amyloid attenuated amyloid pathology. Why was this experiment done? The experiment was done because this was from Athena Neurosciences. Pat McGeer and Joe Rogers were two people who believed that the immune response might be key in the connection between the plaque formation and tangle formation. So what they were actually doing when they did this experiment was they were pumping up the immune response, hoping to get the mice to develop tangles. So they did the experiment. Joe Rogers had been to Athena, and Pat McGeer had both been to Athena about 6 months before this work was started. And they both said, you know, what you need to do is pump up the immune response. So they pumped up the immune response, thinking that they might be able to force these mice to get tangles as well as plaques. That's not how this story is told, because of course what they did is what they actually did was prevented plaque formation. this is where I think certainly me, but the field made a mistake. And that is, we did not really distinguish, at least speaking for myself, I did not distinguish. I saw that these treatments resulted in the mice who'd had the treatment having no plaques and the mice who hadn't had the treatment having a lot of plaques. So I did not really distinguish between the antibodies causing plaque removal and antibodies preventing plaque deposition. And that has turned out to be a key problem in the, if you like, the history of amyloid antibodies. We had not distinguished between preventing plaque formation, which is what this did, and causing plaque removal. And that turned out to be very important. Now I've said this about ARIA. So ARIA is caused by the antibody hitting amyloid, the amyloid angiopathy. The reason in some jurisdictions that the antibodies are forbidden, so to speak, for ApoE4 homozygotes is because they generally have more congophilic angiopathy and they statistically have more problems with ARIA. Of course, this is an overlap, but they statistically have more problems. And that underpins the decision in the UK not to allow the treatment of E4 homozygotes. And it, of course, also adds to the costs because you have to ApoE genotype everybody, and you have to do it quickly. You have to have a real flow through so that the genotype, someone comes into clinic and wants to be treated, you don't want to say, well, we'll give you the genetics results in 3 months. You have to, it has to be organized, so it's really not as simple as you might think. Anyway, it can be dangerous in the context of microhemorrhages and anticoagulants. There's a group of people there's many groups of people of this, which this is true. There's a group of people I find particularly annoying. And the people I find particularly annoying are the people who've never believed in amyloid, but now they say, well, amyloid might have something to do with Alzheimer's disease, but it's too dangerous for us to use as a target. So they've shifted their argument. They used to say it was nothing to do with the disease, and now they say, well, it might be something to do with the disease, but don't use it as a target because it leads to ARIA. And then they publish a couple of dramatic cases where it's been given to somebody who's had TPA, and the combination has killed them. So I think that this is almost a political. This has turned into, like so many things in life, it's gone away from being a scientific discussion to simply those who are for amyloid therapies and those who are against it for any reason they can dredge up. That's my rather sour take on it. But what about Down syndrome? Well, I think that the way in Down syndrome is to go for early treatment. We know when the amyloid deposition starts. We know that the ARIA is caused by the amyloid deposition. You don't almost never get ARIA in the context of no amyloid deposition. It's almost entirely amyloid deposition in the blood vessel dependent. So in Down syndrome, we kind of know when that starts. So we can start treatment at exactly the right time. So these are some of the problems. Large diagnostic error rate has been a real problem. In Downs, we don't need that. We know that if somebody starts to go cognitively off in Down syndrome, and almost certainly if they develop myoclonus, they are starting, that's a side product of the amyloid deposition. So we know that we don't need the large diagnostic relate. So a large proportion of the cost of antibody treatment is all the things which maybe we can avoid with Down syndrome. We don't need to be quite as worked up about getting the diagnosis right because it's there, and we don't need, if we go early, to be, perhaps, this obviously has to be looked at, as worried about ARIA. I tried to, if you like, sell to, I think it was to, well, actually to both ISI and Lilly, that they organized rather short trials, this is before approvals, short trials in Down syndrome where they only used tau P17, phospho-tau P17 and neurofilament as outcome markers. And they started to see, and if they didn't get a signal with those, I thought that they could perhaps do trials rather quickly rather than spending an enormous amount on cognitive testing and so on and so forth with all the problems. Anyway, I tried to sell it to both of them, but they didn't, well, I say buy, it wouldn't have helped me, but they didn't buy it. Anyway, this is a paper which anybody who's interested in the amyloid therapies should read. And the reason I like this paper, I think it's a beautiful piece of work, is because, you know, when a trial fails or works, when a trial fails, let's say a trial fails, you go to an Alzheimer Congress the next week and everybody says, I knew that trial would fail. Everybody is so smart. In the States you call it Monday morning quarterbacking. We don't have a similar expression in England, but I think it's a great expression. But the reason I like this is because it actually predicts the future. What Eric, Karen, and Bart said was that for a trial to be successful, the amyloid had to be sucked out of the brain. That's here. And then the trial had to run on long enough for you to get clinical separation between the placebo and the drug. That was their prediction. And the prediction was right, and this is the real data. This is real data. This is donanemab. This is lecanemab. Both of these, of course, are approved. This is the first aducanumab trial, and I've separated the two aducanumab trials because this one gave a statistical signal and this one did not give a statistical signal. So by separating them, I don't know if many people remember about the controversy about aducanumab. The controversy was that really to get clinical approval, you need 2 successful trials. And actually, Biogen only had 1 successful trial, and the other trial did not reach statistical significance. But when we look at the amyloid removal data, that fits completely with what you're seeing here. And then here you have poor old gantenerumab, which reported at the same time. They'd run the trial out to nearly 30 months, and they did not get statistical. They did not get statistical, a big enough statistical signal. In fact, every marker went in the right direction, but the p-values were nowhere close to significant enough to convince, so they dropped the program. But I mention it again, I mention it here for another reason, in that this drug has now been adapted to the drug trontinemab, which has the transferrin shuttle to take the active part of the antibody straight across the blood-brain barrier. And so this is having a Lazarus-like rebirth in a different format to try and avoid ARIA. So this is the removal by lecanemab, and I am not showing donanemab. It's not favoritism, it's just that it's the same. You see clear removal of amyloid, and here's the placebo, and then here is the clinical data. And 2 things. The first, the good news. It works. You've got clear separation. The second thing, though, is the thing which I think is going to really be part of our research projects for the next period, is that you're not stopping the disease. You're getting almost complete removal of amyloid, but you're not stopping disease. Why not? And that's a very important question for the next period. Why are we not stopping it? Is it that, for example, the tau pathology has become self-sustaining? Just like if you take a match out of a fire, it's too late. You know, is the tau pathology, or is the microglial just exhausted, and is the microglial response getting worse and worse as you get through? We don't know, and one of the things that is going to be very important, and it's going to be very important in Down syndrome as well. And I'm sorry I missed the pathology talk this morning. That was because I got up at 3:30 to come here, and I still hadn't got here for that talk. So my apologies. I'll look at it online if I can. Anyway, we need to know what the underlying pathology is of all the people who've had treatments. We need to understand it. And what we need to know is to what extent are the pathologies that we see in the brains after treatment, are they dependent upon amyloid? Are they independent of amyloid? Or probably most likely, are they semi-independent of amyloid? We need to know what the tangle pathology is. We're beginning to see some of that data in the PET scanning, but we need to know the TDP and the microglial pathologies too. We really need good autopsy follow-up on all of these patients. So this is trontinemab, and the reason I say this is because it may be a route to avoiding ARIA. As I say, it's gantenerumab tied to the transferrin shuttle, and it causes really rapid amyloid removal, and it goes past the blood-brain barrier. And so there seems to be much less of an effect, much less of a danger, much less evidence for ARIA. We don't know yet, of course. We've been fooled by optimistic phase 1 and phase 2 trials before, but this looks good. Let's keep our fingers crossed. So, what is the cause of the residual decline? We need more neuropathology. Are the pathologies dependent or independent? Will earlier treatment avoid or reduce ARIA? I think they will, personally, but that's a thought, not a statement. And will they then have better clinical outcomes? Can we get better clinical antibody outcomes if we treat earlier? I think we can. It's really difficult of course, the DIAN study gives real opportunities, as we're hearing, or to get earlier. And now we just hope that Donald decides that they can go on. And you know, well, I think it'd be a tragedy if they don't. And you know, we need to be really pushing to continue these types. It's much more difficult to predict in the general population who's going to get sick. Genetics gives you an area under the curve of about 75 to 80%. Biomarkers help, and we're going to, obviously we've heard quite a bit about biomarkers. Biomarkers are very good at separating cases from controls, but what we don't yet know is how early in the disease staging they separate, and how well they separate, for example, dementia with Lewy bodies from Alzheimer's disease. We need all of that data as well. So we've got a long way to go, but you know, my overall feeling for the general field is that this is like the Wright brothers on that North Carolina beach in, I think it was 1908. Who would have believed that a bicycle with wings would lead in the 15 years to real powered flight and, you know, actually payment of flights between England and Paris. So, you know, it's once you have something that works, it's much easier to make it work better than it is to make it work in the first case. So I think we're in a really good position. in the general population, and I think it's our duty to push for the same benefits for the Downs community. Thanks very much.
So thank you for the very inspiring talk, John. So speaking next is always complicated. So, but we knew it. Confronting with having worse. So we didn't do a controversy here. So no, here the symposium I'm going to try to run, because I think that what's really interesting is that we're in a very unique forum, and when we were designing this conference, we wanted to have this cross-fertilization, and we wanted in this symposium to discuss, and with all of you, the possibility of expanding the concept of genetically determined AD. We've seen several talks of AD-AD, and still these talks talk only of presenting 1, presenting 2 APP. Mutations, and this is how the field works, and I'm gonna put some arguments that you already know and reflections to see to propose that maybe we can expand this concept. No? This is my disclosures. I consult for several companies, and for those of you into art, maybe you know at this René Magritte, Ceci n'est pas un pipe, and well, he said, try putting tobacco into it, no? So it isn't a pip. But it certainly looks like one, and I would like to throw some arguments, and also I would emphasize from the starting point some rules on how we evaluate things. I think we need some rules, and if we play soccer, we play soccer, we play rugby, we play rugby, but we cannot change the arguments while we are speaking. Because the first thing is what is genetically determined AD, but maybe what is AD? And this seems obvious, but I think that if you look into the evolution of the AD criteria, we haven't reached consensus. We haven't reached consensus. It is two papers, the 2 iterations, and you would say that there's a huge difference, whether you're using the International Working Group criteria or the Alzheimer's Association criteria. And if you include or not include preclinical AD, the penetrance of anything is going to be hugely different. So how are we evaluate the penetrance of any disease? We first need to decide whether we're going to use one criteria or the other. We cannot jump from one to the other. I think that we need rules on how to evaluate those things, and the implications are enormous, enormous. And of course, I'm going to stick to the Alzheimer's Association criteria, not because they support the conference, because I think they are more forward-looking with respect to treatments, but I'm going to give some consideration about the International Working Group also. So first of all, I like to give thanks to all the authors, but especially here Heather, Eric, and Mike, because one of the things that I love of this paper, of course, and in this conference we should thank them, is that the concept of stage 0 that clearly equates for the first time in some criteria DSAD and autosomal dominant AD. So by definition, if you have these point mutations or you have Down syndrome, you're considered to be in a stage 0, regardless of whether you have not only symptoms, but in this case, not even the biology. The genetics is sufficient. And this, I used to, because this is how we understand the genetic architecture of Alzheimer's disease, and I always want to say that here we have the axis broken in the y-axis, that the one talking about whether we're talking about risk or causality in the y-axis, and the x-axis when we talk about frequency. And in the upper right corner, we have this fortress in Alzheimer's disease, and this is what we consider genetically determined AD. And I always argue, that is of course very rare, that this is just neglecting Down syndrome. I think that most people that do not include Down syndrome is just out of neglect because they are not considering them in the research or clinical practice. And of course, you've also heard about this proposal that we made that APOE4 homozygotes should be included. So for the first part, I'm going to try to see, and I'm happy to, because this proposal for APOE4 homozygotes came from the work we've done in Down syndrome. And trying to define a set of rules on how we should define genetically determined AD, we came with the idea that of the main characteristics when you think of a Mendelian disease or genetically determined Alzheimer's disease is that it should have near full penetrance. That it should, we'll discuss a nuance a bit assessment, the statement, but you should have near full penetrance, a predictability in the age of symptom onset, and a predictable sequence of biomarker changes. And so we're going to take AD-AD, and we're not even going to explain anything about it. It's just going to be the model, and we're going to go over the data in DSAD very quickly because a lot of data has been presented, and APOE4 homozygotes. So this has been shown, near full penetrance, the ultra high risk. So I think there is no doubt now, and I think it's well accepted, it's even in the criteria. People with Down syndrome, as John has nicely said, die of Alzheimer's disease. So this is killing over 70–80% of individuals with Down syndrome. So I think that is by far the main killer of people with Alzheimer's disease. So I think that this is now luckily well accepted. In the last 5–7 years, if you go to 2010, you still have complete different numbers. I think that if you need to follow the participants long enough, and this is over 18 years in a nice work from Ireland, and that when you really once you get away from the survival bias, you really get these estimates that are now generally accepted, and you've seen in this conference that how consistently report the penetrance. The other really nice thing is that there was this concept in Down syndrome, and this was even, you don't even need to go 10 years ago, every paper started how variable AD was in Down syndrome. And that came from a clinical bias, the perception that you had seen someone 43 with dementia, and you had seen a 50-year-old or 58-year-old without dementia. And so all papers said this is highly variable. 10 years ago, highly variable. Whether it is Alzheimer's or not Alzheimer's, it was even debatable. So we performed this meta-analysis to see the age at dementia onset in all the studies in the US, Europe, and elsewhere. In the last 40 years, no harmonization whatsoever. So they diagnosed the dementia the best they could. And you can see how conserved this disease, the disease duration, the age at death of 58. And I think it's really nice that now to Eric with the Medicaid data, I mean, with the tens of thousands of individuals, the data is almost mathematical. And this is something quite amazing. But because I did my PhD in autosomal dominant AD, and when we were doing the natural history in Down syndrome, the numbers didn't seem that different between autosomal dominant AD and Down syndrome. And luckily, we had done the meta-analysis in red for all the studies in Down syndrome, and there was another available biomarker by Ryan from London that analyzed the variability in autosomal dominant AD. And here there are different ways in analyzing the variability around symptom onset in these populations. And I think that the main message here is reds are the same as blues. There is the same variability or predictability. I mean, you can say they're variable, you can focus on the predictability, but they are the same in Down syndrome and in autosomal dominant disease. And this, to me, is quite remarkable. This has implications into models, but I'm not going to enter into that. We've also mentioned it, that the natural history in Down syndrome for the clinical biomarker changes is well established. And when you plot the models, it's quite remarkable. The similarities in the biomarker changes in a point mutation and in Down syndrome. And I think this is something that, I mean, of course, there might be subtle differences. We're doing proteomic analysis, and we're also finding differences that can give fantastic clues into the disease pathophysiology, but broadly, all the core AD biomarkers, they follow very, very similar trajectories, and I think this is amazing. So having work in that and having this perspective and looking into APOE4 homozygotes through this lens, we came to the conclusion that maybe in APOE4 we have been doing something out of practicalities. The problem with APOE4 homozygotes is that they are relatively rare. And when you were doing clinical studies, even if you have a fairly large cohort of 300 or whatever, you don't have that many APOE4 homozygotes. So most studies, I'd say, over 99, maybe, lump together APOE4 homozygotes and APOE4 heterozygotes because you don't have a statistical power, and then either you exclude them or you lump them together in APOE4 carriers. So here, and I say that's why I'm talking about rules, in this study we said, okay, if Alzheimer's disease is defined biological, let's look at the biology. And so we did like this was published last year, and actually we presented the results for the first time in this conference. And what we said, using the NAC data and the pathological data, that if you're APOE4 homozygote, of course this sample is biased towards symptomatic participants, you're bound to have AD pathology. But when you look into the AD biomarkers in life, and this is samples, clinical samples that are biased to cognitively unimpaired participants. By far, the largest cohort was the A4 trial, which put here over 4,000 individuals. To enter that trial, you had to be cognitively unimpaired by definition, because it was a secondary prevention trial. So in that sample biased towards cognitively unimpaired, you could also see a near full penetrance of AD biology as assessed by biomarkers. I'm not saying this is a population-based samples, I'm just saying that we have two separate bias and both cohorts converge into an incredible penetrance of AD biology as defined by pathology or AD biomarkers. But what was similarly striking, this was known that the APOE had a stepwise a gene dose on having more risk and on, of course, earlier onset. But what was interesting is what we started to analyzing the predictability of the disease. When we went to the NAC database and say, okay, those that develop AD dementia by genotype, by haplotype, by APOE haplotype, how variable is this disease? And what was really interesting is to see that we also see a gene dose such as that the bars were longer for APOE3 homozygotes, shorter for APOE4 homozygotes. And what we'd like to see is that these bars are as long or as the others. So this is another way to plot it. This is the prediction interval for the age of onset of dementia. And you can see that remarkably, the prediction intervals for DSAD, presenilin 1, the PISA mutation, and APOE4 homozygote is the same. Again, it's as predictable or as variable in APOE4 homozygotes and is this as in these other conditions. And it's nicely, you have a gene dose effect such as the less APOE4, the more variable the clinical presentation that you have. So I think that was a really remarkable finding. The natural history, again, we are lumping together the yellows with the reds. I mean, of course, the reds are homozygotes, heterozygotes, non-carriers. And I mean, it makes the same sense to put the yellows with the greens or with the reds, are just in the middle. But certainly, if we focus on the 4-4 versus the 3-3, and we plot the standardized differences in the same way that the WashU, Randy Bateman did in the 2012 paper in New England, again, we find a really remarkable similar sequence of events in APOE4 homozygotes. And I think that's really interesting. So I think that if we define the disease biologically, I think that we can conclude that we have near full penetrance, the same predictability, and a predictable sequence of biomarker changes. And that's why we propose that APOE4 homozygotes should be included in the family of genetically determined AD. But what about those that are more comfortable with international working group criteria and define the disease clinico-biologically? So I think that's also interesting, and the conversation gets even more interested, because and this didn't come from our work, but there are data there to suggest that the risk for clinical dementia, if you are APOE4 homozygotes in the by 80 or 85, is 40 to 60%. This is Wikipedia. This is these two mutations that are considered Mendelian. Women have these mutations and they take out their breasts. And these are the risks for breast cancers, for pancreas, ovarian cancer. So again, is this exceptionalism? As John was saying, if this was cancer, he said, we would not allow not to be treated. However, we have a completely different set of rules to consider something Mendelian in Alzheimer's disease as opposed to other medical conditions. I would argue, why dementia or not, this does not include MCI, which by definition would include the penetrance. And of course, if we include preclinical, that we discussed, it's even more. So, but even if in the most restrictive of the definitions, dementia, we're still at the same level of risk that we have here. And I think that's something remarkable. But something that's really interesting, and I'm not going to defend the methodology, I'm not a statistician, and certainly not a geneticist, but this is incredible, because Alzheimer's needs to be fully penetrant by definition, 100%, otherwise you're not Mendelian. But then you take all this paper that was found really interesting, and that estimated the penetrance, and I know this, we can discuss whether these estimates are accurate or not, but they estimated that the penetrance in the C9 expansion, for example, which no one discusses is a Mendelian risk gene for ALS or frontotemporal dementia, the risk in this paper was estimated to be 33%. You can say, no, it can be higher. What, 40, 60? Why C9 expansion are a gene for frontotemporal dementia and ALS, and this risk is not in Alzheimer's disease? That's why I emphasize the need for rules and clarify that we should evaluate by the same criteria. But this is something that is really it becomes interesting, because one of the things when you analyze rare mutations is that you really need to have large sequencing in the population, because otherwise you might be biased towards the penetrance in symptomatic participants, small cohorts, and so on. And this paper that was recently published in Nature Medicine found that the carrier frequency of repeat expansions was 10 times higher than expected. That was to be expected if you consider like full penetrance for these diseases. Of course, you still can claim that there is underdiagnosis, and that contributed, but they've clearly said that probably there is much more incomplete clinical penetrance for many of these diseases than previously thought, and that would equate many of the diseases that in neurology that we consider are Mendelian to the case of Alzheimer's disease. So, as I said, this would have dramatic consequences, because basically we would be saying that 15 to 20% of AD dementia cases would have a genetic cause, not genetic risk, and certainly 2-3% of the population would have a sort of presenilin-1 mutation, clearly not with full penetrance, but with this 40-60% for dementia, probably including MCI, maybe it's 2/3 or 3/4. And this is, I think this was really nice, because John mentioned it. And if you take, I've taken here mutations in other genes. So I'm shifting gears. I'm coming back to open a bit the umbrella of those mutations that have been associated with really high odds ratio of developing symptomatic disease, and many have been mentioned by John already. And I think that what's really interesting is that they all converge in A-beta pathways or impaired A-beta clearance. Of course, some of these genes can be associated to other pathways, but there is a clear convergence that anything that confers you an ultra-high risk of developing AD dementia or symptomatic AD, they converge in this amyloid pathway. And I think this is really remarkable and worth discussing. So this is Magritte. This is ChatGPT Magritte. And I hope that here is the space to have this discussion and hopefully reach an agreement on how to define genetically determined AD. And with that, I'll finish. I completely agree with John. Not only should we treat these people with Down syndrome and all these genetic forms, we owe them to them, but it could also be the best way to prevent this disease. We are in this fantastic moment in which we are having therapies that that have the potential to delay, if not prevent, the disease in these genetic conditions. And with that, I'll thank all of you for being here and the unit. Thank you very much.
I think a lot of great points, and I would maybe throw one out to consider. You argued, why are we waiting to give them drugs?
but why even wait to apply the current standards? So you mentioned the match already started, and should we view it more as a fire break, the same way as for cancer, to say, should we be treating people aggressively in stage 0? And is that where we really need to be going, not kind of this incremental to match the current field and then wait to shift earlier and earlier?
Well, let me just put a little public health warning, and that is I'm not a clinician, so it's very easy for me to tell other people what to do. But yes, I think we should. I think we should be giving the therapies we have now. One thing that occurs to me, and it occurred to me as Juan was talking, is maybe this group should write to the regulatory authorities and say, why are we forgetting these people in our midst? Well, I think that's one thing that might come out of here. I don't know what Juan thinks. He's the boss, but yeah, I do think we should be doing this. Yes. I'd love to hear what other people think who are clinicians.
So we're gonna have a whole session on trials in autosomal dominant AD and DSAD with, I think, the two world leaders in this. So Eric McDade and Mike Raffy will have a full session such as this to discuss that. It's been raised. I completely agree. I think that I said it. We've been saying it. All of us, I think, in this room. This is the perfect population to do the preventative trial. There's if you have a drug that is likely to work better the earlier you give it, you have a condition that is telling you, I'm going to develop it. Waiting for symptoms is nonsense. Even if it's nonsense. I always say that if you had the magic pill, Star Trek, that stopped the disease, why would you wait to have dementia? Okay, now you're going to stop at dementia. I prefer to stop when I'm cognitively unimpaired. So it makes absolute no sense. But as I said, I would emphasize that since we have a specific session on treatments, and Eric McDade is running the biggest trial in even primary prevention in autosomal dominant AD, and we have Mike who is spearheading all these trials in DS, I suggest that maybe we can wait for that session to discuss the treatments more at depth. But what about this? How big can the family get? John.
Well, I was pleased you showed, because I thought about it and didn't, at the ABCA7, SOL1, actually ABCA1 nonsense mutations. There's a lot of these rare variants. There's a lot of them, and because they're rare, they're kind of lost. Each one of these family, each one of them has been found in maybe half a dozen to a dozen, and yet they are, they're outside because they can't be in Diane, and that's a shame that they're missing from the table. And for example, in African Americans, there's an ABCA7 deletion, which is very common and has a very high risk. It's not autosomal dominant, but they are doubly missing from this group. It's a shame, and we need to change that.
So, Juan, I think that the argument that you've made for APOE4 homozygotes to be considered a genetic form of AD is very strong. At the same time, what a shame it is if a woman has a bilateral mastectomy when she only has a 40% chance of having cancer. And that's a personal choice, whether she goes through with it or not. Maybe 40% is not a threshold she's willing to consider. Maybe 1% isn't enough. This personal choice is determined not only by their own cultural values, but by really an informed risk assessment. And for APP mutations and trisomy 21 having extra APP leading to AD, that's 100%. That's very clear. But here, you're seeing 40 to 60% risk. And that, maybe what we should be doing is arguing that the BRCA1 and 2 should not be sold as a guarantee, as a being condemned to a life of having cancer. Maybe we need to do better education so that people don't feel like they're harboring a 100% penetrant disease.
Yeah, no, I completely agree with that. And certainly, and it's great because I think this proposal has been highly controversial to the very least. So I think that the controversy should emerge in this forum. So because to advance, we should not. So I really appreciate the comment. The point I make is the same with variability of predictability in Down syndrome or autosomal dominant AD. What I said is what we cannot say is that autosomal dominant AD is predictable and Down syndrome is variable when they have the same predictability and the same variability. The point I wanted to make is that, okay, we need rules. That's why I said that as I said at the beginning. So BRCA1 can be is a Mendelian gene or not. What it cannot be is that is a Mendelian gene and APOE4 homozygotes is a risk gene. And it goes for all the other genes in dementia. What I'm saying is that the exception is in Alzheimer's. So it's the only disease in which you have to be fully penetrant to be considered Mendelian. I think you go to genetic books, and I had to go to the genetic books. I'm not a geneticist. There are many geneticists in this room that know much more about genetics than I'm not claiming. But you go there, and you have lines like that saying that incomplete penetrance is the rule. It's not an exception not to have complete penetrance. So again, it's a question of rules. So how are we going to define Mendelian diseases? And I think that it goes the same with whether you are using the International Working Group and Alzheimer's Association. What you cannot use is the Alzheimer's Association Mondays, Wednesdays, and Fridays. But because you don't want to confer this risk to people, which I understand, that's a human, I mean, that's empathy, but you can just completely change your rules on Tuesdays and Thursdays, and then you consider a clinico-biological definition. So we're using the definition biological or clinico-biological, and we use the kind of the same set of rules to make the discussion, because otherwise I have this feeling that we're kind of dancing with arguments. We're discussing, one thing is how we convey, how we assess risk, but the other thing is the biology, and saying that if we have rules and we apply them to all the diseases, to all the conditions, it's much, we will make more informed progress. And I completely agree with all the concerns that have been nicely written in several articles that were, because this was a controversy, but I agree with that.
So let me put it this way. Think about a trinucleotide repeat disorder. So Huntington's disease, that was mentioned earlier. We can assess risk by the length of those repeats. If you have a certain amount, your risk is higher. That is a genetic basis for risk prediction. We don't have that for cancer, and we don't have that for many of the other neurodegenerative diseases. Once we have that gene dose effect, if you will, then we might be more able to apply the same rules across all of these. Until then, you know, whether it's methylation or post-translational modification or something we can measure, we're not really going to be there, and we won't be able to apply the same rules.
But let me put it the other way. You can tell a 20-year-old that is APOE4 homozygotes that by age 6, that he or she will develop Alzheimer's disease by age 65, symptoms of Alzheimer's disease with 2/3 of probability. This is not 100%, but that's if you put the rule at 100%. That is what I'm discussing. I don't think the rule should be 100%. And if we agree that that's the rule, that's fine, but then we apply it to all diseases. I don't want an exception for Alzheimer's disease. We have to have the same for all diseases. And that's a lot of information. And let me put it the other way. If we followed John's advice and we applied lecanemab earlier now or donanemab earlier, and we could prevent the disease, who would wait and who would discuss not to present? So it's like mastectomy. It's aggressive, but it's preventing. So in the example that you did, I mean, you're taking your risk because, okay, it's very aggressive, but you're saving a 40%, 50% chance of cancer. So, of course, we don't have that in Alzheimer's. But once I think we envision a future where we can have those treatments. And in that case, probably the conception changes. But from an epistemological point of view, the definition of the disease should not be, I believe, contingent on the availability of treatment. So some people say, no, we cannot consider APOE4 because we don't have a treatment. No, I understand the argument in favor of the person and the impact of the diagnosis, but the definition of the nosological entity is not dependent on the availability of a treatment. That, from a logical perspective, that's my approach.
Agreed. And it's not just availability, it's cost of treatment.
Yeah, okay, yeah.
But for your Huntington's example, sure, the person with more repeats is going to get the symptoms faster, but you would never deny someone with less repeats the drug just because that's farther off in time. That person still has the disease. They're still going to get it with a very high probability, even if it's delayed. So I don't know if that's a perfect analogy for this.
And we should also acknowledge that the cancer field is moving. I mean, they are doing their way back, more close to the dementia field. I mean, the first genes that predispose to cause cancer were described in families. So they thought the penetrance was almost 100 or very high. And now with this panel, they are studying many sporadic breast cancer. They are discovering that the penetrance of these genes are not so high. And I mean, the field is also moving. So they don't execute 100% penetrance, but they are also thinking of changing the rules of treatment because of that. So I think they are not so far away. I mean, yes, I think most of these diseases are not 100% penetrance.
I fully agree with your argument, both arguments actually, from John and from Juan, that we should not consider them separate from other diseases, which, if I state from the Netherlands, I don't know how many people are here, I think we have a very strong argument. It's an internal argument with the, that you call it, armchair neurologist, who's a bit older usually. John is maybe an exception. You're not a neurologist and not a geriatrician. That has to, and also not so old. Age is just a number. That they have been looking at patients and just looking at describing patients. So that's basically the argument in the Netherlands. It's a cost argument, and that we cannot do this all at once for this large group. Then considering, so that's the Netherlands. So it's a coin flip if we will start treating the patients within the Netherlands. I really do not know. It will, however, matter for the consideration of risk. And on that note, I do have an epidemiological consideration to your argument that APOE4/4 is a monogenic, you could call it monogenic disease. That would be a better term than Mendelian, because Mendelian Mendel was able to show that traits segregate with certain variants, and with APOE it does not, because it comes from two sides, from your mother and your father, and then you get a certain risk. You call that additive risk, and I think that is the case within Alzheimer's disease. If you start to model it, Alzheimer's disease around age 95 is like where people should all have Alzheimer's disease given the baseline risk. And APOE is just a modifier of that, like your average, and I call it Alzheimer's disease dementia, I'm meaning. It's just a modifier of that. And all the other variants that are out there are actually modifiers, smaller, are modifiers of that. TREM2 is a big one, and there was a paper in New England where you show if you have two TREM2 variants, it's even much more risk than the APOE4/4. It's like it's non-additive. So I'm against putting APOE4/4 as a separate entity, because it's in a scale of where all the genetic risk factors modify the age at onset, and you can very well be with APOE, with just one APOE, a high polygenic score, and maybe a small SORL1 variant and an ABCA7. Like, when do we stop calling it Mendelian? This is not Mendelian, that's a complex disease. So I'm disagreeing with calling APOE4/4 a separate, let's call it correct, monogenic disease. It is a very high risk factor that can still be modified by other genetic factors. And I'm going to give the example of Henne Holsteger's favorite centenarian. She is a centenarian researcher. And our 104 centenarian was included in our study, and she carries an ApoE44. A PhD student was waiting for her to die to actually study her brain. That took her whole PhD because she died at the age of 108. And she got dementia at 107. So likely this patient, but let's call it a patient now because she died with dementia. I think that's unfair. But she both postponed and then delayed the risk of the effect of tau, and proving on her own, this is not a Mendelian disease, it's not a monogenic disease, but a complex disease with ApoE44 being a very strong risk factor that we should definitely take into account, but not forget everything else, because then it's a complex disease.
Okay, so no, I can disagree with myself and close to you. And first I'll start by saying that the term that we liked was, we used is autosomal semi-dominant. That's the one we wanted to use. And that wasn't us that coined it. The first time that was used, it was a nice paper in Molecular Psychiatry in 2011 that they used autosomal semi-dominant Alzheimer's disease. It was based mainly on Ajiwas. It was, and that was they used the term semi-dominant. We presented the data that way. We were crushed because, first of all, you had to define what semi-dominant, and it's a very rare term. It's really interesting, by the way. It's semi-dominant when you investigate a bit more. Is in apolipoproteins, and for instance, you have to talk to endocrinologists, and the risk is this stepwise and everything. So I'd say that yes, I agree with you. I think that the isolation of ApoE4 is just to change the landscape. If you go to the paper and you read a bit more carefully, we focus on the ApoE4 homozygotes, but in the supplementary material we have the heterozygotes, and in the discussion we enter a bit into the semi-dominance, which is a concept that is more complicated to introduce. But for battle reasons, for military reasons, we simplified a bit the concept because it's controversial enough to try to convey these main ideas that are important. Secondly, I would say that I agree with you that this idea, but our point is not to put ApoE4 homozygotes in the castle of presenilin 1, but to break the walls of the castle of presenilin 1s and take them out, which is a completely different approach. Because I would argue that you can also modify the effect of presenilin 1 with other mutations. So that the same argument that you gave for ApoE4 homozygotes, which I completely agree, you could give it to the ApoE4 homozygote. So then you are talking about a dimension probably in the clinical penetrance and how modifiable it is the disease, and it becomes more complex. And maybe the Parkinsonians, the people from Parkinson, sorry, working on Parkinson, the Parkinsonians, that they have a more nuanced approach in that they are kind of putting kind of some gray zones and in the genetic risk in Parkinson's.
Because they have more ApoE genes, like Larkin, they have GBA, they have more ApoE genes. You could also call it bigenic. You could call it, I tried to call it bigenic, but I failed with my clinical geneticist
maybe the
colleague.
argument is that this castle for presenilin 1 is paralyzing. It has a lot of implications. All the disease models have been built because if you don't build a mouse model based on a risk factor, you build a model if you believe that it's causing a disease. So I said that 99% of papers were done with collapsing ApoE4 homozygotes and heterozygotes. That's another consequences that you're neglecting a lot of the disease. How many papers with animal models are based on APP? And presenilin 1, and would you name it? And how many animal models have been built with ApoE now? Because it's so important, there are a few, but if you put numbers out there, I'd say it's 99 versus 1%. The approaches, we have a Diane for rare mutations. We do not have that for ApoE4 homozygotes, and it's another missed opportunity. So I think that this is not, to me, a nominalistic discussion. It has a lot of very important consequences if you think about it. So I completely agree with you, but I'd say that all your arguments, which I agree to ApoE4 homozygotes, could be applied to presenilin 1 and other mutations, and then we start having a dimension, not a castle that we protect. This is a more complex and nuanced picture.
I fully agree, and I think on that part I fully agree. But we have tried with Sorla with to make that the fourth. To start somewhere, and you took the leap to go to ApoE44, but with SORLA protein truncating variants, even my colleagues, Gaël Nicolas, a well-respected colleague, still disagrees that I disclose this variant to my patient, and he would have never. So this is the argument that will change as soon as we, and you say it's different when you have a therapy, yes or no, but for clinicians, and I am one, unfortunately, I am a doctor, it does matter for especially the white coats to what extent uncertainty they're willing to tolerate when they disclose risk to patient. So that is the amount of actionability that follows afterwards, and I think the main argument why we do not call ApoE44 homozygote semi-dominant or autosomal dominant, because then clinical geneticists, the consequence they put after it is that you can do pre-conceptional testing and destroying of the embryo, which we do for presenilin-1, and I find that understandable. But if you have an ApoE34 parent and an ApoE34 parent, you have a 25% chance to have a ApoE44 child. If we find that argument of reasoning logical, so we put them in the same bin, then they say if you say A, you have to say B, C, D, and E is that we have to do preconceptional testing, and people can destroy they can test, and they can decide to terminate the pregnancy if there is, this is the consequence that the clinicians have to deal with as soon as they break the barrier between Mendelian and non-Mendelian. So this is the barrier that you need to break, and that can only be broken if there is a therapy. So this is the main argument why I not get, just like you, do not get the argument closed, because there is this big leap towards preconceptional abortion, basically.
And I agree with that, but that's why I think we need a set of rules, and we need to look beyond Alzheimer's to see what are the rules that we're going to apply, and we're going to be consistent with that. To a certain difference, all the controversy between International Working Group criteria and Alzheimer's Association, to a certain extent, revolves about the consequence of diagnosis. And so the Alzheimer's Association, they include stage one, and whereas in the International Working Group is like, as a clinician, as a white coat, the implications of calling someone with Alzheimer's. But this, again, these are very valid considerations. But we need to discuss on the rules, because otherwise, as I have this impression that we are changing. I've seen papers where they jump from International Working Group to Alzheimer's Association criteria depending on what argument they were valuing more. And that's, to me, it's difficult to follow because I don't have maybe that flexibility to change the set of criteria in the same kind. So I think that's the idea. So it's very valid arguments, but we need, precisely because it's very complex and with huge implications, we need rules. That's the point.
And we are doing performing a theoretical discussion that's very interesting, but we have the practical problem already here, because, I mean, at least in Spain, at this point, the guidelines do not recommend do not use ApoE for genetic counseling, but at the same time, we have to test ApoE for treatment, and we should provide some guidelines or some counseling to the patients with tests for ApoE. So what we should do, because it's difficult now, the knowledge is in internet or whatever, so you cannot say you are ApoE44, you are homozygous, and I cannot provide you lecanemab, at least in Europe, because you are homozygous, and you cannot say anything about genetic counseling, but because the patients will go to the internet and get some information about ApoE and genetic risk of ApoE4. So we need consensus on what should we say to our patients about ApoE. I think the problem is already here. So in the practice. So what will you recommend to say, or what should we say? Tell them.
I completely agree. I think because of these treatment recommendations, we've kind of sleepwalked into ApoE genotyping with no thought of the downstream consequences. I completely agree with that. Yeah, and I sort of feel that the regulatory authorities for the treatments have not really been genetically sophisticated, and they've not really understood what are the consequences of their treatment recommendations in terms of genetic counseling.
But yeah, I completely agree with that, but it's yet another argument of personalized medicine. If you define another reason to this is really interesting, because in a way, ApoE, one of the amazing things is the convergence of these biomarker changes in all these genetic populations. But for instance, the treatment response, or at least the risk for side effects, is completely different. There's no doubt that there is a gene dose effect based on ApoE. So if we are talking about personalized medicine, it would be impossible not to do it. I completely agree. The consequences have not been well studied, considered, and everything. But the need is obvious, because the risks are so dramatically different that there is no way, but which is a good argument to me. The science is what it is. Then there is the consequences and how we handle that. But we cannot, because of the consequences of informing of an ApoE4 haplotype, I don't think we should stop doing an ApoE4 when we know this is going to have completely different risks for the disease treatment. But shifting gears, I wanted to say, because there is this one paper very recent, I don't recall the first author, that what they did is the turnaround, because one of the things of the ApoE is that we are using as reference, obviously, 3.3, because it's the major gene. But the thing becomes extremely interesting if you put the reference at 2.2 as the lowest, because then the risk for Alzheimer's disease, it develops amazingly differently. And I think that putting the mean, we are minimizing the effects of ApoE, because some of them are under and some of them are up on 3.3. And I think that other paper was extremely thought-provoking, and it's been recently published. I really recommend it.
Thank you very much for this fantastic siesta time that you have provided us with a lot of information. As a single question, only one question for a doctor. Imagine that I have already a diagnostic of very early Alzheimer's disease. Don't imagine this for me, it's in general. But imagine that I have this, then I am in a country where it's approved, the lecanemab, for example, and you are the doctors, and you have a genetist here also. Then my question is clear. Are you going to recommend me the treatment with lecanemab if I am black? Thank you.
I guess, yeah, I mean, I think the problem with is, I think you're referring to the lack of evidence in minorities and everything, which is are very interesting also for this discussion, because I think that we haven't mentioned it, but I do completely recognize that the genetic background can influence the ApoE risks. But as I always say, even if it were only, I don't think it is the case, but if it were only in Asians and white people, it would be like a good proportion of humanity that would be genetically determined. The fact that black people might have a lower risk would not change that other fact. But to your point, I think that we're lacking data. The problem with diversity in studies and everything, I think that the data that we have in black people in those trials, if it was positive, in that you would never accept that argument as sufficient proof, because there aren't sufficient participants there. So I think that the jury is still out as to say whether these treatments in minorities that haven't been well represented in the trials are effective or not. I think that that's a big need of the field to have diversity.
Thank you for the answer. I mean, I will never accept it to be treated for something that I have no information about. Then for these 280 lecanemab in the real world, from 250 or 84, there are only 3 black people. That means that we don't know nothing about what is the effect of this drug in people different than white and beautiful people. Then this is the question for Dr. Hardy. what about reelin gene? That some mutations, alteration in reelin genes can protect the disease in this South American reelin. Reelin.
So sorry, what's the question exactly?
There is some. My question is if you can comment about the gene in reelin.
Oh, reelin.
Reelin gene.
Yeah.
Okay.
So
thank you.
yes, well, I can't really. Of course, I can't really comment in the sense that I don't know much more than is publicly available. But certainly, the DAB reelin pathway is important. Actually, it seems to be particularly important in ApoE4 homozygotes. There is an association, the DAB locus and the reelin locus. When you do a GWAS only in ApoE4 homozygotes, they come out as associated risks. So, and that we have interpreted that as being related to the fact that those individuals have very high A-beta amyloid levels. That's how we've interpreted it. But we don't know more than the papers show already. So, we need to do more on it. I will say this also, that Brian Howell had previously suggested, indirectly based upon fly work, that DAB and reelin were involved in APP processing. So there was a precedent in the literature suggesting that DAB and reelin were important, and so we've been collaborating with him, and he's published on those topics, but we don't know more really. Sorry about that ineffectual answer.
So thank you. We should close the session, close the day. Thank you for being here. I hope you have enjoyed the day as much as I have, and see you tomorrow. For those
at dinner.
At dinner, for those coming
It's an expensive dinner, please come.